from Ishbankis, Kelsey recorded for Trin's lab in the Stanford by Behrad, and me and Reza recorded data in Iran. And this is the all author, and this is our lab in Iran at IPM under supervision of Reza, that's here. And I would thank for this cognitive council in the Iran that support us in Iran. Also, Trin and Behrad have other grant from other in US. Thank you, and this is my last slide. Any question?
Okay, thank you, Eza. Yeah, so if you have questions, just post them in the Q&A window. And so we have one question here from Mohammad Amin Samipur. He asks, while your result is in favor of the synchrony view of working memory, how to reject the classic view of working memory? Could you or should you this in your results?
As the important point is that the FEF is, as we know, is a special area and code the location. But our task is the object identity coding. The main question was that for us was that why the FEF maintained this persistent activity during the delay. We are seeking to try any interaction to other sensory area and provide the evidence to shows that this activity have a role in the question in the task. But another main point that we found for response to reviewer of paper that we have positive correlation between object location selectivity and the level of synchrony across areas. And this is another sign that for this part, the FEF has a role. And also we found that the FEF phase is have positive lag to the IT, and we conclude that this signal correlationally maybe comes from FEF to IT.
Okay, thank you. Maybe one question from my side, because we also still have some time. So you showed a couple of different hypotheses or theories, yes, exactly, about how synchrony could play a role in communication. And your results, do they support all of them, or is there one that's not compatible with your results?
This is a good question, but and our question for the next project, we are going to work on that. As we know, the phase locking is a correlational measure, and we cannot rule out another hypothetical, maybe third area synchronize two areas which are each other, and another way maybe two areas, one of them make synchrony between them. We are trying to manipulate PFCs and also recording from two areas in the downstream IT and V4 and try to find which of these hypotheses maybe works. The main point that here we have is that this interaction has a role in the IT's selectivity and IT's role in the object maintenance. And this is the main point that our presentation right now, but that question comes from after our manipulation study, and that we are going to work on that right now.
Okay, thank you again, Ehsan. So we come to our next speaker now. Our second talk will be by Simona Olmi. She currently holds a starting research position in the Math Neuro team at INRIA Sophia Antipolis Mediterranean Research Center in France. And she's going to talk about an exact neural mass model for synaptic-based working memory. So Simona, if you share your screen, great.
Okay, do you hear me? Is everything okay?
Yes, everything
okay,
works, yeah.
Perfect. No, because, okay, let's, good morning, and in this talk I will try to present you a new exact neural mass models that we have developed for synaptic-based working memory. As I think all of you know, working memory describes the capability to memorize and access and manipulate information that are obtained through stimulation, even though the stimulus is not anymore present. So in this framework, we tried to develop a new neural mass model that is able to reproduce exactly the dynamics of heterogeneous Python neural network that are encompassing realistic cellular mechanisms for short-term plasticity. So in particular, in the rest of the talk, I will try to touch the following topics. So I will describe and detail the agreement between the microscopic and the macroscopic dynamics, and I will discuss about memory loading, maintenance, and rehearsal via the persistent activity and the other phenomenon which is related to population burst. I will try to discuss about the competition about working memory items, and I will try to give you a flavor of the role played by different frequency bands in the working memory activity, trying to understand which is the dependence of memory capacity on the presentation rate. I imagine that the first question that arises in your heads in this moment is, why do we need another neural mass model in order to describe this kind of phenomenon, since there are a lot of heuristic neural mass models? So let me say the message of this talk, the take-home message, is that we can have some benefits from this model because I'm going to show you that we combined the exact neural mass that comes from recent findings in in-field theory with the well-established synaptic plasticity, short-term plasticity model developed by Zodix, Matram, and co-authors. And in this framework, I can affirm that our model is really powerful because, as I will try to show you in the following slides, it gives the one-to-one correspondence between the network, so the microscopic dynamics and the mean field evolution, so the macroscopic dynamics of firing rate, mean membrane potential, and synaptic variables, thus allowing to simulate very large networks. And in contrast to the well-known heuristic neural mass models that are usually used, it will allow to show subterranean integration that you will see are very important when retaining and loading the items. And most importantly, it gives us the mean membrane potential, and so it allows to describe the dynamics of evolution of the mean membrane potential that we usually can relate to experimental signals like EEG and so on. So let's go on. The microscopic dynamics, so the neuronal dynamics is described by a network of quadratic integrated fire neurons. For this kind of networks, in 2015, Monbrio, Pasot, and Roxin have developed an exact macroscopic description, exact neural mass models, that takes into account the evolution of the instantaneous firing rate of the network and the mean membrane potential. Together with this, we will take into account the macroscopic short-term plasticity developed by Zodix and co-authors in 1998, where are taken into account the evolution of the depression and the facilitation. In this figure, I've taken into account the interaction and the role of depression and facilitation between two neurons, but this can be easily extended to the interaction between two populations. So we come to an exact neural mass model with short-term plasticity for a single population, so that takes into account the evolution of the instantaneous firing rate and the mean membrane potential, and the evolution of the synaptic resource X and the utilization factor U that are coupled back, so the short-term plasticity is coupled to the dynamics of the network here. in the equation for the mean membrane potential. So this is the basic model for one population that we can extend to a multi-population. And the easiest kind of architectures that we have considered is this one that allows to take into account the memorization and loading of two items, because it is composed by two excitatory populations that are connected to the same inhibitory. In particular, each excitatory population displays short-term plasticity and codes from one item, and it can be stimulated by a stimulation current ES, while there is a common background EB that touches all the populations in the network. So if we start investigating what is going on, we will see that it is possible to observe memory loading and also memory competition just using two excitations. In particular, we have performed an experiment which is inspired by the one that has been shown by Mongillo and co-authors in 2008. And in particular, we tried to see if it is possible to maintain and to load items in one of the two populations by simply varying the population background. So if it is possible to have some memory maintenance with different dynamical regimes. And we see that it is possible to have maintenance through a mechanism that is related to population burst, as you can see here, and with persistent state. So for both the situations that are obtained by stimulating just one population, we see that there is a perfect agreement between what happens at the network dynamics, as you can see in this paragraph, and what happens at the level of the mean field dynamics. In particular, the features that emerge in this kind of memory loading and maintenance is that we observe a delta activity that is related to the excitatory population burst and the beta band activity that is due to the fast inhibitory collective oscillations. So the working memory that we observe is due to a ping-like mechanisms that is driven by the inhibitory beta band. In addition to this neural mass model allows also for a competition between memory items, as you can see here, which is called usually memory juggling. In particular, if we stimulate also the second population after the first one, we see that also the second population starts giving rise to a population burst, and so there is a memory, two items are memorized and compete, thus giving rise to memory juggling. So the entire phenomenon of memory juggling can be seen more in details by looking at the phase diagram, where we take into account what happens if we vary the amplitude of the stimulations and the duration of the stimulation. In particular, if we start from the situation of population burst, we see that it's possible to obtain for small amplitudes and for small duration just the retaining of item 1, so the item that has been memorized by the first population. Then for intermediate parameter values, we see that a frequent outcome is the coexistence between the two memory items, which means that we have juggling. And finally, just for sufficient long perturbation and intermediate amplitudes, we observe that item 2, so the item in the second population, prevails and it is memorized. This is completely different from what we see if we start from the situation where we have persistent state, because in this state we don't observe memory competitions, and the minimal perturbation amplitude you can see needed to observe an item switching is now larger than the other case. Another difference with respect to the other setup is that there exists a narrow stripe where item 2 can be selected, and then we observe more or less the same behavior for intermediate amplitudes and large durations. So things can be more complex if we use a bigger topology. In this case, we have considered a 7-item network where we have 7 excitatory populations that are coupled to one inhibitory In this case, if we present successive items to successive populations, we obtain some delta activity related to the moment where we load the dynamics, but we see, looking at the spectrogram, that there is a dominant signal which is due to the resonance between the delta activity and the beta band due to the presence of the inhibition. Of course, here we are presented the items at a certain rate. You see first item presented to the first, second item presented to the third population, and yeah, I see, and third item presented to the third population. If we do an extended analysis and we see what happens if we vary the presentation rate, we see that we have an interval of a window of frequency presentation where there is an optimal response of the network. So we see that we have a maximal capacity that is equal to 5, but while we see that for slow population, just the last items that are presented are retrieved and thus memorized, while for a fast presentation rate, we see that the first and the last items that are presented are memorized. So I want to conclude and give the time for a question. Just say that I tried to, with this talk, I tried to convince you that this population model is good to be used and can give a lot of, can have a lot of success, since it reproduces the macroscopic dynamics of the networks in terms of the firing rate and mean membrane potential, but is able to reproduce a lot of features that can be seen in the experiment, like the role played by different frequency bands and memory competition among the items. Thanks a lot.
Thank you very much, Simona. So if you have questions, put them in the Q&A window. So maybe I'll start with one question. So you showed that you, in this new mass model, you have several excitatory populations.
Yeah.
So right now it's also, people are starting to look into the different functions of different types of inhibitory neurons. So I assume that having different populations of interneurons with different characteristics would also be possible with your new model, right?
Yeah, of course. In the sense, the idea was to let me try to come back. The idea was to use a single inhibitory setup to simplify the presentation, to simplify also the investigation analysis. Because, but of course, what is clear is that you can couple in different ways the you are free to use the type of topology that you want to use. And what I expect is that then all the role played by the population will grow, in the sense that the inhibitory population is a sort of clock, and in the sense that you really see, if you look at the spectrum, for example, that the inhibition is really interacted, the inhibitory population is really interacted with the other ones, and you have a resonance effect that allows you to have a bigger signal in the beta gamma frequency bands. So all the fast frequency activity, the fast frequency band is enhanced due to the inhibition, and this will be enhanced if we use more inhibitory populations with respect to just one, I imagine.
Okay, thank you. So we still have time, so maybe I'll ask another more technical question. So how much do you gain by this low-dimensional description? So how much faster is it if you simulate that?
Well, you have to imagine that this neural mass model is the infinite size limit of a network. So in principle, you are simulating a network on an infinite size, and you can simulate it very easily in few seconds. In the sense, from a computational point of view, the analysis is very fast. while allowing you to add more and more biological characteristics to somehow to the network.
Okay, so thanks again, Simona.
Thanks.
And we'll move on to our next speaker. The last speaker in this session is Balbir Singh. He's currently a postdoc in Christos Konstantinidis' lab at Wake Forest School of Medicine in the US. And Balbir will talk about LFP phase synchronization as predictive of performance in a working memory task. So go ahead, Balbir.
Yeah. Okay, thank you, Christoph, for introducing me. Good morning, everyone. So today I'm just presenting our new study that LFP phase synchronization is a predictive of performance in working memory tasks. So actually that an individual neuron fire sparsely can't be oscillate themselves. It required a mechanism to oscillate or integrate with neural ensemble or network. So as you know that neural activity in prefrontal cortex is intentionally specialized representation of visually spatial information. Underlying dynamic changes in neural activity during the execution of working memory tasks are still largely unknown. So the memory process role on
Okay, we seem to have lost Balbir.
is to determine the pattern of neural interaction.
Bobby, sorry, you froze for a moment. Could you repeat your last point?
Okay. So I'm the specific effect of synchronization in time, frequency, and space to determine the pattern of neural interaction. So to study our objective is to study the oscillation dynamic, specifically phase synchronization that enable us to select and control neural activity during working memory. So we studied the two monkeys from just these are shows us that position of electrodes array in left prefrontal in one monkey and right prefrontal in another monkeys. So we install that chronicle here, and now this is the behavior training for the monkey. We just have fixation, then Q45, and we have that spatial locations, and after we have delay 25-second delay, and in sample we just giving that match and non-match, and for that match it have to be choose the diamond, and for non-match it have to be choose that edge. And this is the systematic diagram of monkey brain with approximate location of the recording and indicate related to the prefrontal landmarks area 46 and 8. For simplicity, we just divided these 64 electrodes in three regions, that such as that mid dorsal, posterior dorsal, and ventral. For this study, we just consider 255 sessions from both the monkeys. And these electrodes for like mid dorsal and posterior varies for both the monkeys, but ventral it will be same. now for an example, here I just give the example the phase of LFP for correct and incorrect trial. Now, if you just see that in correct trial, they will be more synchronized in Q period and as well as in sample period. And in right side, you just see that for these regions respectively. But in incorrect trial, there is a lack of synchronization, especially in sample period. Now, this is the another phase of LFP from another monkey. Now, after that, we just simply considering that inter-region correlation, just took that correlation from each electrode among that all another electrode from all the sessions, all the trials, all the sessions. And if we just look at that, in correct trial, there will be more synchronization in beta frequency as compared to that incorrect trials. Similarly, in that another monkey. So now that if correlation means we just simply taking that Pearson correlation, it will be comes due to that artifacts also. So after that, we just going for that phase synchronizations. And now what is that phase synchronization? Means simply for that, we just use that phase lag, weighted phase lag index. And phase lag index is used to determine the statistical interdependency in bivariate LFP time series. And it can be just given miscalculated by this one. And for in that case, we just simply consider that imaginary component of cross spectrums, and that range from 0 to 1. Now this is our, we just consider that index of that LFP synchronization. From, if you just see that there will be more synchronized, that will comes from 20 to 35 Hertz. And this is from that second monkey. But in case of that error, there will be no specific band, specific frequency, and there will be no specific electrodes also. Those are synchronized. Okay, now what should be the mechanism behind this, the synchronization? So that phase resetting. So the phase resetting will just I'm audible. So if you just see that here, that low frequency and that higher frequency, it will be just reset that higher frequency. Now there will be no in this way, there will be no phase, and it means that there will be no change in phase. But if you just see here, it will have a phase, but similarly it will change that distal and proximal LFP, or changing phase of that proximal and distal. Now after that, we just considering that distinct bandwidth. So for that, we just considering that beta frequency, and in that case, we just considering that correct trial and incorrect trial. So they will be more synchronized in correct trial as compared to the incorrect trials. So after that, we have to be just divided those time periods, like base period, Q period, and sample periods. And during that, we have to just see that that in Q period there will be in sample period that will be more desynchronized as compared to that Q period. And we just considering only those electrodes, those are more synchronized in baseline periods. So if you just simply see this graph, it will give us that Q period, a sample period will be more desynchronized. It means that it will be only just selected those neurons assembles. And here the summary: the phase synchronized in specific frequency is predictive of performance in working memory, and neural activity during working memory tasks is more synchronized during correct trial than incorrect trial in beta frequency. And neural association in correct trial desynchronize especially the stimulus presentation period in beta frequency, and the preferred phase initiate the synchronization or desynchronize and the response neural activity and induce a neural plasticity in memory. Thank you.
Thank you very much, Balbir. So in case you have questions, to just post them in the Q&A window. We still have some time, so I'll start with a question. So you basically showed that this synchronization in the beta band is basically makes the difference between correct and incorrect trials.
Yes.
And beta activity is usually feedback activity, right? So do you have any idea where this activity comes from already? How it works?
So actually here we just simply just considering that selectivity of that neural ensemble. So if you just look on those things, means in which frequency those are more synchronized, that, and after that it will be it will be like
Okay, you probably froze again, unfortunately. So let's wait a bit, maybe you can back. We still have some 4 or 5 minutes left. And we have another question queued up.
yeah.
Okay, sorry, Bobby, you froze again during your answer.
Sorry for.
But no worries, but we still have some time left. So
Okay.
yeah, maybe if you just quickly repeat your answer.
So I'm saying that the information, it may be the information about Q, it may be information about that spatial location also. So I didn't focus on that, means what kind of information it have, but when it will be more synchronized in that term, so it retrieve that only those neural ensemble will be synchronized, those have some information, otherwise because in this case we have like 64 electrodes, and those are chronicle, means implanted for several months, and it will be like consistency among in that task.
Okay, thank you. So we have another question from Mohammed Amin Samipur. He's saying, I'm not sure if I get your point regarding the phase reset. Are you suggesting that phase synchrony in a specific frequency range causes a synchrony across all different frequencies?
Yes. So here my main focus point is that specific frequency, and that should be synchronized. So most of earlier study also show that beta frequency play important role in working memory, and our study also suggests that it have a like more synchronized in beta frequency as compared to like the alpha and like a lower gamma, what we call as like 40 or 45 hertz. So here we just consider enhance that beta frequency like 20 to 35 hertz range, and in that it will be synchronized. And similarly we just see that another task also. So when that same monkey doing that several tasks, like simple to difficult tasks, so that range should be increased.
Okay, yes, so thank you, Balbir. And well, thanks again to all three speakers, Ehsan, Simona, and Balbir.
Ehsan asks one question.
Sorry,
oh, yeah, of course. If we have like 1 or 2 minutes.
Button doesn't work, and I ask. Sorry, and thanks for a great talk. How would we face synchrony across electrode from different areas? As I see that within area of beta band phase locking happening and in the bunch of the electrode. Is it any significant effect between areas from dorsal, lateral, or posterior areas in beta band or not?
So in this case, when I just simplify, means I just focus on this intra-areal region, means only this dorsal region. But within that, we just divided these areas also like in 3 areas. And in that case, mostly that mid and that posterior dorsal are more synchronized as compared to that ventral region. So in future, we just study that different areas also, means how they synchronize. But right now we just see these only within that intra-area, or within that also only not all the electrode are synchronized. Means it means that not all the neural assembly are synchronized. They have different information also.
Also, and do you have spiking activity formation electrode also or not?
Yes, yes. So we use for that means we already submitted in that study.
Great, thank you, Bobby.
Thank you.
Okay, well, yeah, thanks again to the 3 speakers, Eson, Simona, and Balbir. Thanks also to Melvin, who's running the backend here. And thanks to the audience for the questions and for listening. And enjoy the rest of your conference.
Yeah.
Goodbye, everyone.
Thank you.
Thank you, Christopher, hosting this session. And
thank you.
Thank you. Thank you.
Thank you.
I'm Medina Sarvastani. I'm a postdoc at Max Planck, Florida University, and I'll be your on-screen host. And in the background, we have Manusmriti Agarwal. She'll be your technical and off-screen host. And just a few reminders before we get started with this interactive session, and I hand it over. So with these sessions, with these interactive sessions in particular, I don't know if you guys have seen a few already in the conference. What we're doing is really running an experiment to try to find out the best way to transfer in-person poster sessions to this new virtual online conference format. So each speaker will give a 5-minute intro that'll be uninterrupted about their like an overview talk using a slideshow or a poster version of a slideshow. And this is like the initial overview you get at a real life poster session. You go up and you say, hey, can you give me a broad overview of your talk? Then we'll open up to the room for a more casual and non-structured discussion. So here is where the audience comes in and can really kind of influence how engaging and detailed the session gets. So based on your interest, you can post questions in the Q&A, and there's no dumb questions. So it's like a real poster session. Often I go up to a poster presenter at SFN and I say, hey, I'm not in this field, but I'm interested. Can you clarify this aspect? So all those sorts of questions, clarifications, comments, all of that sort of thing is really is totally okay and proper in this context. You should also upvote other people's questions in the Q&A because we'll sort of go through them in order. And let's see. And when you post your question to the Q&A, you can indicate if you want to come physically online. So this means you want to be on stage with us. So we can, you can indicate that, and we can enable your microphone for audio only, or your microphone and your video. So you can really be a panelist. And this is like the closest thing to you physically being there at a poster session with the few of us. So indicate that in your Q&A question if you'd like to come on stage, and we'll let you do that. Also feel very free to use the chat as an even more informal way to ask questions or post relevant papers or make comments. this conference, I mean, its inception, volunteer recruiting, really every aspect of it has been very linked to social media, especially Twitter. So please help us get the word out by tweeting about this session or the conference in general, and you can use the hashtag we've put in chat, NMC3 for that. Okay, I think that's all the session generic info. So today we have two very interesting talks. The third one had to be canceled, unfortunately. But two very interesting talks focusing on unraveling the relationship between genetic mutations and their associated cognitive deficits using multiple scales of cellular and subcellular investigation. Our first speaker is Francesca Van Tartwijk, and she'll tell us about the influence of FUS on axonal mRNA localization and local protein synthesis. Francesca is a PhD student in the laser analytic group at the University of Cambridge, where she's applying fluorescence microscopy-based methods to investigating biological questions of a broad range. She's previously obtained degrees in natural sciences, specializing in biochemistry and sensor technologies, also from University of Cambridge. And we'll put her Twitter handle and group website in the chat if you'd like to get in touch with her to continue the discussion. All right, Francesca, take it away.
I guess this is my cue. Perfect. Okay, so let me quickly find PowerPoint to share and start my timer. So hi everyone, my name is Francesca and thank you for the introduction, Monday. And I'm a PhD student at the University of Cambridge. And in this short 5-minute start to this session, I would like to give you a short overview of the general idea behind my PhD project, which is to investigate the influence of a protein called FUS on axonal mRNA localization and local protein synthesis in neurodegenerative disease. So FUS is one of those genes in which the mutations occur in certain families that make them much more likely to develop certain neurodegenerative diseases. In the case of FUS, that's mainly ALS, motor neuron disease, and frontotemporal dementia. Now, of course, a lot of work has been done since this was initially discovered to try and find out what causes, why these mutations cause disease. And it has been found that FUS that these mutations make FUS much more aggregation prone, as in it adopts this kind of semi-fibrillar state. And the interesting thing about this is that you could consider this to be a phase transition between the normal state of FUS and a more fibrillar form. And FUS actually normally also dynamically transitions between different phase states. In particular, it can be found in a soluble state or solution, or phase separated as a liquid droplet, which is important for its function. Now, of course, FUS is, the FUS gene is not expressed not just in neurons, but also in other cell types, but it is mainly associated with neurodegenerative diseases. So the question that we're trying to answer, that I'm trying to answer with my PhD research, or contribute to answering really, is why disruption of FUS function is particularly detrimental to neurons, and whether that's because the fibrillar aggregate is particularly toxic, or because neurons are particularly sensitive to that, or because loss of function of FUS is particularly detrimental for neurons. So FUS is an RNA binding protein, which means in the context of axons in particular, that it regulates transport and translation of mRNAs towards the end of the axon. And it does that because it has both RNA binding domains that enable it to recruit specific mRNAs into these liquid droplets, but also low complexity domains that allow it to self-associate and form droplets in the first place, and then further tether it to transport machinery, and so take the mRNAs to where they need to go. Now, this local translation of mRNAs in axons, so to be fair, also in dendrites, lets axons react rapidly and autonomously to external stimuli, which, as you probably all know, can be quite important if you have an axon such as that of a motor neuron that is 1 meter long. And this contributes to a range of functions, whether that's survival signaling and continued maintenance of the axon, local alterations to the cytoskeleton, such as microtubules or actin, or energy homeostasis and production of mitochondrial proteins as needed, as well as lots of other functions. Now, the hypothesis that we're working with is that because local translation or local protein synthesis is important in axons, and we know that to some extent microtubules are affected in, for instance, ALS, that the fact that fuzz is an RNA binding protein might imply that in fact that axonal translation is being affected, and that's why neurons are particularly suffering in these diseases. So to quickly recap, normally what you have is droplets get transported into the axon, mRNA gets released, and then proteins are locally produced as needed. Now, there are roughly two kind of ways in which this go wrong, at least within the axon. And that's either that mRNAs get sequestered in these slightly more fibrillar aggregate forms, and that influences, it makes protein synthesis much harder because they're not accessible, or in fact, that there is some kind of transport effect and either the mRNAs don't get packaged properly, or there are issues with the cytoskeleton along the axon that might make efficient local delivery of mRNA difficult. And in particularly, since there are indications and in many cases also good evidence that cytoskeletal proteins are locally translated, these could in fact be interacting with each other and progressively making the situation worse, where local production involves where loss of local production of cytoskeletal elements or proteins occurs. Now, the system we use to investigate this is Xenopus retinal ganglion cells. And the way we do this is we take a newly fertilized Xenopus embryo, inject in vitro synthesized mRNA into the forestal stage, and then let it develop a bit further. So I'm working with 4 different constructs, just GFP as a control, normal wild type 16R, which is an artificial aggregation prone mutant. And what we do then is wait for the embryo to reach the stage where normally the retinal nerve would start extending from the eye primordium towards the brain, and instead take out the eye and plate it on a dish. And what happens then is that you end up with something that kind of looks like this. So you have the eye primordium on a dish and axons just fanning out quite excessively, either to further manipulation or for imaging. And you don't have any dendrites in this particular system, so it's quite an easy way to do biochemical or imaging assays of specifically axonal function. And we've been using this for a range of experiments trying to investigate what fuzz is doing in these axons. So I think that was my background chat. I'm happy to answer general questions about the project idea or about some of the things that we've been trying to do.
Okay, awesome. Yeah, so attendees, so this really is like a poster session right now. There's about 10 of us sort of gathered at your poster. So guys, please feel free to ask questions. I have a general questions. One that's going to sort of give away my ignorance, and then one more specific about your work. The first is, can you tell me what kind of proteins need to be or are usually phase separated, and what proteins aren't?
Well, I think that's a very good question. And it kind of, well, phase separation is a way of dividing the cytoplasm into compartments without using membranes. And so one of the advantages of that is that it's very dynamic and it can be regulated by post-translational modifications as potentially other triggers as well. It also happens quite regularly in the nucleus. And it is, I would say, probably a relatively new field. So more and more membraneless organelles or liquid droplets, etc., are being discovered on a regular basis. I'd say quite a lot of it is to do with RNA processing or RNA targeting. So you have within the nucleus or within the cytoplasm, and it's generally mediated by local complexity domains because they have the ability to self-associate, but also by proteins that include both RNA binding domains and low complexity domains. So that what you have is a very multivalent interaction with lots of proteins interacting to some extent sequence specifically, but also a lot more flexible because the structure isn't exactly determined. And then what you end up with is like these kind of droplets. So I'd say it's a lot of RNA binding proteins, RNA processing proteins to some extent also within the nucleus. In some cases, as part of the DNA damage response, or as part of like the stress granule formation can also be considered to be an occasion like that, where you have dynamic regulation of accessibility of mRNAs. So those are the main examples that I would highlight.
Cool. We're being asked, Rabia is asking you to please speak a little louder. I can hear you clearly, but I guess
okay, sorry.
The audience might not be. No, it's okay. Yeah, so now a more specific question. Well, okay. I'll ask this one first and then I have another one. So you mentioned that it's possible that there's these sort of two mutually unexclusive effects of FUS mutations. One is sequestration that sort of disallows the proper, and the other is that there's a transport defect in the first place from the soma down the axon. I missed, or maybe you didn't tell me, or if you did, I missed it. I missed how you're gonna test those two hypotheses.
Well, I didn't really explain it in much detail, but happy to go into it further. So generally, we have within the lab several established assays to look both at RNA transport and local translation in this particular model system. So what we're doing for transport, actually what we've already done so far, so I can show you the hidden slide. Generally, what we do is we inject together with the mRNAs Cy5 conjugated to UTP, like in the very early embryos, and then gets incorporated into lots of, basically all RNAs that get synthesized during development. And then as the axons grow onto the dish, what you see is these lots of different RNA containing granules, which will both be ribosome RNA, tRNA, as well as mRNA. And we can see these move. And then if we just acquire a video over several seconds and build a kymograph, what you can do is extract motion parameters from that. So we've done several replicates of this. We have a data set now which still needs a bit further analysis to try and separate which populations of granules might be affected in different ways. So that's the transport side. At least what we can tell already is that there aren't dramatic differences when it comes to, if you just aggregate all the different granules. And as far as the local translation side, we can look at specific target proteins, either assaying their mRNA in axons or their local translation with specific reporter constructs. Or what we do for general bulk translation rates is a pure mycin stain, where you expose the axons to pure mycin for 10 minutes or so, which gets incorporated into any newly locally synthesized proteins, and then you can fix that and stain to see the intensity of the overall signal.
I see, that's pretty cool. Okay, I have another question, but let's go to the attendees. Yosefa Zaldivar is asking us, you covered this a little bit, but if you could tell us more about the fluorescence microscopy-based methods you use.
Yeah, so actually, the lab that I'm based in is called the Laser Analytics Group. So effectively, what we have is a lot of different microscopes for different resolutions that we can achieve. So this kind of thing you can do quite regularly on a confocal, but when we're trying to get higher resolution studies of like slightly less fast process than this kind of transport, quite often what we do is use structured illumination microscopy, in which case we illuminate the sample with a grid-like pattern rather than with one specific, just wide field. And what that allows us to do is extract slightly higher frequency information from the sample and just get a bit more specific knowledge of what's going on there, particularly if we also want to see, which is something we may or may not do in some way, how these events co-localize with organelle positioning, etc. So it's like relatively fast when it comes to super resolution.
Okay, awesome. And yeah, another broad question. So if I remember correctly, FUS mutations are related to particular types of dementia and motor neuron disease. So do you have any sort of speculating hypotheses about why these mutations that may have to do with protein synthesis and transport and axons have these particular downstream cognitive deficits?
Well, when it comes to motor neuron disease, the argument that is suggested is just by the virtue of the length of the axons. So of course, any kind of transport from the soma will take much, much longer. So we think these are relatively dependent on local translation, etc., and particularly sensitive to disruptions in transport. When it comes to the dementia aspect and the frontotemporal cortex, that argument obviously applies slightly less. And I would say because FTD is also very commonly associated with aggregation of another protein to which fuzz is closely related, which is TDP43. Probably we also, to explain that entirely, we also need to look at the interactions with that as well as other proteins.
I see. And is there, are there non-neurons, maybe in the peripheral system, that still have very long processes? So you can test this idea about protein synthesis and transfer and the effect of FUS, particularly in neurons, your sort of opening hypothesis.
Well, right now we have also tried some of this together with a collaborator in some primary hippocampal neurons, which obviously don't have this similar length of processes. And also, to be honest, with this model system, we're not seeing the kind of lengths that compare with motor neurons, because obviously that's quite hard to replicate in vitro. But I would say because these axons are shorter, if anything, we're underestimating the effect of FUS, so we won't be the disruption of local translations. So in that sense, we are unlikely to draw false conclusions. But yes, I agree that it would be interesting to try and do this with slightly longer nerves. I think SCGs might be slightly longer, but also we can grow these cultures for several days and they keep extending. So we can also try it at different time points.
Cool. Okay. Okay, well, that's just about time, so perfect timing. Thanks, Eva, or I'm sorry, Francesca, and good luck with the rest of your research.
Thank you.
Thank you. All right, so Eva, you're up next, and let me grab my notes here.
Should I already start sharing my screen?
Sure, yeah. Okay, great. So our next speaker is Eva Cristina de Oliveira Figueiredo. That's a great name. And she'll tell us about the importance of BCL-XL and the onset of cognitive deficits associated to 22q11 deletion syndrome. Eva's a PhD student in the lab of Professor Paolo Bezzi at the Department of Fundamental Neurosciences in Lausanne. Her thesis project right now is aimed at understanding the postnatal maturation of astrocytes and their role in the formation of brain circuits. That's something we really know very little about, so that's great. And she's going to take advantage of the 22q11 animal model, the 22q11ds in this project. But today she'll tell us something a little bit different. She'll talk about the neuronal dysfunctions associated to this disease. Thanks for being with us, Eva. Take it away.
Thank you. So welcome. I will guide you through my poster. I did it a bit interactive. Let's see how it is. So 22q11 deletion syndrome is caused by a microdeletion in chromosome 22, and the most common microdeletions are of 3 or 1.5 megabases. However, the severity of the disease of the phenotype does not depend on the size of the microdeletion, and it is more likely that the genes responsible for the phenotype are encoded in the short deletion. Patients display variable features that include cognitive deficits related to psychotic symptomatologies. In fact, 30% of young patients develop schizophrenia. Cognitive deficits are related to dysfunctions of the prefrontal cortex, so the PFC, that is one of the most important brain areas in schizophrenia. In fact, patients show problems in the development of the PFC, and many studies have shown molecular and morphological alterations of pyramidal neurons in this area, and the neurons have structural abnormalities and in dendrite maturation and spine formation. The cognitive deficits of 22q11 deletion syndrome patients are particularly related to the working memory and cognitive flexibility. That are two behaviors related to the prefrontal cortex. Therefore, this is consistent with the defects found in the patients suffering from schizophrenia. Besides the cognitive deficits, patients also show metabolic problems, and it has been shown that schizophrenic and 22q11 patients have altered transcript levels of proteins regulating mitochondrial functions in the PFC. In fact, 6 out of the 45 genes that are deleted in the syndrome encode for mitochondrial proteins. To study this syndrome, we took advantage of the LGDL mouse model that presents the deletion of all but one ortholog genes to the human, 1.5 megabase microdeletion, and also present cognitive impairments and brain abnormalities that are very similar to the ones found in human. Because of the deletion of the genes that encode for mitochondrial proteins, we decided to examine mitochondrial gene expression in brain tissues of the LGDL mice. We found that among other mitochondria-related genes, BCL-XL was specifically downregulated in cortical tissues, as you can see here, but not in hippocampal ones, which was consistent also with the transcriptome analysis performed by Sarkeol in 2008 in another model for 22q11 deletion syndrome. We just checked if the decreased levels of BCL-XL was specifically downregulated in cortical tissues. We checked if this could have a role actually in neuroanatomical abnormalities associated to 22q11. So at first we analyzed the mitochondrial distribution and the morphology of neurons in vivo by labeling the single cells with in utero electroporation using a plasmid that was produced by Tommy Lewis and Frank Polo at the Columbia University. And this plasmid expressed a cytoplasmic fluorescent protein, the scarlet, you can see here, and a mitochondrial matrix targeted fluorescent protein, the YFP. Then we performed morphometric analysis, and this analysis revealed remarkable differences between wild-type and LGDL animals, as you can see. Mitochondrial length and mitochondrial dendritic coverage were reduced in LGDL mice compared to the wild-type ones. Then we analyzed the morphology of neurons, and we measured the dendritic length and performed shoal analysis that showed decreased length of basal dendrites, and the arborization of pyramidal neurons was also decreased, as you can also see clearly here from the reconstructions. Then we performed the same analysis in vitro on cultured neurons, and we found the same differences regarding mitochondrial length and coverage, and also about the dendritic arborization that was decreased. Moreover, to evaluate the functionality of mitochondria, we performed OCR and ATP content analysis and found that LGDL neurons have compromised oxygen consumption and reduced ATP content. So these findings suggest a possible link between the decreased dendritic complexity of the neurons and the mitochondrial dysfunction. Then, in accordance with the reduction of the BCL-XL previously reported, we decided to rescue the levels of BCL-XL in vitro and in vivo in order to evaluate the possibility to ameliorate the phenotypes observed. We employed a pharmacological approach using the TAT BH4 peptide that you can see here. It consists of the BH4 domain of the BCL-XL fused to the protein transduction domain of the HIV virus, so the TAT, And in vitro, we also used a genetic approach where we transfected the cells with the BCL-XL plasmid. Here I show you the effects of the treatment in vitro, where you can see that the treatment with the genetic approach on top and the one with the peptide below were able to recover the mitochondrial phenotype in LGDL neurons. In vivo, we treated the animals with, with the TATPH4 peptide, and we were able to rescue different phenotypes. Here, the mitochondrial phenotype, mitochondrial length and coverage were rescued, as you can see here. Then, with the in utero electroporation, we saw an impairment in the dendritic length and complexity. With the treatment, we could see an increase in the complexity and the length of the basal dendrites. Here again, you can see some reconstructions of the neurons. Then, since in the literature circuits dysfunctions were described, we decided to look at the miniature postsynaptic currents using electrophysiology. And LGDL animals presented a significant decrease in the frequency of the currents, but not in the amplitude. And with the treatment, this difference was rescued, as you can see. Then, to have an outcome of the dysfunctions observed, we decided to perform some behavioral analysis, and we performed the T-maze test to evaluate the working memory, and the two other discrimination tasks to evaluate the cognitive flexibility. And we obtained a significant rescue for the cognitive flexibility and a partial one for the T-maze. So in conclusion, we observed a decrease of BCL-XL in the prefrontal cortex of LGDL animals, together with a morphological impairment in pyramidal neurons and their mitochondrial complexity. And we were able to rescue these phenotypes by restoring the level of BCL-XL. So this is all from my side. I thank you for your attention, and I'm happy to take any questions.
Okay, great, thank you. We'll go to questions in a second, but I just want to say I found this zoom in poster format pretty effective. That's cool. I'm gonna have to remember that.
Thank you, I'm happy.
Yeah. And also, this seems like you're at a completion point with this project. You know, you sort of came for full circle. It's cool to see behavioral deficit rescue and the last part. That's awesome. Okay, we have a couple questions. Rabia Hashim is asking if you could elaborate on how you developed the animal model, including which strains you used.
So it's the 22q11 deletion syndrome.
Yeah.
So it's the LGDL animal.
Yeah.
Yeah, we got it. I mean, we didn't do it ourselves. It came from another lab. It was developed in another lab and was characterized really well. Actually, is just one of the many, because you can see, I mean, you can go back.
Yeah, it's quite a common animal model, right?
Yeah, here you can see there are different ones, and the LGDL one is the one we are actually using. And there are different deletions. We have longest deletions and shorter deletions. And we are using this one because it's very similar to the deletion that we found in humans.
Cool. And another question from Yosef Zaldivar. He's asking if their recordings, I'm assuming he means the mitochondrial recordings, were performed in head-fixed animals.
This the in utero electroporation is done like post-mortem. So we do brain slices,
okay.
And actually we use a filler to see the morphology of the neurons and the mitochondria. So we take images with the confocal microscope, and then we analyzed these images.
Okay, cool. And someone is asking, and I think you answered this, but if you could clarify, did you try the genetic rescue with the animal model? With the
in vivo, no, we didn't. We did only in vitro, but it's something that we want to try. Yeah.
Okay. Oh, I misunderstood that. Okay, that's great. And I have a couple of broad questions now. So it's interesting that BCL-XL is downregulated, particularly in cortical, but not hippocampal, subcortical issue. Why do you think that is? Do you guys have ideas, or can you speculate?
Honestly, I don't know.
Yeah, fair enough.
Yeah, I'm sorry.
No, that's okay. I mean, I like an I don't know honest answer. Okay, and then this is something that I just maybe missed. So it seemed that the BCL, where BCL-XL was downregulated, one of the side effects was that mitochondrial length was reduced everywhere, but there was also just less mitochondria in the dendrites, producing sort of lower basal dendrite length and affecting neuronal morphology in general.
Yeah, the coverage inside the dendrite was decreased. Yeah.
But was the mitochondrial length lower everywhere in the soma and the axons?
Yeah, we checked for the dendrites, so the dendritic coverage, and yes, this was for every neuron.
Okay, that's interesting. Tamara Zender is asking if you looked at, that's interesting. Yeah, mitochondria, fusion, fission, movement in general.
No, we didn't, but would be nice to investigate this, since BCL-XL is actually playing a role in mitochondrial fusion and fission.
Yeah, okay, that's interesting. Yeah, that would be really cool. And then I guess we'll finish with one last very broad speculative question. So therapeutic targets here. What's the path there?
Yeah, so here we saw the impact of BCL-XL. We rescued the levels of BCL-XL, and we had the rescue of all the phenotypes we observed also in the behaviors. So I think that like restoring the levels of BCL-XL could have a great impact as treatment for these patients.
Absolutely. I think you made a pretty compelling case for that. Okay, great. That's our time. Thank you, Eva. Thank you, Francesca. Yeah, thank you, Manu, for running a very smooth technical session. And thanks to all of the attendees for engaging our audience. I think the interactive sessions can work well, guys. This was kind of cool. All right, enjoy the rest of the conference, everybody, and bye.
Thank you.
Bye.
Hey, hi everyone.
Hi.
Yeah, the broadcast has started and we're live on YouTube and we should see attendees. Okay, yeah, and attendees are starting to slowly roll in. Great, so good morning, good afternoon, good evening. Welcome to the second day of Neuromatch 3.0 conference. I think this is the first day where we have full and temporally reasonable participation from Europe, since we have morning sessions. So welcome to our European attendees. My name is Medina Sarvastani. I'm a postdoc at Max Planck, Florida, and I'm joining in right now from Florida, US. I'm your on-camera host for this session, and we have Sophia Bachelor in the background running all of the various technical things. Thanks, Sophia. And just a few reminders while everybody rolls in and before we get started. So, we're trying to reproduce a real online conference with all of its human participation aspects. So the session can, based on attendee and panelist participation, it can really sort of influence the feel of the session. So please feel free to use the chat to post papers or make comments or ask for clarifications. And if you have questions, post them to the Q&A and upvote, importantly, upvote other people's questions, because if we're short on time, we'll sort of go through them in a democratic fashion. And finally, so much of this conference, its inception, volunteer recruiting, advertising has really happened on social media, mainly Twitter. So please help us continue to get the word out. Registration is free right now. And actually, you don't even have to register because we've made the schedule, YouTube links, everything just available for the public audience. So help us get the word out about this session and specifically, but also the conference in general. And you can use the hashtag Sophia, if you can drop that in the chat if it's not already, hashtag NMC3 for those tweets. Okay, let's see. Yeah, great. I think we can get started now. So this looks to be a very interesting session focused on using mathematical modeling to understand the dynamics, control, and stability of various muscles and general posture. We'll have 3 talks, each a total of 15 minutes, including a 3-minute Q&A, and I'll help you guys keep time because unfortunately we don't have much wiggle room. So with that, our first speaker is Lahiru Wimalasina from Emory University. He's going to tell us about denoising EMG using dynamical systems models of muscle population activity. Lahiru, why don't you take it away?
Oh, thank you very much. Does everyone see a white screen?
Yep, looks good.
Great. So thanks. My name is Lahiru. I'm a 4th-year PhD student working under the guidance of Dr. Chetan Pandarna in the Systems Neural Engineering Lab. Today, I'm going to be talking to you about a tool we have developed to denoise EMG recordings using dynamical systems models of muscle population activity. To start my talk, I want to highlight an example demonstrating the capability of animals to produce complex movements. In this video, the great Rodney Mullen does 2 tricks back to back. To achieve complex, accurate movements like this, we must activate sets of muscles that may span a limb or appendage in the appropriate time-varying pattern. This raises the open question as to how the nervous system solves this high degree of freedom control problem. In order to answer this question, we must understand the neural command signals that underlie muscle activation. A major barrier to progress, however, is our ability to interpret the electrical activity of muscles through electromyographic recordings that will henceforth refer to as EMG. Consider recording EMG from multiple muscles in the hind limb of a rat during locomotion over many cycles. Rectifying and aligning the EMG to a key behavioral event, such as when the foot strikes the ground, allows us to observe variability that is common across muscles, such as this feature I've highlighted. But we also see a lot of variability that appears highly stochastic across the many repeats of the aligned behavior. This poses a challenge in analyzing EMG activity. We have to find a way to remove this noise or denoise the recordings to extract the embedded neural command signal. The typical approach to denoising EMG is to apply a low-pass filter to the activity. The same low-pass filter is applied independently to each channel and effectively treats high-frequency features as noise, which is evident from the frequency response of the filter, where the boundary between what is considered signal versus noise is defined by the frequency cutoff we set for the filter. This choice we make affects what we consider signal, sometimes completely smoothing over features that are evident in the original activity. Further, finding the optimal filter cutoff requires a supervised process that requires testing whether the filtered EMG corresponds to some external recorded measure, such as decoding behavior. This may potentially bias our denoise estimates toward representations that can explain behavior well, regardless of whether key features in the activity are suppressed. Here, we present an alternative unsupervised approach that denoises EMG by modeling muscle population activity as the output of a dynamical system. We call this approach dynamically estimated EMG, or DEMG for short. Our model assumes that the observed rectified EMG is a noisy observation of an underlying muscle activation command that is generated as the output of an underlying dynamical system. The dynamical system is represented by the state S, which starts from an initial state S0 and evolves through time by following a state update equation F, which is determined based off of the current state of the system and a time-varying input U applied at each time step. Our assumption is by modeling the underlying dynamics that govern EMG, we can better separate signal from noise to extract the underlying neurocommand signal to the muscle. Two key previous studies serve as motivation for our approach. First, Cecilio et al. 2015 demonstrated that recurrent neural networks, or RNNs, could be trained to generate EMG activity. The internal state of RNNs are nonlinear dynamical systems, where the state update rules are implemented by the weights of the network. In this study, the authors showed that an RNN provided with limited input could train its weights to infer the underlying dynamics needed to generate recorded EMG activity of multiple muscles recorded from monkeys performing a variety of reaching movements. Second, Panarnath et al. 2018 demonstrated that RNNs could be integrated in an architecture called latent factor analysis via dynamical systems, or LFADS for short, to create a model that denoises spiking activity for a neural population. LFADS infers an underlying rate for each channel of the input population spiking activity. These rates are generated from an RNN that was trained to model the dynamics that govern the evolution of the population activity over time. We adapted the LFADS architecture to implement our dynamically estimated EMG approach. The generator serves as the underlying dynamical system that models the state S. The network takes as input the rectified EMG of a muscle population and passes it through a series of RNNs that estimates an initial state and a time-varying input. These are passed to the generator to evolve S over time using the learned state update rule F that models the underlying dynamics of the population activity. The output is then passed through a nonlinear transformation to estimate the parameters of a time-varying gamma distribution for each EMG channel. To get our denoise estimate of muscle activity, we take the time-varying mean for each channel. We train all of the network's parameters together via backpropagation of the reconstruction costs of the gamma log likelihood of observing the noisy EMG activity given the estimated time-varying gamma distribution for each channel. We tested our DEMG approach in two separate model systems. We first applied it to activity recorded from 12 muscles spanning the hind limb of a rat walking on a treadmill at constant speed. The stereotypical cyclic evolution of locomotion made it amenable for initial testing of our dynamical system modeling approach, which attempts to learn consistent rules that govern state evolution. Multiple joint angular kinematic parameters were inferred from simultaneous high-speed video tracking of joint positional markers. It also enabled annotation of key behavioral events, such as the timing of when the foot strikes and lifts off the ground for each stride, allowing us to segment the data into different phases of the locomotion cycle as shown, with the gray stance phase when the foot is in contact with the ground and the orange swing phase when the foot is lifted in the air. We next evaluated the generalizability of our approach by applying it to a different model system, muscle group, and behavior. We modeled the EMG activity recorded from 12 muscles in the forearm of a monkey during a wrist force isometric test. The monkey performed a series of trials to control the movement of an on-screen cursor by applying forces to a 6 degree of freedom load cell. 1 of 8 targets was presented in a location surrounding the central hold position, resulting in the monkey producing 1 of 8 distinct force trajectories. Additionally, brain activity was simultaneously recorded via 96 channel microelectrode array in the hand area of M1, allowing us to investigate the relationship of EMG to recorded brain activity. We first visually compared the input and output of the DEMG models. DEMG output, colored in blue, is overlaid on the input rectified EMG for a subset of muscles from both model systems. The DEMG output captures the time-varying changes of the muscle population activity while substantially reducing high-frequency fluctuations. To capture the activity of the entire muscle population, we use dimensionality reduction via PCA to reduce the DEMG output for the 12 recorded muscles down to 3 dimensions. For the rat locomotion data, each trace represents an individual stride, and each part of the individual trajectories is colored by the corresponding phase of the locomotion cycle. The white dots signify the start of the trajectory at the time of foot strike. They are clustered in a small region of state space and evolve along a consistent trajectory that loops around in a figure 8-like pattern. Our assumption of an underlying dynamical system is intuitive for the rat data, since the behavior follows this consistent cyclic pattern over many repeats. However, this assumption is less intuitive for the monkey data, where the behavior is neither cyclic nor predictable from the initial state, where the input of target information at the beginning of each trial has a profound effect on the resulting EMG activity. This poses a greater challenge since our model must be able to infer this input, since we don't provide it with any information about target direction. The visualization shows single trial muscle population trajectories for the 4 cardinal reach directions. The individual trials all start at a common baseline state, but then separate to distinct locations of state space corresponding to the reach direction by the time of target hold, suggesting that the model was able to infer the inputs needed to describe the muscle activity for different conditions. We first validated our DEMG approach by asking whether the representation is still predictive of behavioral variables recorded in the model system. To do so, we used optimal linear estimation to get these predictions of the behavioral variables. We compared our predictions to multiple low-pass filtered EMG representations with different frequency cutoffs ranging from 40 Hz down to 2 Hz. We first mapped EMG onto predictions in the rat locomotion data of joint angular position and joint angular velocity for 3 joint angles of the hip, knee, and ankle. DEMG prediction performance is represented by the blue bars, while the gray bars represent performance for different low-pass filtered representations ranging from the highest frequency cutoff to the lowest frequency cutoff. We found that across all 3 joint angles, DEMG improved joint angular prediction across the board when compared to the standard approach. Here's an example of the trial average predictions of the joint angular velocity of the hip for different representation predictions overlaid on the true data. Visually, we see the DEMG predictions better match the true hip joint velocity than the standard low-pass filtered representations. Importantly, we find that the optimal frequency cutoffs for predicting joint angular position versus velocity were different, highlighting the limitations of having to choose the filter cutoff with the standard approach. With DEMG, you get higher prediction performance for both behavioral variables, even though the model training was completely unsupervised to behavior. We saw a similar result in the monkey wrist force test when we compared predictions of DFDT between DEMG and the same low-pass filtered EMG representations tested in the rat locomotion data. Here again, I show an example of the trial average predictions for one condition. Again, DEMG provided more accurate predictions than any of the standard approaches. We next asked how the filtering properties of the DEMG model differed from a static filtering approach like a low-pass filter. To analyze this, we estimated the frequency responses of the models during the stance and swing phases of locomotion, where we observed large differences in the change in behavioral state, as evident from this example of the joint angular velocity of the ankle. We first estimated the frequency response of the static linear filter during the 2 periods and found the expected result that the responses were near identical in these 2 phases of behavior. However, if we do the same analysis for DEMG, we found that the model adapts to allow more high frequency content to pass during the swing phase than in the stance phase. This trend was observed across multiple muscles, where DEMG adapted its filtering properties to coincide with observed changes in the behavioral output, even though the model was trained in a completely unsupervised manner with respect to behavior. A clear example of the high-frequency features preserved by DEMG can be seen in the activity of the vastus medialis and lateralis muscles, which play key roles in stabilizing the knee during locomotion. Aligning EMG around the foot strike event and averaging across all the strides shows a consistent response across the vastus muscles in the rectified EMG output following foot strike. This feature is preserved in the DEMG output. However, if we compare it to the output of the static low-pass filter at 10 Hz, this feature is completely smoothed over in this representation. An important thing to remember here is that the 10 Hz representation led to the optimal prediction of joint angular velocity. So if we would have used decoding as our supervised process to choose the optimal filtering cutoff, we would have lost this feature in our denoise estimate using standard approaches. DEMG is unsupervised to the behavior, yet is able to capture both these high frequency features and predict behavior with high accuracy. Finally, in the monkey data, simultaneous recordings of motor cortex via microelectrode array allowed us to ask how well EMG representations could infer simultaneously recorded brain activity. We used optimal linear estimation again to predict smooth threshold crossings from each of the 96 channels. To summarize the results, we compared them on a scatter plot where the vertical axis shows DEMG prediction performance and the horizontal axis shows low-pass filtered prediction performance. The dashed line represents equal prediction performance for both representations. So if the scatter is skewed to one direction, that would suggest that that representation is predicting better than the other. Each dot here represents a single prediction performance for one M1 channel. As we see, we found that DEMG significantly improves prediction of motor cortical activity over the standard EMG approaches we tested. So in conclusion, DEMG is a dynamical systems modeling tool that uses RNNs to infer underlying dynamics of muscle population activity to denoise EMG. It is trained in an unsupervised manner with respect to behavior, yet can generate EMG representations that outperform standard approaches at predicting behavior. It adapts its filtering properties to different periods of fast behavioral changes to preserve high-frequency features in the EMG that are relevant to behavior. And finally, we saw that it improves our ability to infer brain activity that is actually relevant in generating the muscle activity that we tried to denoise. So with that, I would like to thank my PI, Dr. Chetan Panarnath, for his guidance, as well as all the collaborators who have helped so much with putting together this work. So with that, I would like to take questions.
Okay, great. Thank you for a very interesting talk. We're a little bit over time, so maybe we just have room for sort of a joint question. Jungwoo Lee from the audience is asking, how well does your method predict new behavior different from trained behavior? And that's sort of similar to my question, which is, can your method infer hidden unmeasured dynamics or alternatively tell you which are the joints that you should be measuring and the others are correlated?
Yeah, so it's a great question. So sort of asking about the generalizability of the approach to be able to explain more dynamics. So right now we sort of tested on 2 separate model systems and only tested within that. But I think one way to sort of test the generalizability of this approach, with the rat locomotion data, we can look at different speeds of walking or different inclines of walking so that we can test whether, when we change those parameters, as our model, if we train it on a subset of that behavior, can we still explain the changes when we sort of change those kinematic parameters? Or in the monkey data, we can apply different dynamic loads to the wrist while they're performing this task to see if we can still explain the underlying dynamics of the muscle, even if we don't train our models with respect to all those behaviors.
Great, thanks. There's a couple of really interesting questions about common noise rejection underlying dynamical systems model. We're gonna move on, but if you wanna answer those questions in the Q&A, that would be great. Okay, sure. Thank you. All right, our next speaker is Jungwoo Lee from the Newman Lab at MIT. He's going to tell us about identifying human postural dynamics and control from unperturbed balance. We're going to put some info about Jungwoo in the chat. Specifically, he's organizing a workshop, and we'll put that information on. All right. If you want to stop sharing your screen, Lahiru, that'll allow Jungwoo to take it over. All right, that looks good.
Hi, can you hear me?
Yep, sounds good.
Okay, so hello everyone, I'm Jongo, the PhD student at MIT. And thank you for coming and listening to my talk. As the title indicates, I'm going to talk about the method we have recently developed to study human postural dynamics and control in unperturbed balance experiments. So, where is it moving? Right. So we all know that we humans have amazing ability to maintain our body upright. And this video is just one particular example of many, many remarkable examples that show how incredible capability we have. Standing upright in our daily activity might look tedious and very easy compared to this walking on a slack in a valley. But it's actually quite an interesting and challenging task because our many degrees of freedom body is inherently unstable. So we have to apply corrective torques to stabilize our inherently unstable body. And that postural control, how do we do that, has been a very interesting question to all of us. We all want to understand how humans do balance our body. Now, who are we? We would include neuroscientists, physiologists, physical therapists, engineers, and roboticists. And they have approaching this question in all different perspectives. So I think I have to clarify that I'm a mechanical engineer, and I'm interested in enhancing our understanding of math models of joint space dynamics and control. So there are two experimental paradigms in balance studies, perturbed and unperturbed. In perturbed balance, you apply external perturbation to the human subjects, and you examine how they react upon the external perturbation. A typical way of applying perturbation is to translate or rotate the platform the subjects are standing on, and then record the motion of the subjects in response or interact to those applied transformation or translation or rotation of the platform. So you have a direct measurement of the movement of the platform and the motion of the human body. With these two signals, you can apply the well-established input-output system identification techniques developed in engineering society. From here, you can identify input-output dynamic relation, and actually you can tease out the postural control or the control strategy that humans are using. This approach has been well established and very popular since last few decades ago. There are a lot of interesting studies out there, and you can actually learn a lot from them. On the other hand, unperturbed balance, as the name says, you don't apply perturbation. So if you don't apply perturbation, would subjects move at all? Yes, they would, because they are kept challenged by the internal noise in our system. So you can still measure the output motion of the human subjects, and you can characterize human behaviors. But the problem here is that the driving signals or driving forces here are internal noise. So you cannot have a direct access to the measurement of them. This actually hinders you from applying the input-output system identification method that has been well used in the perturbed balance cases. So perturbed balance is a perfect setup to apply system ID, and we can learn a lot from that. So why do we bother? And actually, humans are notoriously adaptive to external perturbation. For example, as reported, the identified dynamics and control of a human subject is depending on the type and the size of the perturbation that you have applied. Moreover, if you want to apply the external perturbation in a systematic way, you actually usually need and sometimes bulky, a lab-mounted robotic systems in your lab, which is a little bit, how to say, like demanding. So on the other hand, unperturbed balance allows you to study the natural and daily balance activity. And sometimes it is also useful when external perturbations are not applicable. And for example, if you want to study a human balancing on a challenging environment, like the balance on a slack line you have seen just before, or when you want to study patients or subjects that have impaired balance, it is kind of dangerous to apply external perturbation. So if it exists, a systematic system ID method for unperturbed balance is desirable. And we propose a presentation. Okay, so our goal is to identify the joint level human postural dynamics and control. And without any external perturbation, without any direct measurement of the internal biological noise. This can be mathematically formulated. So I promise I will not exhaust you with math, but please bear with me a little bit. So our human postural control system can be mathematically described as on the left panel. So the human body can be described with the states, the joint states X, and the input joint torque U. And we model the controller, the postural feedback controller, as with the controller gain matrix K with sensory measurements Y. And these sensory measurements and the execution of the joint torques are corrupted by the internal noise. Together, we can actually summarize this all together as a closed loop dynamics of the system of human body. Now our objective is to estimate the closed loop dynamics ACL, and then estimate controller gain K to understand what control strategy the human subjects are using. And here we're going to only use the measured time series of the Z. I think I forgot to mention what Z is, but the Z is the measurement of human body, which is a function of the joint states. We also are equipped with a known A and B matrices because we can get them from gravitary inertial mechanics, but we don't know what all the noise sources are. We actually found that it is possible to solve with the autocorrelation of the output signals with non-zero time like K, without I'm skipping all the details of the math, but you can actually estimate your ACL, the closed loop dynamics, and the controller gain K in a very reasonable and reliable way. All right, so to validate our method, we modeled a balancing human as a double inverted pendulum, which is, I guess, the simplest possible model that we can use right now. And we stabilized the model with the linear quadratic regulator just for simplicity and just for make an example. And we tested two different controllers that was studied before in our lab. The case 1 beam balancing, case 2 quiet standing. Okay, so I know I'm very bad at aesthetics, so please do not blame me for the choice of colors. But here I show you example simulation results of single trial of two different cases. So each model performed 90 seconds of standing, which is reasonable for like human balancing studies. And so each model performed the 5 trials of that. And from all 5 trials, we can actually use the method presented before to estimate the A and K matrices. We iterated this 10 times to calculate the mean estimation error. And also because we are in the simulation world, we know the exact A and K matrices, so we can actually compute the estimation error as shown below. we tested multiple different combination of biological noises, noise and measurement noise. And here you are seeing the mean estimation error of dynamics A on the left and the control K on the right. Note the color code indicates a very small error in all combination of noise strength. And the advantage of this method is highlighted when you actually compare it with typical least squares method. So you can find very large errors in the certain conditions, as on the top right corner, reaching up to 150%. The difference between two methods is more evident in the second case. And again, the proposed method on top row gives us very small error, while the bottom row shows you, like, especially the controller gain error reaches up to 400%. So in summary, we have developed a system ID method for unperturbed balance, and which would not require you to externally perturb human subjects. We validated this method in simulation, and it's applicable to real experimental data is ongoing. Of course, there are limitations. We assume linear and time invariant dynamics and white uncorrelated noise, and each assumption may need validation. Interpreting the result requires care. We only identify apparent joint level dynamics and control. This actually means that the contribution of intrinsic impedance, neurocontrol, active neurocontrol, feedback, feedforward, predictive, whatever, are all blended in our result. And moreover, the time delay is not explicitly incorporated. Nevertheless, I believe the method allows us to better understand human balance in an unperturbed case, which has not been tackled before. This has a practical implication because clinicians may use our method to assess individual balance quantitatively and accurately, and engineers may use this method to improve their technology. The mathematics behind this method is quite general, and actually it could be relevant to many different fields, such as motor learning model or assessing stability of rhythmic movements, such as walking. For details, I suggest you to check our paper, which is under review. And with that, I will stop here and thank you for listening, and I'll welcome any questions or feedback or comments.
Okay, thank you. That's a pretty interesting approach, Jung-Woo. We have time for one very quick question. So I'm just going to ask, have you tried estimating the dynamics in one participant and applying it to another? How well does it generalize?
It's a good question. Are you asking if I applied this method to one subject and applied it to the other subject?
Yeah.
I think the point of this method is you can use this method to identify the dynamics and control of individual subjects. So all of the subjects may have different control gains, and we do have different body parts. We may have very similar skeletal mechanical dynamics, but the controller gains could be quite fairly different. So
that makes sense.
Yeah,
okay.
So this method provides you to identify individual subjects rather than the group results. And
but it doesn't have to be. It doesn't have to be, active inference online, right? You could estimate the underlying model one day and then apply it the next to sort of initial. Yeah. Okay, great.
Yes.
Okay, that's really cool. Okay, we are out of time. Thanks a lot, Jung-Woo. I'll let Neelima take it away.
Oh yeah, sorry.
No, it's okay.
This is.
It's okay. I'll introduce her so you have a minute. Our final speaker is Neelima Sharma from Yale University. She's interested in understanding the consequences of the mechanical constraint of stability on animal morphology and neuromuscular control. And we'll put her contact info in the chat. Neelima, the stage is yours.
Cool. Hi, so I'm Neelima. I'm a graduate student in Madhu's lab at Yale University. Today, I'm going to talk about some of the work we have done on understanding finger stability and precision grips. So human hand dexterity is something to marvel and something that robots aspire to achieve. Here is an example of an extremely dexterous human. This is Okatanpe, a contact juggler from Japan. He manipulates this acrylic sphere effortlessly. However, the effortless action relies on precise fingertip forces, stable finger postures, and soft and flexible fingers. So one of the key grasps that he and we rely on while holding objects is called precision grip, where we produce stable and precise forces using just our fingertips. Now, forceful precision grips are related to stone tool making in early hominins, and therefore have implications for studying human hand evolution. The ability to precisely grip objects is sometimes lost, such as due to diseases like arthritis, and there are various modes that a finger can become unstable. One of the modes of instabilities is having high contact stiffness. So and this has been shown by a lot of robotics and control theoretic studies. For example, here is a video. A person manipulates the manipulator with a relaxed arm posture, and then the manipulator moves smoothly. However, when he tenses his muscle and increases the contact stiffness, you can see these high frequency oscillations, which indicate instability. One way to improve stability properties is to decrease the contact stiffness by making the manipulator soft. However, that leads to another problem, which is related to buckling of fingers. So fingers have internal degrees of freedom, and if the stiffness is not enough, then your fingers can become unstable and buckle. And you're welcome to try this experiment at home. Just take your finger, put it in a flex posture, and push on a surface as hard as you can, and you'll see that your finger buckles sometimes. So why do we not become unstable while grasping objects is the topic of my talk today. So to characterize the mechanical response of the finger and to assess the severity of buckling instabilities, we performed an experiment where we asked people to push on a surface as hard as they can. And we saw that sometimes their finger became unstable, which was characterized by the sudden change in posture. And the DIP angle, which is the most distal angle of your fingertip, showed an exponential increase with time. So we see that the time constant related to this exponential increase varies between 9 to 23 milliseconds. Now, neural feedback control cannot stabilize these buckling instabilities because the time for the neural signal to travel from your finger to spinal cord and back takes at least 65 milliseconds. So how is it that humans maintain stable finger postures when they apply contacts? So we hypothesized that joint stiffness due to muscle contraction maintains finger posture during fingertip force production tasks. So we all know that muscles are formed of actin and myosin proteins, and these proteins form a cross bridge upon neural activation. The higher the neural activation, the more the muscle tension because the number of cross bridges increase. In addition to tension, each cross bridge is also associated with a certain stiffness, and because these cross bridges are attached to each other in parallel, the net stiffness of the muscle also increases with activation. This has been experimentally verified as well. You take a muscle, put it in isometric condition, and apply a muscle twitch, overlay it with length perturbations, and you'll see that the muscle stiffness with respect to muscle tension shows a linear increase. The way it is modeled is, is that muscle tension, it's shown on bottom left. Muscle tension is related to muscle activations given by A linearly, and F naught is the isometric tension of the muscle. Similarly, muscle stiffness matrix, which is a diagonal matrix of muscle stiffnesses, is also related linearly to muscle activation by this muscle tension M1. And joint torques and joint stiffnesses are simple transforms using the muscle movement arm matrix of the muscles about the various joints, and therefore joint stiffness and joint torques also increase linearly with muscle activation. So the point of the slide is that if you're increasing your fingertip forces, the postural stiffness will also increase linearly, and therefore it's possible that stability is simply a byproduct of increased fingertip forces, and you don't really have to control it. So we test this by using a biomechanical model. Here, the finger is modeled as a 3-link manipulator on the left, and it is driven by 7 muscles. The finger is hinged or attached to the ground at the fingertip E and the knuckle, which is the most proximal joint of the finger. So because of the constraints, this 3-degree of freedom system actually has only 1 degree of freedom. And our objective is to find out whether muscle stiffnesses can provide enough postural stiffness for stability. And to do this, we find the minimum stiffness that's required for keeping the finger posture stable. And it is simply a scalar because the system is 1 degree of freedom, and it is directly proportional to the fingertip force magnitude F0 and a posture-dependent length scale L. So now we design an optimization experiment. We ask what is the optimal activation pattern of the 7 muscles that maximizes the fingertip force. We find the optimal activation pattern, use this to calculate the muscle-induced joint stiffness, and use the maximal forces to calculate what the minimum stiffness required for stability is. And comparing the two will tell us whether or not the finger remains stable. So this is what we find. On the right bottom is the activation pattern, which is optimal. It produces a distal fingertip force of 42 newtons. And we find that the minimum stiffness to keep this posture stable is much larger than the joint stiffness induced by the muscle. And therefore, when we apply a perturbation at t is equal to 0, we see that the finger becomes unstable and buckles. So previously I told you that neural feedback cannot stabilize a buckling finger. And now I'm showing you that even muscle stiffness is unable to stabilize a finger posture when people are producing maximal forces. So what is it that keeps our finger posture stable during day-to-day tasks? Well, it is possible that neural control's primary objective is to select muscle stiffnesses for stability, and that takes priority over the instructed goal of producing maximal forces. So we test this prediction. And we proposed an experiment where we ask people to produce fingertip forces in the presence and absence of a finger stiffening splint. Now, if neural control actually prioritizes stability, then attaching a splint will remove the constraint of stability and people will be able to produce higher fingertip forces. So here is the control trial where a person maintains a prescribed finger posture and produces forces, which is shown in the graph just below the finger. We also measure activity from 2 muscles, the flexor digitorum profundus and FDS, which are shown on the right bottom. And we see that the activity varies. It correlates with the force activity. Next, for the experimental trial, we dorsally attach a finger stiffening splint and see that the people can produce much higher forces in the presence of the splint, and their corresponding muscle activity also increases. So this is one trial for 1 subject, but the collated data for 22 subjects is shown at the bottom, where we see that the percentage increase in force for 22 subjects is around 70%, and the corresponding percentage increase in EMG is around 80%. So we also do a similar experiment with a shorter splint and see the force and EMG increase again. The increase is smaller than what we saw with the full splint, but it is a positive increase nonetheless. So on the right here, we plot all the data for percentage change in force versus percentage change in EMG and see that these quantities are correlated for both the full splint and the half splint condition. So what this means is that when we remove the constraint to maintain stable posture from the finger, people are able to produce higher forces. And this may be because people are able to access muscle activation patterns, which were earlier precluded to them because they were associated with unstable postures. All right. So now I have spoken until now about maximal fingertip forces. However, these results all have consequences and implications for light grip forces, and which is because even though there are multiple activation patterns that people can use to produce submaximal forces, experiments that measure EMG activity from all 7 muscles of the index finger find that as people modulate their force or scale it down, the EMG muscle activation pattern also scales down. So you're simply relying on scaling of the activation pattern. So now the question is, can scaling the maximal activation pattern for producing smaller forces allow you to become stable? And the answer is no, which is shown in the middle graph here. The minimum stabilizing stiffness as a function of the distal fingertip force is linear, as we discussed earlier, but the joint stiffness due to muscle activation is always lower than the minimum stiffness required for stability. And this is because the stability properties are preserved under linear scaling. However, we know that people can co-contract their muscles and become stable at submaximal forces. So here we numerically calculated various muscle activation patterns that produce the same fingertip force, where the distal fingertip force is 9 Newton, and each dot represents one activation pattern. The y-axis shows here the stability. So eta is the eigenvalue, b is the damping of the system. So here we see that the minimally co-contracted muscle activation pattern 1 is unstable, and the maximal co-contracted activation pattern 4 is stable. And there is a trade-off between stiffness and stability, such that if you co-contract your posture to increase your stiffness, that improves the stability. However, you are stiffer, and that can lead to other problems. So this is again an example that linear scaling of an activation pattern preserves stability properties, and this is shown for the marginally stable activation pattern 2 in the graph on the right. All right, so in conclusion, I have told you that muscle stiffness is unable to stabilize finger postures when we are producing fingertip forces, and we must rely, neural feedback is unable to stabilize finger postures, and we must rely on muscle stiffness. And the way neural control decides chooses muscle stiffness for allowing you to remain stable limits the contact forces that you can produce. So this is our paper, and preprint is available upon request. Thank you for coming to my talk, and I will take any questions.
All right, thank you, Neelima. That was cool. We are going to get kicked out in about a minute, so we've put your contact info in the chat if people want to continue the conversation and ask questions. But very quickly, can you comment on potential applications of this work?
Oh yeah. So a lot of neuropathologies cause people to become highly soft in their muscles, which causes them to buckle when they are holding objects. So one way this research can be applied is to design splints that allow them to access muscle activation patterns that do not destabilize them. And then there is also implication for open loop control in robotics and prosthetics, where you do not really have to rely on feedback control all the time, and can use the mechanical properties of the design to keep you stable at short time scales at least.
All right, great. Okay, thank you, Neelima. Thank you to our other 2 speakers. And thanks to Sophia Bachelor in the background for running a smooth tech session. And thanks to everyone in the audience. Bye for now.
Yes, I can hear you now.
Okay, great. I'm sorry, I think the problem is that then my Hopkins address.
Ah, okay. I just promoted you to panelist, so I believe you should be able to share.
Okay, let me just, my computer crashed in the process, so let me just find my document.
There we go, all panelists, yes.
Okay.
All right.
Oh, and you can't see me. I just realized.
Voila, there we go. Time to rock and roll. All right, glad you're able to make that work. This is maybe not uncommon problem with Zoom when we have multiple university accounts.
Yeah.
Okay, so so we have about 40 to 45 minutes together. And just for everyone in the audience, we will have, the speaker will present her work, and then we'll have about 10 to 15 minutes left for Q&A. If you have any questions, please use the Q&A feature at the bottom of the Zoom window. You can post questions throughout the talk, and please upvote those questions that you find most interesting. So with that, if you want to go ahead and share your screen. Let's see. Okay, great. And so for this morning's, or wherever you are, whatever time it is there, today's keynote will be from Kathleen Cullen, a professor in the Department of Biomedical Engineering at Johns Hopkins. And the title is Predictive Sensing, the Role of Motor Signals in Sensory Processing During Active Self-Motion.
Okay.
Take it away.
Yep, thanks very much, James. And apologies for the little snafu with the two dueling email accounts. So I'm going to start by just introducing the vestibular system in terms of sensory coding. So there are really two parts to my talk. And as most of you appreciate, during our everyday lives, our sensory systems are both activated by stimuli that are a result of our own behaviors. So if you're in the case of the vestibular system, as you move through the world, you can activate through your voluntary behavior the vestibular system, as well as things that occur externally. So if someone's driving you around in a car, that would be an external stimulation to the vestibular system. And the ability to distinguish self-generated versus externally applied stimuli is really essential both for our perceptual stability, but also for accurate motor control. And recent work in my group is really focused on understanding how self-motion information, particularly vestibular information, is encoded during active versus passive conditions. And in terms of terminology, we refer to vestibular sensory inputs that are self-generated as reafferents, and those that are externally generated as exafferents. Okay, so just to kind of remind people what the vestibular system is, from an engineering point of view, it's a really cool system. You have 3 canals on either side of your head in the inner ear, which effectively act like a 3-dimensional gyro system and tell you your head rotation relative to space in 3 dimensions. And they're basically organized at right angles to each other, so you can encode all of 3-dimensional rotations. In addition, we have 2 otoliths, which form 2-dimensional maps of linear motion, and thus give us 3-dimensional linear motion. So together we have 6 dimensions of movement encoded 3 linear and 3 rotational, much like a MEMS platform that you might now put on an animal for tracking. And the vestibular system, most people think of it as being synonymous with the vestibular ocular reflex, which is there to generate compensatory eye movements when your head moves relative to space to give you stable vision during your everyday activities. And we'll see if this little movie works. This is a movie taken by my postdoc back when I was at McGill in Montreal, just walking down the street. Oops. It doesn't look like that's going to work. I'll try again. So he's not doing anything really crazy. He's just walking, and you can see he has one of those old GoPros on his head that didn't have gait, stabilization feature. And that sort of bouncing movement is what you would get if you didn't have compensatory eye movements for your head movements, right? You would have your eyes just kind of locked into your head like you were a doll, and the world would move up and down on your retinas. But that's not what we see, because we have this very tight relationship between head motion and eye motion, which is mediated by a 3-neuron pathway. In addition, we have the vestibular system generating to other really essential functions like head and body posture. So if you start to slip and you catch yourself before you even realize it, you have vestibular spinal reflexes that are helping you do that, as well as our ability to compute our self-motion and perceive how we're moving through the world. So as I mentioned, the first thing I'm going to do is really talk about the vestibular system from a sensory coding point of view, sort of the old, standard approach that people have used. And I'll go through this pretty quickly. It's just to give you a flavor of how beautiful this system is. So a lot of people who study the vestibular system back in the day started off as engineers, generally electrical engineers, and we realized that we could do things like take linear systems approaches and apply them to this system, because again, it's very, this beautiful readout of how you're moving through space, and you can put an input in, which would be, say, a head motion, either rotational or translational, and then you can look at the output, which would be a neuron's response or an eye movement, and change the frequency and use these sort of basic linear approaches to understand the system. And the transfer function in this condition would be, say, you're putting an input in, which would be head motion, and then you look at the afferent response, and you try to understand what all the channels, et cetera, are doing in order to shape the responses. So this is really the standard approach that people have used. And what we did in our lab is recorded from individual afferents, and we used monkeys mostly as a model. We do some work in mice, but what I'll primarily talk about is monkey work, and record from single afferents in the vestibular system. So again, you have these 5 end organs, 3 rotation and 2 translation on either side of the head. We can record from afferents, which come from only one of these end organs. And then you can see how they respond to different inputs. So in this case, we're recording from horizontal canal afferents that respond to horizontal yaw rotations. And we can apply different frequency rotations and see how the afferents respond. And I should just note there are 2 classes of afferents. There are regular afferents and irregular afferents, which when the animal's not moving have a baseline firing of about 90 to 100 spikes per second, but there's big differences in how regular the interspike interval is between the 2. And what we know is that the irregular afferents primarily hook up with these type 1 hair cells that are in the sensory periphery. And that type 1 hair cell evolved more recently. It looks as it evolved effectively when animals left water. So when, from being a fish and an amphibian to moving on to land is where you see this evolution of this type 1 hair cell. So presumably it's there to deal with the more dynamic and transient stimuli you would have in a terrestrial-based lifestyle than an aquatic-based lifestyle. So what do we see when we do this sort of systems analysis, linear systems analysis for regular versus irregular afferents? So you can put in, say, a 0.5 hertz sine wave and rotate the animal back and forth, or a higher frequency sine wave. And what you see right away is that you get an increase in modulation with increasing frequency. So these neurons are high pass. And it's even more pronounced for the irregular than the regular afferents. And that's what's shown here. In this curve, you can see the gain as a function of frequency. So generally over our range of natural head motion, if we look at the frequency band over which we generally move, irregular afferents are actually much more sensitive. So this then raises the question of why do we have regular afferents? So Mohsen Jamali, who was a PhD student in my lab at McGill, and Jerome Carriot, who was a postdoc with me, around the time that these MEM sensors first became available in sort of a limited way, we took advantage of them, and we basically packaged in our own sort of little platform a MEM sensor, 6-dimensional MEM sensor for rotation and translation. We added a cell phone battery and a little data storage card, and we strapped this kind of awkward looking now that we have these smaller sensors that people are using, but this at the time lightweight package to people's heads and had them running around the city of Montreal. And we really wanted to see how people moved, because sort of surprisingly at that point, we didn't really know. There'd been some work on treadmill, but we didn't really understand how people moved. So what we found is that people actually move a lot more dynamic, vigor or activity than you might imagine, right? So people reach very high intensity movements up to 8 Gs, like just getting up and swinging around to get your cup of coffee, 15,000 degrees per second of rotations. If you look at the distribution of motion, this is showing rotational motion. It's very non-Gaussian, very peaky in the middle, which means that we spend a lot of time just sitting and not moving like most of you guys are doing now. Significant power up to 20 hertz. And for those of you who know something about stats or natural statistics of sensory inputs, they don't follow power law. And what we find is actually the high passness of the afferents, for example, that I showed you is really perfectly balanced to whiten the spectrum of the natural stimuli. And by the time you get to central neurons, you have this really nice whitening that we've described in some recent papers. Okay, so we did a similar type of analysis of monkeys and mice, because those are our experimental animals. And fortunately for us, the statistics of self-motion in monkeys look actually very similar to what we see in humans. So it does argue that they're a good model system for us to look at. The other thing that's important is if you look at the tested range, and this is where sometimes techniques limit our ability to really understand more important questions, what effectively we've been taught as students and researchers is that in order to use our linear systems approaches, we had to stay in a linear range, right? Because you don't want to go into saturation or cutoff. So this is where we've been probing for decades. But this is, if you look at the natural range of motion, really what we're encountering every day, right? And to me, this was a really kind of, maybe not a big surprise, but it really was striking how different the two are, and how, in sort of playing, in order to, do things properly, we'd actually maybe missed the bigger question, right? So I'm just going to give you a little bit of insight into what we've just, discovered recently. So using natural head motion, or little epics of natural head motion that we recorded in our freely moving monkey experiment that I showed you, what we could do is then take those epics of head motion and play them into our turntable or our hexapod, and move the animal. And when we do that, using more naturalistic stimuli, we can do things like look at information coding. We can also look at spike timing and temporal codes, and really push things out of this sort of linear range. So I'll just give you a couple examples to give you a flavor of the type of work we've done. What the top panel shows is a head motion stimulus in black. The firing rate of the neuron is this dot, this sort of digital line at the bottom with the spikes. And then what you can do is convolve the spike train with a Gaussian to get this black trace here, which is on the top, which is the spike density. But we can also convolve that spike train with the optimal linear filter to do the best stimulus reconstruction possible, to see how we can actually turn this spike train into the stimulus. And what you can see right away is for the regular afferent, the spike train looks really like the stimulus. It's very easy to get a good stimulus reconstruction. If we go through that same exercise for the irregular afferent, even though it has higher gain, what you see right away is it's much harder to reconstruct the stimulus, right, which is in the middle based on the spike train. So our best stimulus reconstruction is that red dashed line. And that's pretty typical. So this suggests if you have a larger coding fraction, you have better information coding, and indeed that's what you see. So this is information coding of the regulars across that same frequency band that I showed you for gain, right? And what you can see right here is that even though the irregulars have higher gain, this is for the canal system, they have lower information, right? So just compare this, right, with the sort of standard approach. So that's what it shows is that the signal noise is better for the regular afferent channel. The other thing you can do is things like look at spike timing precision. So that's another thing we did. So we basically looked at performance using spike timing classifiers. So you would, instead of just playing those little epics once, you'd play them over and over and over again in sort of a frozen noise type experiment. And then look at how much precision there is in the firing rate of the neuron for this repeated stimulation. And what you can use different classifiers that are available, like Victor Popora or the von Roscombe classifiers. And what we find is you can do classification close to perfect with the irregular afferents. If you look at this, spike timing precision, with spike timing precision on the order of 5 milliseconds. So it's really precise, well outside the range of anything in the stimulus itself, whereas it's much less, the performance is much less for the regular channel. And again, this is sort of interesting, right? Because the regulars have higher information, but they have less spike timing precision. So this just shows you these two channels, regular and irregular, one with rate coding, one with temporal coding. And what was sort of interesting to us when we looked centrally, and we focused to start on the neurons that generate vestibular spinal pathways and project up to the thalamus, what you see is that these central vestibular nuclei neurons look actually very much like the irregular afferents. Okay. So what I've show using sort of classical approaches, we have two channels of information going into the vestibular system from the periphery. We have the regular afferents that are low gain but high information. They use a rate code. And it looks like they actually project based on some preliminary work we do to the VOR pathways primarily, or transmit information. And then we have the irregular afferents, which are high gain, low information, use a spike timing code, and they seem to be transmitting their information best to the vestibular spinal and ascending thalamic cortical pathways. So now I'm going to switch gears and talk about happens when you actually generate your own self-motion. Because what I've done in the first part of the talk is we recorded self-motion and we played it into our turntable. But what happens when the animal's actually generating it? And I'll go through this pretty quickly, but the point is that, you know, you want to be able to make this dissociation between what you do and what happened to you. So there's that old observation by Helmholtz that if you make saccades, the world is stable, but if you tap your eyelid and your eye just moves a little bit, you get this jumping of the visual world, right? So normally we have our eye movement command that the brain can use to keep track of, you know, what you've done and where the world is so that you have this stable percept. You also need to make this dissociation for the case of accurate motor control. And that's what I show you here. So vestibular, so for example, vestibular spinal reflexes are essential for providing robust postural responses when you have an unexpected perturbation. But if you're trying to actually purposely move through space, they can be counterproductive. So this is an example I often show of somebody walking down a hill, say in Montreal, less the case in Baltimore, slipping on black ice, right? And hopefully they're going to catch themselves right before they fall. And it would be this vestibular spinal reflex afferent central neuron down to the spinal cord that could help them catch themselves. Then so this is unexpected vestibular input or X afference. But you could imagine somebody maybe experiencing very similar vestibular inflow, right? Like maybe their head is moving very similarly through space, but she's not going to want to stop herself. She's not going to want to stabilize her body in space, right? So you could argue that, you know, you have this 3-neuron pathway in its shortest form from the afferents to the spinal cord, and you don't have a lot of real estate to do some sort of switching. So you might want to see, you know, a differential encoding of active versus passive movement very early on. So that's what we looked at. First, we start at the beginning, right? We have afferents, and then we're going to have these central neurons, and then we project in the pathway shortest form down to the spinal cord. So what happens at the afferents? And the reason it's not just a kind of, you know, silly question is because there is actually a vestibular efferent system that projects back out to the periphery. And the argument's long been that this is its role. Its role is when you generate active movements, you send a signal out to the periphery and you bring up the resting discharge of the neurons and decrease the modulation so you don't saturate your sensor. So we found that that's not the case during active head movements. So this is a monkey generating his own movement, active head and space motion. This is a nice firing rate of these central neurons. And then this is when we take that same head movement and play it into our vestibular motion device, and the neuron behaves pretty much in exactly the same way. We can fit a little model to it, and then we can look at things like differences in the sensitivity or the bias. We could look at differences in the phase lead, and there are no differences for these active versus passive conditions. We've more recently started looking at locomotion, and this is a paper that's currently under review. So we can basically use deep lab cut to monitor and accelerometers how the animal's moving through space. And this just shows an example of passive stimulation, passive pitch. This would be a posterior canal afferent that we're recording from the walking and running, head motion. And then what you can see is that regardless if the movement is generated by the animal or by us moving the animal, we get the same responses across all these conditions. So the argument is that for afferent inputs, everything I told you in the first part of the talk is true. Afferents really act like MEM sensors. They don't care, or IMUs. They don't care if they're context independent. They don't care what kind of behavior generating. They bring a non-ambiguous signal into the brain. Okay, next part we record from the vestibular nuclei, right? So the next stage of processing. And here we see something really different. So this just contrasts with the sort of parallel figure I showed you for afferents. And what you can see right away is when the animal generates its own active head motion, we get this pretty much, for this afferent, negligible response, whereas the neuron is very sensitive to that same input if we take that head motion and play it into our turntable. And what's even more striking is this next slide where we have the animal's head unrestrained. We rotate him passively through this sine wave, right? But he's actually generating his own head-on body motion, which is this black line. The total head and space motion is this black line up on top, and the same here, right? So we have passive, active, and then total, which is the sum, and the neuron's only encoding the passive component. So it's a really striking computation, right? And this is at the first stage of central processing. So just to make it really clear to people, if you're recording from an afferent, it would encode this dashed blue line, right? It would encode total head and space motion like a sensor, like a MEM sensor. But these neurons actually specifically will encode in this condition just the passively applied component, the X afferents. So the big question now is, how does that occur? And that's where we've been spending a lot of time trying to understand this really beautiful computation that happens at the first stage of processing. We know we have robust afferent input, so that suggests that something's canceling that input at the first stage of processing. And I should note, we see this for rotational motion, for linear motion, for combined motion. We see it across species. So it's a very general mechanism. So you could argue maybe you have, during passive motion, just vestibular input. During active movements, you're also doing things like stretching proprioceptors, generating a motor command, you have knowledge of motion. So maybe it's just one of these inputs that's getting summed in and combined. And that's all we need to really think about. So we looked at that, and that seemed like the logical place to start. So what you can do is look at neck proprioception by having the animal's head fixed relative to space and rotating its body under a world fixed head. So there's no vestibular input. And we never in normal monkeys see, rhesus monkeys, see any modulation in this condition. So there's no proprioceptive, passive proprioceptive response. We also did an experiment where we had the animal making normal head movements, and then when he didn't expect it, we put electromagnetic clutch on, so he could not move his head, but because he was attempting to, we could measure the neck torque. And in that condition, we don't see any sort of inhibitory response. So it's not just a motor command that's coming in and turning things off. And I just put this electric fish up for comparison for people who know the electric fish work by people like Curtis Bell and more recently Nate Sawtell, who trained with Curtis. If there's a kind of this beautiful mirror image that electric fish people see in sort of the analogous experiment to this one. So it suggests that the mechanism that's being used in the vestibular system is a little different than what people have reported in the electric fish. It's a bit more sophisticated. And then we also did this experiment, which is looking at higher level percepts of motion. If you can predict, in this case, we had the animal or the monkey trying to drive and chase a goal with his target light. So he's controlling the light that's beaming off his turntable by rotating a wheel and moving. As he moves his turntable, the light moves with it, and he's chasing our goal. And when he does this, we were sort of surprised. It took time to train the monkey, but this actually is encoded in this condition as if it's passive. And I can take questions about that at the end. Okay, so what we found is that we see cancellation of vestibular reafferents when sensory feedback matches that expected as a result of the motor command that was generated. So this is what is the case during active movement. Specifically, the brain's computing an estimate or internal model of the sensory consequences it predicts based on the action that was generated, and then compares this internal model, this expectation, to the actual sensory feedback. So the comparison then results in a sensory prediction error signal that guides the updating of a motor program. So this sounds like a motor learning story, right? But in the present context, this computation of a sensory prediction error enables this distinction between X afferents and reafferents. So I'm just going to talk a little bit about the work that Jess Brooks did for her PhD to test this idea. And she recorded from the output neurons of the cerebellum, particularly the vestigial nucleus, rostral vestigial nucleus, and to test this idea. And if our hypothesis is true, then we should not see reafferent suppression if we alter the relationship between the motor command that was generated and the sensory afferents. So what we did is set up a little learning experiment where we had the animal making active head movements, and then we put a load on his head so that when he moved his head initially only moved half as much as he expected it to. Then over time, he learned he needed to work harder to accomplish the task. So that's the learning phase. Then occasionally we would turn, once he was in static sustained learning phase, we would turn off the torque motor every so often, turn off the load, and have these catch trials. So this just shows what you've already seen, passive and active sort of control movements. Then we put on this resistive torque. So he initially only moves his head half as much. Then over time you get learning so that the behavior matches what we saw for the normal active trials. And these are all active movements. And then once the animals learn this new condition, we can turn the load off and get these catch trials where they're expecting a load, but the load's not there. So you get this big overshoot. And what do neurons do? So this shows you what you've already seen for the first two rows. Passive is encoded, active is not. And then what you see right away is even though this head movement is less than half of what the normal active head movement is, the neuron's more responsive. And it's responsive in a way that you would predict based on the neuron sensitivity to passive motion. Then throughout learning, you see that the head movements get bigger, but the responses get smaller, right? So it's pretty cool. These are active head movements, all of them, but what you're seeing is this sort of updating in the internal model as the animal's learning to deal with this new load. And then for the catch trials, again, even though they're huge active movements, they're encoded as if they're passive because there's a mismatch between what the animal expects and what's actually happening. So you can look at the sensitivity relative to passive. So passive is encoded, active is suppressed. And then these initial active movements are actually encoded as passive. Then you see this reemergence of reafferent suppression. And then again, catch trials are encoded as if they're passive movements. And they follow very nicely the head velocity error that is occurring during this learning protocol. So we can also look on trial by trial basis, and basically this neuronal sensitivity follows very nice percent change from control head movements. So what this shows you is when the relation to a motor command to move the head resulting actual motion is altered, neuronal responses are consistent with the rapid updating of an internal model. And this is really important for calibrating motor pathways, right? So you initially want to make that difference between active and passive, so you make the right postural change if it's unexpected. But if you have a persistent mismatch, you want to calibrate your system. I don't think I have that much time because I started late. So I'll just note, we also showed, I won't go through this in detail more recently, this is work by Isabelle Macros and Jerome Carriot, my group at Montreal, who we looked at what happens when you actually move your head relative to gravity. And that's interesting because you have neurons in the vestibular nuclei that encode net acceleration, which is effectively what's encoded by the otoliths. But you also have neurons that encode tilt and translation. And in order to do that, they need to combine canal and otolith afferent inputs in very specific ways. So this is in passive condition, we can find these different classes of neurons using a series of protocols. And what we found is in the active condition, these gravity-related responses are canceled during active movements. So again, these tilt-selective neurons and net acceleration sensitive neurons that are sensitive in very specific ways in passive conditions, when the animal's generating its own movement, are no longer sensitive. And if we look at the afferents, again, they're of course acting like IMUs. So the responses of the cerebellar output neurons are consistent with the existence of an internal model that also predicts the sensory consequences of gravity. And this is really, interesting because it implies you need this really complementary suppression of the canal and otolith input to match in order to do this properly. And now we're investigating the processing of how it's altered during self-motion at the central level during locomotion. Okay, so I focused on thinking about this, the utility of this from the point of view of calibration of motor circuits. But I also mentioned at the beginning that one of the reasons you want to have differential processing of active and passive motion by the cerebellum is for perceptual stability. So we have looked a bit at that. In particular, we looked at what information is encoded by ascending thalamical cortical pathways during active versus passive self-motion. I put Omid's picture here because he's going to be talking later in the meeting about what's actually happening at the level of the cerebellum. We've been primarily looking at the cerebellar output neurons. He's looking at what computations occur within the cerebellar Purkinje cells. So this work was done by, in the thalamical cortical pathways by Alexis Dale, as her PhD research. And this should look very familiar to you. We have the dashed sine wave is what we apply as experimenters. The black line is total head and space motion, which is what the afferents would record or encode like a MEM sensor. And then the red line here is what the animal's actually generating himself. So total head and space motion is the black line. And again, these thalamical cortical neurons that are in the ventral posterior lateral thalamus, which gets direct input from the vestibular nuclei and projects up to the cortex, encode preferentially the unexpected component of motion, the part that we are applying, and ignore the part that the animal generates himself. So this is interesting because, again, the VPL projects up to the areas of cortex that we assume are involved in self-motion perception. So we believe this mechanism is important in suppressing vestibular reafferents to help us ensure perceptual stability. Did I just move or did something just move me? So this is a sort of grand summary showing you that the canals and otolith afferents encode passive and active motion the same way. But as you go centrally, you see more and more suppression. So by the time we get to the thalamus, we have about 90% suppression. So just to summarize, I will note that we have descending pathways. We know the vestibular spinal pathways. It makes sense to have this, I guess, preferential encoding of unexpected motion. It's important for vestibular pathways that mediate vestibular spinal reflexes. And then we have this ability to update based on a mismatch, which is important for calibrating these circuits. And then we also see the same mechanism contributing to ascending pathways in terms of mediating perceptual stability. So in summary, I said there are two distinct information channels in early vestibular processing, regular and irregular afferents, and they're context independent. It doesn't matter if motion is self-generated or externally applied. In contrast, neurons at the first central state of processing, which get input from these afferent classes, preferentially encode X afferents. Vestibular sensory responses to dynamic self-motion and the sensory consequences of gravity are canceled when there's a match with the brain's expectation. So if you have a match, you suppress. And then you just leave coding for what's unexpected. The output of the cerebellum reveals a rapid updating of an internal model as unexpected sensory inputs become expected. And this distinction between active and passive vestibular stimulations is important for ensuring both perceptual stability and accurate behavior. So I'll stop there, and I'm happy to take questions.
All right, well, thank you for an exciting and interesting talk. And so we'll open up for Q&A now, and I see we have a few questions that have already been posted. So the first question is from Byron Price, and the question is, are there any ideas about whether the irregular spiking afferents encode something different from the regular spiking? The irregular cells showed lower mutual information with stimulus, but are they just noisy?
Yeah, so the irregulars, I went through that pretty quickly. The irregular afferents actually have much more precise spike timing, and we're trying to understand what the implications of that are. So you can think of, for example, the auditory literature. If we played a sine wave in that was big enough amplitude, you would see phase locking, where you would have a very systematic, firing at a certain point in the phase of the stimulus. So what we think that this is involved in the case of the irregulars, they're more dynamic, so they're noisier, but they also have more spike timing precision. So that if you have a really dynamic stimulus, you're probably more likely to fire off a synchronous volley of these afferents. And what that's probably important for, the thought would be, is if you think about the inertia of, say, the eye plant, right? For the VOR, the eye plant doesn't have much inertia. Think about the body and what you need to do to move the body and to generate a vestibular spinal reflex, quickly and rapidly. So what we think is that the higher sensitivity and the precise spike timing is important for generating really dynamic, synchronous drives to the vestibular spinal pathways to most effectively overcome the high inertia of the body and generate the most, I guess, effective vestibular spinal stabilizing reflexes. So that's one thought for that.
All right, so the next question is by Rebecca Boom. We see this sensory attenuation for all senses, and I believe that's maybe related to predicted versus externally driven sensory input. Do you think the very basic or first part of this mechanism is the same for all senses?
Yeah, we actually did a review on this recently. Jess Brooks is an author on it as well. While all systems show this type of modulation, it's done differently across systems. So, and I should also note, I mean, I don't know if my slide is still up.
Yes.
Yeah. That it's not a trivial thing. So, for example, we see this in the vestibular only neurons, which are the ones that go to the spinal cord and the ones that go to the thalamus. We do not see this in the VOR neurons. So if anyone was really paying attention at the beginning of my talk, right, people, when you're walking down the road, you want your VOR on when you're walking, right? Because you want the world to be stable. So the VOR neurons are not turned off during active movements. They're turned off, when you want to redirect your gaze, but if you're moving through the world, you don't turn these neurons off. So again, if you look at the visual pathways, if you look at dorsolateral geniculate, the first central neurons in the visual pathways, they're not suppressed. You get very subtle effects that people have described in the magnocellular pathways. If you look at proprioceptive pathways up from the spinal cord, they're actually enhanced during active movements. So it's more complicated than all pathways show the same phenomena. There is context-dependent gating that goes on centrally, but it really actually is different strategies are used in different systems.
Okay, the next question is from Robin Mildren. So with the driving monkey experiment, and she had a question of whether that was coded as passive, do you think if you did this for long enough, this might become more similar to active? In other words, and then the follow-up is, what if the driving was controlled in some other way via, for instance, neck EMG?
Yeah, I'm really glad that Robin asked that question. So I have to say, it's the most frustrating experiment we ever did. It took a lot of time to train the animals to drive, and it was very exciting. They actually blew up our turntable initially because they were trying to drive too fast. And we thought we'd see something, and in the end we didn't. So we thought about a bit about this, and I do agree that if we had perhaps made the task a little more complicated, had to use a window or something to ensure they had better control, so that we might have seen something. I always think that I always make the analogy, the only time I have issues like banging up cars is when I rent a car, right? My own car, I feel very much like I know exactly where the car is. So I think if we had perhaps trained the animal more and gotten him to be more, I guess, tight in his control of the steering, that perhaps we would have seen a difference. So we're looking for somebody to take this on.
So I actually have a question related to, that's a more methodological question. So one of the things that I found that was really striking during your talk is you talked about this discrepancy between the range of head movements that you observed during natural behavior and what you tend to study experimentally. So I guess the general question is, do you have any sense as to how well the models you build will generalize to that range? And then in terms of how those models are identified, from a sort of system identification perspective, I often think about using, combinations of signs or things that are maybe less predictable. Do you think that has any influence on the models or how you relate input to output?
Yeah, I mean, for some of signs, we actually did some of that. We did, for the vestibular only neurons, which are the central neurons that mediate vestibular spinal pathways, we did the sum of signs, and they actually, we see even for within what looks like the standard linear range, we see a big nonlinearity where you get this math. So I didn't talk about that, but there are, some very interesting nonlinearities centrally. As far as the afferent, so one of the other projects that we do in the lab, I didn't have time to talk about, is we have working with vestibular prosthetic, and so there are clinical trials now. It's like a adaptation of a cochlear prosthetic. They're clinical trials with maybe 10 people, maybe more at this point, enrolled. So you have effectively, it's replacing canal function. So you have a three-dimensional gyro, and then a mapping function between the analog signal recorded by the gyro and a pulsing of electrodes in each of the three canals. So you project the gyro plane into the canal plane, and you do the proper mapping. So for that, what we can do, and to represent the afferents are pretty linear with some saturation. So we find the afferents we can represent with sort of a linear function fit over the linear range, and then cascade like a linear, nonlinear cascade with something that's very sigmoidal. So I rushed through the recent work we've done on walking. We really need to account for the nonlinearity. And what's kind of exciting is that's a case where our basic physiology has made it into the mapping box, right, of what these patients are currently experiencing. So it's the kind of biomimetic, at least in the dynamics of this linear, nonlinear cascade model.
Great. So I just want to be mindful. So it's 7:45 local time here. I think we should have about 5 more minutes in the room. I'll go to another couple questions, but just if people have to leave and go to another talk, I just wanted to thank everyone for attending and thank you, Kathleen, for the wonderful presentation. So we'll move on to the next question here. So this is from, I believe it's Daniil Markov. Can you speculate on possible neuronal mechanisms and pathways that result in inhibition of activity in vestibular nucleus if expectation matches reality?
If expectation matches reality.
So
when you have,
go ahead, sorry.
Yeah, so you mean, I guess, does she mean when we have a match between, so a match between what happens, so why do we have suppression? So that's one of the things that we're currently trying to understand. So presumably, so we've looked at vestibular nuclei neurons, we've looked at the output neurons at the cerebellum, and they both show this suppression. So that suggests that we're looking, if you look at the output neurons, let me just backtrack. So the vestibular nuclei are effectively output neurons of the cerebellum because Purkinje cells project both to the deep cerebellar nuclei and to the vestibular nuclei. So both in the sort of deep cerebellar nuclei proper, which would be the, say the rostral fastigial nucleus, and the vestibular nuclei, we see the same suppression. So this then bumps the problem up one level to the cerebellar cortex, and that's currently where we're looking. So Omid Zuberi, who's in our group, will be presenting, I believe on Thursday, I forgot what time, his recent work looking at what we see in the cerebellum, Purkinje cells, and trying to actually understand the computations there. So our guess is, at least part of that computation is done in the cerebellum. It seems like a logical place to be building an internal model.
And then I think this may be our last question here. So speaking of the cerebellum and internal models, Christopher Versteeg has a question. Sensory prediction errors can be caused either by a faulty motor plan or a faulty forward model, maybe a combination. How do you think the brain figures out whether to change the plan or just the sensory prediction?
Yeah, I guess the point, in our, whether to change the plan or the prediction. Yeah, that I don't know. I mean, I guess at this point we're not looking at the inputs from sort of higher levels. Like ultimately, obviously motor cortex is says generate a head movement, right? We don't know what the feedback loops are. In the cerebellum, you have different time scales going on. So, probably the cerebellum is working at one time scale, and these other loops would be working at another time scale. So I would, I guess, say what people would generally say is that perhaps by looking at the time scale of different effects, that might give us some clues. At this point, I think we're really focused on the cerebellar loop in part because if you're thinking about dynamic models that are changing within 40 movements or so, that would be where I would look. But for other types of paradigms where perhaps you also give people different types of feedback, right? Here we have the feedback to the animal is just his getting his reward or not getting his reward and the target at the end. So I think depending on how we set up the protocol, we might be able to play with these sort of different time scales.
Great. All right, so we will have to wrap up here. So thank you again, Kathleen, for an excellent talk. Thank you everybody who posed questions for for interacting here. And just as you go through the remainder of the meeting, as usual, feel free, reach out to people, meet new people, as that's sort of the purpose of this format. And so I hope to see you all around in subsequent talks. Take care.
Yep, thank you.
So as people kind of pour into the room, because it takes a little while for them to come in. Great. Okay. So welcome to this session. My name is Megan Peters, and I'm the MC for this session. I'm one of the co-organizers of NeuroMatch. And in the back end here, we also have Ishant Agarwal. I hope I pronounced that correctly. And he's going to make sure that all of the tech works well and that we have our live stream on YouTube working as well. So we have 3 great talks for you today from Robert Nowosielski.
Yeah, good job.
Sorry,
No
Jessica
problem.
O'Brien and Abby Hutton. And so why don't we just go ahead and get right into it. So I'll ask the other 2 speakers to turn off their cameras, and we will start with Robert from the University of Toronto. And he is going to tell us about age-related differences in visual vestibular integration, over time of exposure in VR simulated driving environments, or the vestibular system for short.
Okay, so let me just, we had it working. Okay.
We did.
Oh boy. Okay, can everyone see just the slides?
Yep, looks good to me.
Okay.
Here we
So,
go.
everyone.
I will hold up a, sorry, I'll hold up a 3-minute warning when you've got 3 minutes left.
Okay, perfect.
Great.
So, hello everyone. My name is Robert Novoselsky, and I am a PhD candidate from the University of Toronto. Thank you for giving me an opportunity to share my PhD work with you, titled Age-Related Differences in Visual Vestibular Integration Over Time of Exposure in VR Simulated Driving Environments. It's quite the mouthful. So, it's a beautiful sunny afternoon as you drive your car up to the cottage. Captivated by the rugged Canadian landscape, you miss a sign telling you to reduce your speed. Suddenly, you encounter a valley and you get a sinking feeling as you reactively reduce your speed. You are driving too fast. But how is it that your brain knew? So this feeling is in part caused by the vestibular system. It's one of the many sensory systems that allows us to judge our own movements while driving. The vestibular system are organs in the inner ear that detect acceleration using hair cells, and along with vision and other sensory inputs allows us to perceive our own movements through space. Self-motion perception while driving is dependent on the optimal combination and integration of relevant multisensory information. However, of all the sensory inputs that have been studied in the context of driving, vestibular cues are amongst the least studied. Like all of our sensory systems, vestibular functioning, processing, and perception have been known to change throughout the lifespan, with adults over the age of 60 exhibiting an estimated 25 to 40% reduction in vestibular hair cells compared to younger adults. This leads to changes in behavior in the context of balance and mobility, such as slower gait speeds, as well as an increased occurrence of negative events, such as falls. Furthermore, age-related changes to vestibular functioning have been found to lead to abnormal gait stabilization and increased perceptual thresholds for acceleration. But it is not clear to what degree these changes affect driving performance and safety. There are also age-related changes to the way we integrate information across the sensory systems. Specifically, older adults demonstrate larger performance gains compared to younger adults on tasks where information is presented via more than one sensory input, for example, seeing and hearing an object, compared to just one sensory input alone, for example, seeing an object. This heightened multisensory integration may be a compensatory mechanism for reduced acuity across the sensory systems with age. Furthermore, while older adults exhibit more errors and greater variability in performance when learning new tasks than younger adults, they can improve at a greater rate than younger adults over time of exposure. This suggests that older adults may demonstrate greater improvements as they adapt to a simulated driver task, but it is not clear what the magnitude of this improvement can be. Therefore, it is important to study vestibular function in the context of driving in order to understand how these changes can influence driver safety and performance, especially now, as adults over the age of 65 are quickly becoming one of Canada's largest demographic groups, making up to 20% of the population within the next 10 years. Maintaining mobility in older age is critical, as over 55% of adults over the age of 80 continue to drive. However, older adults have a 3 times higher likelihood of being involved in a collision, and it's important to understand the role that changing vestibular functioning plays into this. Therefore, in order to understand how age-related changes in vestibular functioning affect driving performance, we have 2 main objectives. The first, how does the addition of different kinds of motion affect older and younger adults differently in a driving task? And 2, how does driving performance change over time of exposure to the driving task in younger and older adults? We recruited 34 healthy younger adults aged 18 to 35 and 30 healthy older adults aged 65 and above, with no diagnosed vestibular disorders. All participants went through a series of baseline sensory tests before going to the experimental procedures in order to make sure that, that there were no differences in vestibular functioning between groups. This experiment was conducted at Kite's Driver Lab, pictured above, which consists of a real car surrounded by 360-degree projection screens on top of a turntable, placed on a 6 degree of freedom motion platform. This allows for the ability to stimulate the vestibular system precisely and systematically in a highly controlled manner. I programmed and designed 3 15-minute drives that participants drove through while following the rules of the road. Each drive was comprised of rural, urban, and suburban environments, which included curves, turns, hills, and intersection, and these elements can be seen in the slide above. We collected data on mean speed, speed variability, lane positioning, lane departures, and steering revolutions. The reason why we chose these measures is because they provide information on how well or how poorly participants drove. For example, greater speed variability would reflect poorer driving performance. Participants were then randomly assigned into one of 3 groups based on the type of motion they were exposed to. And the first condition is the fixed base condition, where participants did not experience any kind of motion. As you can see here, the vehicle does not move as it enters the turn. The second condition was the turntable condition. Participants were exposed to rotational motion, which provides information about turns and curves in response to steering behaviors. As you can see here, the vehicle rotates as it moves through the curve. The third condition was the full motion condition, which, in addition to rotational motion, provides information about forward, backwards, and up-down acceleration and deceleration. In this video, we can imagine the vehicle moving through the same turn, but the motion platform adds a more compelling and realistic sense of motion. For our procedure, we had everyone first drive through our fixed base condition before moving on to their respective motion conditions for the second and third drive, allowing us to conduct analyses on the effect of adding motion, as well as how performance changes over time of exposure, using both between subjects and within subjects analyses where necessary. We had 3 hypotheses in this research. The first is that younger adults will have better overall driving performance than older adults. The second, there will be an additive and beneficial effect of motion on driving performance, going from no motion to rotational motion and no motion to full motion, with older adults demonstrating larger improvements in driving performance. And the third is that older adults will demonstrate a larger improvement in driving performance than younger adults over time of exposure. Before analyzing our driving performance metrics, we wanted to make sure that younger and older adults did not differ in their baseline sensory data between conditions. We performed 2 additional sensory tests measuring vestibular functioning in order to provide evidence to suggest that older adults did in fact have age-related changes to vestibular functioning. Our posturography data, which can be seen in the top left of the screen, measured the stability of one's posture. It was found that older adults had less stable posture overall, with both their eyes open and with their eyes closed. Our analysis of the dynamic visual acuity test, which assesses the vestibular ocular response during perturbed head movement, found that older adults had greater declines in visual acuity during head movements horizontally and vertically. This suggests a poor vestibular ocular reflex, which is highly reliant on vestibular functioning. So our main objective was to investigate whether the addition of motion, with addition of different kinds of motion, affect driving performance, and whether these effects are different in younger and older adults. Every participant drove in the fixed base condition in drive 1, and then in their respective motion condition in drive 2, with the fixed base group acting as a comparative control. Our findings demonstrate 2 important trends. The first is that the addition of motion generally improved driving performance, or in the case of the 2 graphs on the left, lowered speed variability for both older and younger adults in the turntable and in the full motion group. This was, however, not the case for the fixed base group, which can be seen on the right. For our second observed trend, we found that the addition of motion affected younger and older adults differently, with older adults benefiting more from the addition of motion than did younger adults. However, this depended on the type of motion being added and exhibited greater relative improvements for speed variability, lane departures, and steering revolutions. While in the case of full motion, older adults exhibited greater relative improvements in lane departures and steering revolution. And both of these cases illustrate that older adults benefited more from the addition of motion than did younger adults. Our final objective was to determine how driving performance changes over time in older and younger adults. Broadly speaking, we found that younger and older adults demonstrate what can be described as improvements in driving performance over time of exposure, regardless of motion condition, with older adults generally improving more drastically than younger adults. This can be observed in the graphs above, where older participants had higher speed variability and speed revolutions and lower mean speed in their first drive, but then improved their performance for each drive over the course of the experiment. So we can see a greater relative decrease in speed variability on the two graphs on the left and an increase in mean speed on the graphs on the right, all of which suggests an improvement in driving performance. We found a main effect of age, exposure, and interactions between age and exposure for each of our dependent variables. As hypothesized, older adults exhibited poor performance on their first drive, but demonstrated larger improvements in performance over time. We had two objectives in conducting this research, and we were able to answer both questions. With regards to how the addition of different kinds of motion affect older and younger adults differently, we found that motion leads to better performance overall in younger and older adults, but that older adults demonstrate a greater improvement than younger adults. This finding is consistent with the literature, where older adults experience a multisensory enhancement. With regards to how does driving performance change over time of exposure, we found that older adults exhibit greater improvements in driving performance over time, despite driving more poorly overall. These results are also consistent with the literature. But this research has some important limitations, however. With regards to the effect of adding motion, we do not know if this improvement in performance was a result of adding motion cues specifically or motion cues generally. Future studies can address this by utilizing a within subjects design where each participant experiences all three motion conditions. With regards to the effect of exposure, participants all began driving in the fixed base condition before moving to their respective condition. So there's a confound of motion. There couldn't be a confound of motion despite it not being significant. Future studies can utilize a fully between subjects design where participants only drive in their respective motion condition in order to see how motion and exposure interact. So I'd like to thank you all for your time. I'd like to give a special thanks to my co-advisors, Dr. Bruce Haycock and Dr. Beiran Keshevars, and my primary advisor, Dr. Jennifer Campos. Also to my research assistants, Catherine and Gayatri, and to Dr. Jeff Fernie for funding this research with a CIHR impact grant. Thank you very much.
Fabulous. Thank you, Robert. And because we cannot applaud in Zoom, please drop a word of praise in the chat if you enjoyed the talk. So we have time for a couple questions here.
Should I stop sharing?
Sorry?
Should I stop
sharing? That's good.
Okay.
Yeah, great.
Perfect.
So the first question is from Carly Sombrick. And the question is, do you think that older adults' slower improvement is related to general learning impairments?
Yeah, so we do find evidence for older adults having learning impairments, and that what we might actually be seeing is a very quick adaptation to the simulator in younger adults. So they might be adapting very, very quickly. While in the case of older adults, because of these impairments, we're seeing a larger trend, but it might just be that this is a delayed learning effect. So that might be one of the ways that we could interpret this research.
Great, thanks. Do we have any other questions for Robert before we move on? Can I ask a quick question if we don't have any? I was wondering, you have these measures of vestibular precision and ocular precision.
Yes.
And normally in a multisensory case, you might have, you might do kind of a Dora Angelaki experiment where you kind of like have like an optic flow and you move the person through on like a kind of platform. And then you can get a measure of like the actual like optimal or suboptimal cue combination, like an Ernst and Banks kind of approach. So do you have any plans to look at how that kind of mathematical approach might interact with your more realistic naturalistic tests?
Yeah, I definitely think like these are dynamic visual acuity and posturography. They're really proxies for vestibular functioning. We don't really know. They could only suggest it. So perhaps by adapting more sophisticated models or a greater battery of tests to really look into vestibular functioning, will we be able to make these changes? Because it's not just vestibular functioning, like older adults experience changes all across. So the degree to which we could really point to vestibular functioning as the driver of this effect still needs to be worked out a little bit. Like this is still the first research we've done using this paradigm. So we really expect that this will evolve and develop as we continue researching this.
Cool. I'd love to talk to you more about that, actually.
Yeah, me too. Thank you.
So, well, we have, we're setting up for the next talk. Jessica, why don't you go ahead and get set up? There is one more question from Carly Sombrick. Do younger adults with no motion act similarly to older with motion? And so, Jessica, as you get set up, let's answer this question.
So what we found was that in the no motion condition, the driving metrics were similar. Younger adults still generally drove better, but we really find these bigger differences in interactions in the two motion conditions. So yes, there was a slight, but it wasn't necessarily for all of them. For example, with our speed variability measures, we found that there's no difference between the two. However, we had so many variables and so many ways of looking at them that I really only wanted to highlight the ones that really exemplify these effects.
Great, thank you. If other questions come in, you can feel free to answer them in the Q&A box directly. But for now, let's move on. Let's thank Robert for the great talk, and let's move on to Jessica O'Brien, who will be talking about, she's from the University College Cork, and she will be talking about audiovisual training in older adults, 2 interval force choice tasks, improves performance. So please, Jessica, when you are ready, take it away.
Perfect. Thank you, Megan. So hi, everyone. My name is Jessica, and I'm calling here from Ireland, where I am doing my PhD in cognitive psychology. And today I'd like to share just one piece of research I've done recently, which is audiovisual training in older adults. So my research area, kind of similar to Robert, really focuses on multisensory integration, which of course is the brain's ability to put together information from different senses into a kind of a coherent perception of the world around us. And when we talk about multisensory integration, we often talk about a temporal binding window or a window of integration. And this is just, I suppose, a window within the brain during which the brain will integrate information from different senses because they probably come from the same source. And I just really like the example of lightning, which is why I have the image here on screen. So of course we see the lightning flash before we hear the thunder. So this window of integration allows for that time lag between the different times that sensory information kind of reaches the brain. The interesting thing with, I suppose, multisensory integration, and kind of Robert touched on this as well, is that as we age, I guess, the way we integrate changes, and we find that the window of integration is actually enlarged in older adults, so they integrate more extensively. So that means they have a wider window, and so they're possibly putting together information that may not be coming from the same source, so they're integrating a wider across time. We also find that older adults show kind of greater susceptibility to audio-visual illusions. And this greater susceptibility is actually linked to, I suppose, cognitive and functional conditions and decline, such as MCI and balance and falls. So I suppose the question for us as kind of applied researchers is, can we do anything about that? Can we train multisensory integration if it is or becomes inefficient as we age? And there is some kind of promising good news. So Powers and colleagues back in 2009 was kind of one of the first to come up with perceptual training for multisensory processing, particularly audio-visual integration. And Powers has shown kind of 2 different versions. So we have the 2 IFC, which is interval force choice task, and the 2 AFC, which is alternate force choice task. And both kind of play on simultaneity judgment and I suppose test someone's multisensory perception in terms of the temporal domain, so of audio and visual information. Using a kind of a feedback training of this task, Powers was actually able to train young adults to improve after just 5 days of training. So it's about 30 to 40 minutes training each day, and they actually improved on the task. So it is possible to alter this particular process in the brain and to improve it. And Powers actually also found that it generalized to an audio-visual illusion called the sound-induced flash illusion, or SIFI. And now, while it didn't actually reduce, these are young adults, I should mention, it didn't reduce the amount of illusions they saw, but they did improve their visual temporal acuity on the task. So it generalized beyond just the 2 IFC, it actually went to a different perceptual task, which is kind of important because we don't just want to improve just one task, we want to improve actual multisensory processing itself. There's been a few variations since Powers' initial study. So we've had a AFC staircase done by Sethi and colleagues back in 2014. And again, this was 5 days, but importantly, this was with older adults. So this took a kind of a similar procedure to Powers and found that this was effective in older adults. And they actually also found a generalizability, to a reduced susceptibility to the CFI illusion that I just mentioned a second ago. So that was kind of a big deal. And that was, again, just 5 days was all that was necessary to see improvement. Then more recently, only actually earlier this year, Thieves and colleagues used the MEG or the MEG, and with 1 day of training in younger people, they found that was enough to improve on 2IFC. So that exact same training or task that was used by Powers. And they actually looked, I suppose, at the neural underpinnings and found increased beta band amplitude with that improved training. Where my study fits in here is where I'm taking the original training that Powers used back in 2009 and 2016 and taking it not with young adults, but straight into older adults. And I tried to see if I could reduce the time a little bit. So to make it 3 days as opposed to 5 days, because I guess 5 days is quite, it's a lot to ask of an older person in particular. So Powers used it with younger people, and with older people asked them to sit, at a computer for like an hour, over an hour, 3 days or 5 days in a row is quite a big ask. And we wondered if it was, be more feasible to have a much shorter training. So we took exactly Powers' task and training and did it with older adults. As I said, with 3 days. We included a young comparison group simply because we changed it up so much. It wasn't 5 days, it's now 3 days. We had to make sure the 3 days was effective in younger as well as older adults. And the last thing I should mention, I suppose, is we chose 3 days because in those past studies, that Powers studies, that SETI study, 3 days was sufficient to get kind of the maximum benefit. And the kind of day 4 and 5 of the training was quite minimal benefit for the amount of time the participant was spending. So we thought, can we kind of tailor this down to just 3 days for training? Now, of course, sorry, I'm just skipping ahead. We expected, I suppose, similar results to Powers, that both the younger and the older should train after 3 days of training, and we hoped and expected to reduce susceptibility to the illusion, just like SETI found. Our participants, so I had an older adult group, which were 23 older adults. They were your typical healthy, neurotypical adults living in the community. I just note here at the last bullet point there that we had 11 fall-prone individuals of those 23. And I guess the reason I included those is my PhD is kind of broadly on multisensory integration, but particularly in fallers. But actually we decided to throw the fallers in amongst the kind of put the fallers and non-fallers together, because their baseline kind of pre-test or pre-training performance was very similar to normal non-falling adults. So we put them together for the purpose of this particular research. And our young adult group is your typical university students that kind of get roped into or cornered into helping out with these projects. So the age range of 18 to 35. The task itself is exactly the one that Powers used back in 2009, 2016. So if you just have a look at the top row there, so it's a 2IFC, so a 2-interval forced choice task. And the top one is the pre-test, I should say, and post-test. So what you have is you have 2 pairs of stimuli, a flash and a beep, or a circular kind of a white circle, as you can see there. And one of the 2 pairs, the flash and the beep, are simultaneous, so they occur at exactly the same time. And the other pair, there's a lag. And if you just look at the bottom, the lag, varies by SOA, so stimulus onset asynchrony. So it could be 50 milliseconds or all the way up to 300 milliseconds between that initial flash and the beep. And what we're doing here is really testing people's temporal window of integration. So if your window of integration is very small, maybe at 100 milliseconds, you're not seeing, or you're well able to discriminate. But for others, it could be at the 300 milliseconds. So we expect better accuracy if it's outside of your window of integration, so you won't integrate. On the bottom row here then is the training, which again is exactly the same as the pre and post test, except you have feedback at the end. So once the participants respond, so they actually verbalize the response. So they say either the first or the second pair, and then myself presses 1 or 2 on the computer. And then they get a smiley face if they're right, or a sad blue face if they're wrong. And this is after every single trial. So it's immediate feedback they're getting. And that's the training that Powers used, and also Thieves more recently. And then we come to the CFI. So the sound-induced flash illusion was included as another perceptual measure because we wanted to see, could we generalize the training, or does training generalize beyond just task-specific training or improvement. So we included the CFI, and for anyone that doesn't know this, it's kind of does what it says on the tin. So it's a visual illusion induced by sound. So what happens if you're presented with a flash and a beep that occur at the same time, and then if I present another beep at you quite like in quick succession, so a couple of milliseconds after, you'll actually experience a second flash. So it's an illusion as such. You see an illusory flash that isn't there. And that just means that your brain is working, so it's integrating. But for older adults, the sound-induced flash illusion, they're more susceptible at longer SOAs. So even when that second beep is very far away from the initial pair, they're still seeing an illusion as such, which is kind of an indicator of maybe not so efficient multisensory processing, an indicator of a very large temporal binding window. Now, I'm not sure if this will work, but I'll give it a go. So this is a little video of the sound-induced flash illusion. So hopefully we see it. Not the end of the world if it doesn't work. Not sure if the sound will work on this, but what you should see is flashes here and beeps. And every single time there's actually one flash. But at the start, you should probably see two flashes because your brain is integrating. But as the video goes on a little bit, you should be able to discriminate there's only one flash, but two beeps. So I'll just try that there now. Okay, so it's a fairly tricky one. So so it is quite hard, as you can see there, to actually judge how many flashes are happening because the beeps, you know, are kind of throwing your brain off, I suppose. So the CFI has really come into prominence as about 20 years of research on it now, I suppose in 2020, and it's just emerging as a very good indicator of a person's multisensory processing. Just to note here, having the side that D prime is sort of what we use as the outcome variable. So we do look at proportion of correct, so you know how many illusions they're experiencing, but the D prime is just a more sensitive measure, so it takes into account on this task how well you perceive the actual stimuli. So it takes into account your two flashes and two beep conditions. So are you perceiving two flashes when there actually is two flashes? And your false alarms. So are you seeing that additional flash when in fact it isn't there? Our procedure then was 3 days, as I said, of training. So we had our pre-test measures with the illusion in green there and your task. And then you have the training, which is just the task with feedback. Day 2, you just have training. Day 3, you have your training again and back to your post-test measures. So what did we find? Well, if we look here, this is actually the training. So the day 1, 2, and 3. And we might just look at the young adults first, so over on your right-hand side there. And along our y-axis there, we have the mean proportion of correct responses on the training. So you can see the green line is 50%, so that's chance. And remember, participants are forced to respond, so they have to guess even if they don't know. So there will be an amount of guessing, I guess. But if you look here at, I don't know if you can see my cursor now, but if you look here at the young adults, so at 50 milliseconds, they're integrating quite a bit. So they're close to 50% accuracy. So this is almost pure guessing, right? Which is what we expect. So the brain is integrating correctly at 50 milliseconds. But as the SOAs get longer, this is kind of the curve we'd expect. Participants, the younger adults, are getting better at discriminating which of the stimulus pair occurred simultaneously. So they're almost at 80% there by 300 milliseconds. In comparison, look at our older adults. So even after day 3, which is our gray line here, they're at 60% accuracy. So there's still a fierce amount of guessing going on, I suppose, even after 3 days of a training. So that's just giving an overview of how they trained. And then let's look at the actual results, so the stats, I guess. So the TWI is our temporal window of integration, and this is on the task. So this is the task without feedback. So this is us testing pre and post. And if we just have a look at the young first, I suppose. So over here is the size of their window, or the average size of it. So this is actually each of the participants here. So it's 20 young adults here. And you can see in general, post-training, the window is reduced, which is exactly what we're hoping to do. So they're integrating less extensively and they have a narrow window. And this is a significant result. And also the older adults. So you see that's a bit more variety here. So some are unfortunately getting worse. So their temporal window or integration is getting larger post-training. But on the whole, so most of them are also having that narrower window after training. And if you just have a look, the older go from 56% accuracy, so very close to chance, so very close to guessing, to 62%. on average, and the younger go from 73 to 76%. So this replicates the results of Powers and of SETI, and with just 3 days was effective in both young and, more importantly, in the older adults. Next, we go to our CFI. So I guess this is a really important one because the CFI is an indicator of their multisensory processing efficiency. So could we generalize the training to this particular perceptual illusion? If you just have a look on the left there, so the y-axis, we have D prime. So as I said, the perceptual sensitivity score. And I guess what we want here is high scores. So high scores up here, so the closer to 4 they are, the better their performance on the CFI. So what we're seeing, we'll just have a look at the young. The young is always easier to look at and make sense of. So you're seeing here, we have our D prime, and then they're heading up. So they're all getting quite better, okay? So their performance is much better post-training. But unfortunately, this actually doesn't reach significance. It's only a trend. In comparison, our older adults are actually showing the reverse. So the opposite of what we expected. Their sensitivity is getting worse. They're experiencing more illusions. We weren't quite sure of the reason for this. This isn't what we expected and not in line with past research. But we presume that part of that must be possibly due to fatigue, because unfortunately we didn't counterbalance our tasks. So the CFI was always the third. So the last task they did after day 3 of training, and perhaps a tension or fatigue is setting in and leading to this. Unfortunately, the reverse of what we were hoping to find. Again, it's approaching significance, but not quite there. So these are trends. So I suppose the good news is that they do train. So older adults, just like younger adults, train, and we find a narrower temporal binding window post-training, just as in those previous studies, but with older adults. So we suppose we extend the research with older adults, and 3 days was sufficient to get that. Unfortunately, we don't find the generalization to the CFI task like was found a few years ago. And I suppose the limitations just might explain possibly the reason that we don't find that. One is the range of SOAs we chose. So perhaps the SOAs were too short, that we didn't capture, I suppose, the extent of older adults' binding window, because we capped it at 300. And actually, more recent research has shown that older adults can go almost as up to 400 milliseconds. Their window can be extended to. So perhaps we missed that, and the task was a little too difficult. And unfortunately, no objective measure of sensory acuity. So while on the CFI task, we asked them about, we presented trials that were just 2 beeps or 1 flash. So we got their unisensory conditions. We didn't get any objective measure of visual acuity, say pre or post test, which may again have accounted, like perhaps we had participants that had very poor acuity, but we weren't able to discern that. So I guess our research is just adding to a kind of mounting evidence that perceptual training is emerging as a novel intervention for populations that might have underlying multisensory processing impairments, such as dyslexia, autism, or, as I said, older adults with falls. So I'm going to wrap up there. Thank you very much.
Great, thank you, Jessica. And unfortunately, we are way out of time, so we will go straight to our next talk, but there are some questions in the Q&A and in the chat, so you can feel free to ask them or answer them there, okay? Thank you, Jessica, for a great talk. I've got a question for you too, but we'll save it till later. So we're going to go straight to our last speaker of the session now, who is Abby Hutton from Wichita State University. And Abby is going to tell us about associations of pickleball play with cognition among older adults. And this is a longitudinal study. Great. All right. Take it away whenever you are ready, Abby.
Okay, I just need to get this working. Oops. All right. Okay, you can see the wrong one now, right?
Correct, yeah.
Is this the right one?
That is the right one, yep.
I don't know how to make it bigger than that.
It looks fine to us.
Okay, all right.
Yeah, go for it.
So yeah, thank you for that introduction there. I am pursuing a PhD in human factor psychology at Wichita State. And today, like she said, I will be talking about the associations of pickleball playing with cognition among older adults. And this was specifically our 6-month longitudinal study. So the amount of older adults is going to increase by 2030. And it is said that older adults will make up 25% of driver fatalities. Cognitive functioning declines with age, and it is necessary for tasks such as driving. Driving uses many visual functions, including working memory, selective attention, processing speed, decision-making, and many others. So it is difficult to perform tasks while driving if those decline. The useful view task, or UFOV, examines 3 factors, including processing speed, divided attention, and selective attention. In a study done by Osley et al. In 1998, they examined people between the ages of 55 and 87 and found that overall, older adults who had a 40% or more decline in their ability of the UFOV task are about 2 times more likely to be involved in a car accident. Additionally, inhibition declines throughout the aging process as well. Inhibiting is the process of dismissing irrelevant information while responding to a target. So when distractors arise, older adults are more likely to pay attention to them, which could lead to accidents or other types of accidents if you can't inhibit. Cognitive functioning affects everyday life of older adults, and if not maintained, it can be detrimental to, due to vehicle accidents or falls or things of that matter that could injure them. So aerobic exercise has been studied in previous literature to help maximize the quality of life cognitively and physically among older adults. So previous studies have shown that 6 months of consistent aerobic exercise training, such as walking or running, can help maintain cognitive functioning, which can lead to enhanced driving performance, not only cognitively, but try to enhance driving performance. So here's a list of some different studies that have happened in the last about 15 years or so with aerobic training. And so why has aerobic exercise, been able to enhance driving performance or just in general cognition? So a study showed exercise can increase these levels of oxygen to the brain. So this exercise, they had 2 groups. One was an exercise group and one was a control group. And in the exercise group, they increased 16% of amount of oxygen to the brain, and the control group only increased 5.3%. And so as we age, cognitive function declines, but with the help of exercise, this process can be slowed down or prevented. Overall, the most prominent improvements in cognition have been from longitudinal training studies. So with this in mind, aerobic exercise has been studied the most, but there is some literature studying racket sports such as like tennis and badminton that can potentially have the similar results as aerobic exercise because of the similar components that it has. So one to talk about is tennis. So some literature by Marks in 2006 shows that older adults who have participated in tennis regularly have positive health benefits. So an increase in oxygen to the brain can help players' cognitive abilities. And this study found that older adults who play tennis had quicker reaction times. So older adults who engage in tennis can possibly maintain their cognitive functioning, but definitely more research is necessary. Another racket sport, badminton, a study showed that the players have, they have to use their visual functioning to be successful. So implications can be made that badminton players had better visual capabilities than non-athlete controls. So even though they did not study in this specific study, the Jafar Indifer et al. 2012, I probably butchered that, but they studied, they only studied younger adults, but it can definitely be translated to older adults just because these visual functions are needed in general to play this sport no matter your age. So racket sports are different from aerobic exercise because they involve a more lateral movement. So like where swimming and walking and running do not. So these are known as open skilled exercise. So these kind of, they're always they kind of create the flexible environment for the athlete, to have to constantly be, knowing their environment, where's the ball at, different, it just takes a little bit more of an unpredictable environment than maybe running or swimming would. So for the current study, we're examining the effects of pickleball and how that has on, what that has on cognition. So pickleball is one of the most rapidly growing sports in the US, specifically among older adults. And it is recommended for older adults because it's easy to learn, does not involve a lot of running, burst of running. It just kind of involves that lateral movement side to side because of the smaller court. And there's a lot of social interaction involved in that. But there is still that competitive factor that maybe previous tennis players are missing out on. So it is additionally is classified as a moderate in intensity. So it's still, getting at that high heart rate and that kind of thing where, running and swimming might, take over there, but it still is having those components. So a study with 15 participants who engaged in pickleball playing twice a week for 6 weeks resulted in similar health benefits to traditional aerobic exercise. And that was done by Smith et al. In 2018. So training had health benefits, including blood pressure and cardiorespiratory fitness. So it looks like pickleball playing can increase oxygen levels to the brain and increase cardiorespiratory health, and these can be associated with healthy aging and produce similar results to traditional aerobic exercise. So lastly, recent literature made the connection between pickleball and improved self-esteem and life satisfaction. So this study resulted in retired older adults who engaged in pickleball had a higher score on the satisfaction with life scale. So overall, previous studies examined pickleball are missing the visual information processing and cognition part of it. This lack of previous literature is why pickleball was chosen for our study. So like I mentioned, this is an attractive option for older adults because of their decline in speed and muscle, because racket sports like tennis are just too intense. But pickleball can be an easy sport for them to learn. So with that, it's hypothesized that older adults who engage in regular physical activity will demonstrate superior performance on cognitive and balance measures compared to physically inactive older adults over time. And pickleball players will show a greater performance benefit compared to aerobic exercisers. Additionally, novice pickleballs are hypothesized to improve on measures of cognition and balance. So why did we pick this? Just kind of like the reasons I just went over. It's an open skilled exercise. It's easy to learn. There's lateral movements. It's still considered a moderate in intensity activity for them, and it's still fast paced, has that competitive aspect to it. And there's just a lack of research so far with how it affects cognition. So a non-experimental longitudinal study was launched. 83 participants were put into groups based on their exercise characteristics. So we have advanced pickleball who have played more than a year, like longer than a year. Then we had novice pickleball who have played less than a year, so anywhere from a few months to a year. And then aerobic exercisers, and then inactive controls. And you can see here how many we had in each group, relatively similar group sizes. Okay, and next we did some different assessments. So we had 4 standard assessments and 4 cognitive assessments. And today, obviously with time, I will not have enough time to go over the standard assessments, but definitely I will go over each of the cognitive assessments. And I would like to mention that the multiple object tracking was in VR, and so it was the useful field of view task as well, but the other 2 were on a PC. So now the results. So a linear mixed modeling approach was used in RStudio to analyze the relationship between accuracy and exercise for MOT. The model that fit the best included the random effects of subjects, and the fixed effects were group, age, and session. So session being pre and post. And so what we noticed was the aerobic group were more accurate overall than relative to the overall mean. And so then the inactive group, like we hypothesized, was less accurate overall. And so we also found a significant main effect of group and age, or like that interaction between group and age. But we did find some interesting results where older participants in the aerobic and advanced pickleball groups were less accurate. So that was a little interesting, and maybe the MOT might not be sensitive enough. But a little discussion about that is that the aerobic actually had an average of 16.3 years of being active, like generally active. And so due to their many years of being active, their visual functioning, such as depth perception, motion perception, and divided attention may be more intact after playing, different sports, different exercises all these years. Additionally, the interactive group, like I said, performed poorly on average. And the reason we may not have found the pickleball groups performing like the aerobic group is that these visual functions may take several consecutive years in multiple methods of exercising. And so, we had a lot of aerobic exercises. I think I noted in the limitations later that we had, I think it was 6 different modes of type of exercising for the aerobic, whereas the pickleball usually just played pickleball. So that could be, having multiple methods of exercising could definitely have, made their visual functions for the MOT more intact. So next on the UFOV, again, we used the linear mixed modeling approach. The model that best fit included the random effects of subjects and the fixed effects for group, age, and session. Here we had, they had aerobic group and the advanced pickleball group had quicker processing speeds than the inactive group had, slower processing speeds on the UFOV task. So like our previous literature, that kind of is supported by that, and that could lead to the inactive having more driving accidents, because I can't remember the study off the top of my head right now, but the study that did the UFOV that I'd mentioned earlier said that, who have poor performance on UFOV could potentially result in more accidents, vehicle accidents. Okay, and then we had an interaction between group and age, where the advanced pickleball groups, so as they were older participants had, let's see, slower reaction times, right? I just lost my notes. Older participants in the advanced actually had faster processing times. So they had faster processing time, which could lead to, you know, that same idea of, you know, longer active lifestyles. They, you know, have more intact visual functioning and more intact processing speed in general. So again, they had, you know, 16.3 years, they had long active lifestyles. And additionally, the aerobic or the advanced pickleball players who were older were playing on average 4 times a week on average. So they were playing a lot during the week, whereas novice pickleballs were only playing a couple times a week. Okay, so next is the flanker task. So same thing here, the same model fit similarly to the previous ones. The aerobic group and the inactive group again performed as they have on the other tasks. The aerobic group was quicker with their reaction time, and the inactive group was slower on average with their reaction time. And then there was an interaction between group and session where the advanced pickleball players had quicker reaction time at the post-test, and the aerobic group had quicker reaction time at the post-test as well, whereas the inactive group had slower at the post-test. So they were getting slower from pre to post, whereas the advanced pickleball and the aerobic were getting quicker. The aerobic group, yeah, they had quicker RT. And so on average, the advanced pickleball players and aerobic had quicker reaction times, whereas the inactive were slower. So participants were playing about 4 times a week in the advanced pickleball players, whereas again, the novice only played 2 times a week. So that might be why we didn't see that result for the novice players. But it looks like playing pickleball more times a week can increase comfort level and familiarity with an unpredictable environment, which could be a factor in why their reaction time is quicker. Okay, so the last one, the in-back. So the model that best fit here was the same as the previous with the fixed effects were group, level, and or session. And so the aerobic group was more accurate on average. And between the interaction between group and session, the advanced pickleball and the aerobic pickleball were less accurate at the pre-test, so they were more accurate on the post-test, so they got better as they went from pre to post. And so the results of the in-back had mixed support for our hypothesis as the other 3 tasks. In general, the aerobic group was more accurate. This could explain that aerobic exercise can strengthen working memory more than pickleball can, or that the inactive is not sensitive, or the in-back is not sensitive enough to assess changes in working memory in individuals who have not been exercising for more than several consecutive years. So we might see some, we're currently investigating a second post-test, so we might see that the in-back could be a little bit more, could pick up more of those changes between groups. So here again, like I've mentioned throughout the study or throughout the presentation, is that some limitations. It was an observational study. In the pre-test, we did it in the summer. In the post-test, we did it in the winter. So that could kind of change the way they play, even though there are some, you know, indoor courts for pickleball and indoor gyms and such like that. And like I said, we had 6 different aerobic exercising methods, which could be one of the reasons why the aerobic were kind of performing better on each task. We had some different medical conditions, but relatively most people were healthy. And then self-report of duration, frequency, and total involvement in exercise in general. So that could be a little bit of a limitation. So overall, cognitive aging is unavoidable, but there are many ways to maintain the quality of life. The current study suggests that pickleball may take more time to show cognitive changes in older adults, but regular physical activity leads to better performance on measures of cognition compared to inactive older adults. It is possible that our follow-up that we do, which we are currently studying right now for currently collecting data and should be done by the end of the year, will show more you know, differences between groups, and you know, these tests that we have, these assessments may not be sensitive enough to pick up them. So hopefully with this, you know, COVID has pushed that back, but hopefully with this second post test, we will be able to find some more differences and maybe, you know, more analyzing more of the in-depth factors that we have, you know, about the times a week they play, the different sports, which is all currently underway right now. So as the population of older adults grows, maximizing their quality of life in every aspect is important. And a game of pickleball could potentially be part of that solution. Thank you.
Great. Thank you, Abby, for a great talk. We have a very, very short amount of time before, well, now at 9:50, we're going to get kicked out of this room soon. But if anybody has any burning questions, please stick them in the Q&A. Otherwise, I would like to invite all of our speakers to turn on their cameras again and wave and say thank you very much to all of our speakers for a great session. And if you want to connect with any of the speakers, of course, you know where to find them. You can, I'm sure everybody is Googleable, or maybe you will even get matched with them via the NeuroMatch algorithm. So I want to thank all of our speakers once again for a great series of talks, and I'll see you guys next time. Okay.
Thanks so much.
Thank
Bye-bye.
Bye.
Methods on how we interpret the results that we look at in neuroscience. And so as a reminder, this is a traditional talk, so each presenter will have 12 minutes to present, and then you're all welcome to ask questions for 3 minutes. To ask a question, you can use the Q&A panel, and also remember to upvote your favorite questions because we'll answer them, we'll ask them in order based on how many upvotes they have. You're also welcome to use the chat to share thoughts and related information to the talk. And so with that, I'll introduce our first speaker, Eric Bridgeford. And so Eric is a graduate researcher in the Department of Biostatistics at Johns Hopkins University in the lab of Joshua Vogelstein. His talk is on batch effects or causal effects applications in human functional connectomes. And with that, I'll let Eric start.
Great, thanks. I appreciate the introduction, Stephen. So to start us off, I will discuss a little example here. So let's say we have N participants and we measure functional connectivity. Let's say pairwise correlation between the ROI time series in this case. My goal is to effectively address whether or not, at specific edges, is there a difference between the FC of males and the FC of females? So let's say I just do an associational analysis, maybe a linear regression at each edge, and I identify which edges are significant after some sort of an adjustment for the many comparisons that I have here, like Bonferroni correction. I find I have 99 edges that are significant in FC after multiple comparisons, and I go in and report my finding and ship my paper off to bioRxiv. Now some other investigator comes in, and let's say he gets a hold of a new group of people, takes a look at the data, and finds a new subset of edges. And kind of the problem that arises is that some of the edges are similar and agree between the two analyses, but a lot of the edges are a lot different than the ones I had originally found. And so sort of the question is, which edges are the actual signal edges? What went wrong in this case? And so in this study, there are effectively two problems with making this comparison. So the first problem is when somebody tells you, that I have a difference between two data sets, your mind should immediately jump to, tell me something about the study population. Were the study populations similar? How did the study population differ? And the problem with the local associational study is that we're not really seeing the full picture here. My inference is going to apply well enough to a population identical to the population I actually, measured FC for. But then when I try to go and generalize that to, whatever my target population is, or a new target population, that becomes a lot harder. And so basically the problem here was in data sets 1 and data set 2, the issue that we had was that the two data sets differed, by an order of magnitude, basically, in the age, sex, and racial composition of the individuals, as well as a number of other factors, which we basically term our signal producing covariates. And so in this case in particular, data sets 1 and data sets 2, as you can see, the sort of age regions of the individuals, or the age ranges, are just totally disparate between the two data sets. And I can subvert this problem somewhat by doing things like, quote unquote, adjusting for age and sex. But the problem I run into there is that can end up basically aliasing the signal of interest that you actually want to measure, which would be the actual batch related effect. And so what we do in a few slides is we'll actually just try to restrict ourselves to the shared regions of the covariate distributions in which the two data sets are basically similar enough in these sorts of signals of interest. And let's say now we have a sort of a third study where the experimenter realizes this issue. He goes to a new group of people who are effectively identical in covariate distribution to the people in data set 1. We'll call that data set 0. And he measures FC again, but he's at a different university. So and this time we end up with a much more similar agreement, but we're still getting disagreement in about 20% of the edges. And so now the actual statement that you start to be able to make is that you have a piece of evidence that the batch, the factors that relate to the site, the MR machine, the experimenter, the protocol, et cetera, is leading to this disparity in the detected signal. Kind of stated another way, we start to be able to have some piece of evidence that something related to the batch is what's actually causing the signal disparity between the two data sets. So to put this into practice, these sort of two pieces, we're going to look at the prominence of batch effects compared to local associational effects in the core metadata set. So for those who aren't familiar, the core metadata set comprises a few thousand individuals measured at a couple dozen of sites. And we basically restricted ourselves to the subset of data sets where we had a bunch of phenotypic covariates such as the sex, age, and continent of the individuals who were measured. So we estimated FC using the AAL parcellation, and our procedure was pretty straightforward. So first we compare the association between the dataset identifier with functional connectivity for two pairs of data sets. And next what we did was we estimated a causal effect between the dataset identifier with functional connectivity if such a comparison was possible. And so, by possible in this case, what I really mean is if the two data sets had sufficient overlap in the covariate distribution, very similar to what we saw with data set 0 and data set 1 in that mini example, basically after removal of individuals who are outside of the region of overlap in the covariate distribution. So in this first figure, we took a look at whether or not associational or batch effects were actually present in the data set. So we have a total of 276 possible comparisons amongst all of these 24 times 23 pairs of data sets where we removed duplicate comparisons since our test statistic in this case was symmetric. So we had 170 pairs that differed pretty substantially in the covariate distribution where we did not think such a comparison was warranted. Either this was, the covariate distributions didn't overlap whatsoever, or there were only a very small subset of the individuals in that sort of shared region of the two covariate distributions. And second, we had 106 pairs of data sets where we had a very similar covariate distribution. And those are shown on the right. And so effectively in both plots, what we ended up seeing was that, sort of associational effects between the data sets, as well as, effects where we can begin to make a more causal statement about batch effect, were both present and extremely prominent in the data set. So in the next figure, what we were trying to do was, how can we remove this sort of batch related alias in the data set? And so we used a strategy called combat correction. So basically what happens with combat correction is across all the features, you fit an empirical Bayes model to identify components of signal that are specific to each batch. And you try to take that out. And so, what we can see in both of these figures is that, if we compare this to the previous figure, practically none of the effects are significant in this case after adjustment, which, it indicates we've done a pretty good job of removing, whatever batch-related signal we were detecting previously. So our next question was a little bit more high level. So on one hand, we've removed our ability to detect a batch effect. We don't really know what happened to the rest of the signal that we might want to investigate in our neuroscience investigation or whatever. And so the problem basically boiled down to the fact that, whenever you remove something, you're not really sure whether or not you're also removing other useful things that, might either alias or might be scaled in with whatever your batch removal strategy was. And so to kind of quantify how prominent this was, we identified a few signals of interest that we considered to be, sort of ubiquitous in that they're pretty often or nearly always present, but were unknown to the combat algorithm at the time it was applied. So to start off with, we took a look at a few between individual comparisons that we might be interested in making in connectomes. So before doing combat, while we couldn't compare two data sets due to the problem that we mentioned at the beginning, we could still do inter-individual comparisons within a single data set and look for associational effects. And so what we did here was we took a look at whether or not sex and age effects were present before and after combat. And we found that after adjusting for multiple comparisons, both sex and age effects were fairly prominent. So we haven't removed this sort of inter-individual signal. The second question was, there are also a bunch of within individual properties of the connectome that we're interested in. So here we were seeking to check that properties that were present at the individual level before and after combat were still present. So our individual level signals in this case were the homophilic and the homotopic differences in connectivity. The homophilic difference is the difference between edges in the same hemisphere and the opposite hemisphere. This effect doesn't tend to be super prominent. And on the right plot, we can see what happens with the homotopic difference in connectivity, which was the difference in connectivity between edges that are bilateral homologues, for example, region A in the left and right hemisphere, and edges that were not bilateral homologues, region A and region B, irrespective of hemisphere. And this tends to be one of the strongest signals in functional connectivity that we tend to find, in that it's present, I think 100% of the time across all of the connectomes in the core data set. And so the conclusion here was that combated connectomes tended to yield the same inference as the raw connectomes at the individual level, which was not the case with a couple of other fairly naive methods for batch effect correction that are used fairly prominently in like the genomics literature, like z-scoring, ranking features within a data set, things like that. And so in summary, we proposed a strategy for basically more precisely posing the batch effects problem as a causal problem. We proposed a method for estimating batch effects and differentiating batch effects from associational effects at the interdata set level. We showed that batch effects could be successfully removed with combat correction, and we showed that combated connectomes preserved between and within the individual properties of interest. So finally, some people I'd like to thank are my PIs, Josh Vogelstein and Brian Caffo, a few colleagues that I work extremely frequently with, Mike Powell, Anton Eliakin, and Noam Finkelstein, and finally, the Consortium for Reliability and Reproducibility. Thanks, guys.
Thanks, Eric. That was really exciting. So yeah, just a reminder, if you want to ask any questions, feel free to ask in the Q&A. Just have a few minutes for those. I'll jump in real quick. So you talked about looking at covariates such as age and sex. Are there other covariates that you're interested in looking at, particularly what comes to mind for me is like different scanners or something. Are there, variations across the measurement devices themselves that you might want to correct for?
Yeah, no, I think that's a great point. And so when I say batch in this case, that's kind of one of the signals that we would be interested in summarizing with some sort of a batch effect, would be things like, if I measure person A at MR machine 1 and 2, and MR machine 1 and 2 are totally different models, that would be one of the batch related effects that I would kind of want to pick up. With however I was measuring batch. Within the core data set, what tends to be the case is I think the overwhelming majority of the data sets are collected on similar MR machines in that I think most of them were Siemens and Philips, as well as using very similar scanning parameters. So that kind of made the fact that the batch effect was, I think it was present like 85% of the time. I haven't specifically looked at how it differs when you're looking at identical MR machine and identical scan parameters versus otherwise. But there's definitely a lot of examples where it was, same MR machine, same exact scanning parameters, still batch effect present. So
yeah, that seems to be a thing for a lot of other neuroimaging recording modalities as well.
Yeah,
Awesome.
Absolutely.
Great. Well, thanks again, Eric.
Great,
so yeah, I'll introduce our second speaker, who is Robin Ince, and he's a research fellow at the University of Glasgow. And his talk is going to be on Bayesian inference of population prevalence. So take it away, Robin.
Well, thanks very much. So I'm going to talk about how we can reconsider population inference from this prevalence perspective and present a Bayesian method for estimating population prevalence. So as a bit of background, what's the goal when we do statistics for experiments in neuroscience and neuroimaging? I think that often it's to infer a causal relationship between some aspect of the external world or behavior and some aspect of the measured neural activity. So could Eric mute because he's typing.
Hey, Eric. Eric?
Thanks.
Oh, yeah, what's up?
So we want to sort of draw in the connection in this sort of causal DAG between some experimental condition and some aspect of neural activity. And because we use sort of carefully designed experiments that serve as randomized controls trials, that is sort of the gold standard for inferring a causal relationship. So the purpose of bringing to bear our statistics is that we want to be fooled by randomness and have some sort of confidence and control our error rate for drawing in these sort of links. But of course, we want to do this at the population level and not just in the participants we measure. So if we just evaluate an effect within the participants we measure in our study, that would be a case study. But what we need, especially for publication in neuroimaging, is we want to have a population inference to say something about the population from which these subjects were drawn. But one point that I think is not sort of made explicit enough, but is usually implicit, is that to make an inference about the population requires a model of the population. So the standard approach, at least in neuroimaging, is to use a linear mixed effect model with participants as random effects. So this implicitly models the population with a normal distribution. So how does this work? I'm just going to recap a little bit. This is the standard sort of statistical model used in neuroimaging. We have a hierarchical model where each subject mean, mu I, is drawn from a normal distribution at the population level with a population mean and a between subject standard deviation. And then the trials within each subject modeled is being drawn from a normal distribution with the subject specific mean and a within subject standard deviation. So normally we proceed in a two-step procedure where we calculate the effect within each subject, which could be, a simple mean like this, or a difference between conditions, or a D prime, or a, more fancy decoding analysis, or a complex, behavioral learning model. And then we apply a one sample t-test on these per subject effects. And, in the words of the original sort of seminal paper, this means that our estimated activations are being assessed against their variance across subjects, with the goal being whether to infer a non-zero population mean. So we're doing inference on this parameter, the population mean. So then, of course, there's been a lot of work studying, the power of such tests, when we're doing a null hypothesis significance test to reject a 0 population mean. We're interested in the power of this for different effect sizes. And one preprint, which I really liked, so I wanted to highlight, and it also motivated a bit of this, was about power contours. So here they're comparing, looking at the effect not just of sample size in terms of number of participants, but also in the number of trials per participant on the sort of power of this standard t-test approach. And to motivate this, they show these figures at the top, which is simulating data from the null model. And they show this figure, I think, just to illustrate how increasing the number of trials, so we go from 20 trials to 200 trials, increasing the number of trials can affect your inference on the population level because it reduces this population, your sample population standard deviation. But I want to highlight this because for me, this is really the sort of the key of our point in this paper. This under the normal mode of neuroimaging is a null effect, right? It's a 0 population mean. But when I look at this, I see, many subjects in which you're reliably measuring, this is the standard error. So this is the effect of each subject with a bar showing the standard error of that subject. So we're reliably measuring effects within a number of subjects. But, within the t-test framework, within their paper, this doesn't even appear in the power calculations because this is a null effect under the population mean. So we're starting from this, we can actually do an explicit inference within each individual participant. So now at alpha equals 0.05, I've rejected the null hypothesis of a 0 effect in all the subjects that are colored orange. And so we can go from this, so we can count them up. And the first thing we can say is that there's 30 out of 50 participants where we reject the null hypothesis of no effect. So I want to go back to the question I started with and say, what do you think this means? Like, is this a reason to draw in this link? And I think for us, it is. For us, this is evidence of some effect, but it's something that's not really covered in the standard sort of statistical sort of, at least in neuroimaging, the very standard approach of just focusing on the population mean. So what is the difference between these views? I mean, formally, you can say that for the population mean, our null hypothesis is that this population mean is 0. Whereas in this alternative view, our null hypothesis is that the individual subject effects are 0 for all subjects in the population. And that's called the global null hypothesis. But of course, this global null hypothesis can be rejected if there's at least one subject in the population which has an effect. So that's relatively weak, and why it wouldn't normally be considered as meaningful sort of random effects population inference. But still, we can think about how they compare, and I would draw the example. So this is just a range of simulations from this hierarchical model. I draw your attention to panel B. So this is like 200 trials, 0 population mean. So we correctly failed to reject the population mean null hypothesis, but we have 32 out of 50 in this random simulation having a significant effect. So the p-value for seeing 32 out of 50 accounting for false positives at alpha equals 0.05 is extremely small, smaller than floating point precision. So in a sense, this provides enormously strong statistical evidence that you have a non-zero proportion of the population that shows an effect, way beyond the normal standards of evidence for publication. And then I would draw your attention to panel C, where this is a much more conventional sort of study, so only 20 trials per participant. There is a difference in mean, and we detect it nicely with a p-value, 0.08. This is 50 participants, so this would be, absolutely fine to publish, and I think would be, considered a solid result, at least in neuroimaging. But again, even in this case, if we look at the percentage, the proportion of participants that have a significant within participant test, the p-value is much lower. And what this means that actually this result is much more surprising at the individual level than this is. So in that sense, it's less likely. So that's sort of the background, but then we want to model this and get a bit more quantitative estimates. So we model the population as a binary sort of 2 states. One proportion gamma, has this property and the remainder don't. So if we could measure this property directly, it would be just the binomial distribution, but we can't. We measure with our error-prone, within participant statistical tests, which has a false positive rate and a sensitivity. So I'm not going to go too much into mathematical details because of the time, but we model it under this sort of modified binomial model, which has also been used in sort of disease studies in epidemiology. And then we apply sort of standard Bayesian methods to estimate this. So we end up with a posterior distribution of prevalence from which we can extract, various Bayesian quantities like the MAP estimate, which is the most likely value, and these highest posterior density intervals, which is a type of credible interval. So what does that look like? Well, if we take this example, in this random running, there was 24 out of 50 significant. We can then convert that to a sort of Bayesian posterior. This is the graph of the Bayesian posterior. We have the MAP illustrated with a line. The large wedge is 50% highest posterior density interval, and the other is the 96% interval. So we've gone from within participant tests to a population estimate. So the point I really want to emphasize is that this is, a valid, random effects population interval estimate. And in this case, we've got 50 subjects. And, what we've learned, obviously we can't pinpoint exactly with very high precision the proportion of the population, because that would take much more subjects. But even with what we have, we can be pretty sure the proportion of the population that shows this effect we're testing for is greater than 25% and less than 75%. In all these examples, I've done two-sided tests, but I mean, it works exactly the same just with one-sided. Just to give an idea what these look like, if you did a subtest with an experiment with 50 participants, and this is, how your posterior changes as a function of, number of participants with a significant test. So you can see that, 4 or 5 significant is very close to what you'd expect by chance if there was no effect, but it increases as you get more. And this works even with relatively small numbers of participants by neuroimaging standards. So you can think of the Midnight Scanning Club, a very nice data set, which has many hours of recordings from just 10 participants. And you can see that even with just sort of 6 out of 10, we start to be pretty sure our proportion is not 0. And if we got 7, 8 out of 10, we start to be pretty sure, you have good evidence that the proportion of the population that shows this effect is greater than 40%. So you can learn meaningful things about the population even from small numbers of participants. And this is how obviously the posterior narrows as you get more participants. It narrows much more at the start, and then you get diminishing returns. Obviously, if you want to estimate the population prevalence to within sort of 0.05, you're going to need many hundreds of participants, and we quantify this a little bit in the preprint with these kind of looking at this scaling in a bit more detail, which is in the preprint if you're interested. We can do between group comparisons, so we can apply the same test to 2 different sets of participants, or we can apply different tests to a single set of participants, and then we have to use numerical methods here. There's no closed form Bayesian solution, but we can get Bayesian posteriors on these prevalence differences. And so I think that makes it more applicable for common scientific questions where you want to know how your experiment is different between groups or between different conditions. So to summarize, we have this sort of binomial model of statistical testing, which provides a bridge from within participant testing to the population. So the main thing that I want to get across is this switch of perspective from thinking about inference on the mean, to which assumes normality, to this sort of alternative approach. There are other things we can ask about the population. So in a sense, it's more general. There's less assumptions. We only require that the test is the same across participants and has the false positive rate controlled. And the output is not a binary inference. We have a quantitative estimate with associated uncertainty. And it's very easy to apply. You just have to do your inference at the first level and then take forward to the second level, integer counts of the different conditions, a number of significant subjects. So it can apply to any statistical test in psychology, neuroimaging, electrophysiology. Today, I've mostly been talking about experimental units being human participants, but they could also be, individual animals in electrophysiology, or even single units within a brain region where you could use the prevalence different functions to, report differences in the population of the proportion of responsive units that respond in a certain way, say between different brain regions or between different species. But again, it would be a link from single units to the population of cells within the area recording from. We argue that in psychology and neuroimaging, cognitive effects might be more interesting to quantify at the individual participant level. And also that when you do within participant statistics, every participant is a replication, and so you may get more replicability and robustness. Provided the family-wise error rate is controlled within a region per participant, we can then combine our results across participants at this second level Bayesian inference without actually having precise alignment within individuals. And I think this could be a big advantage for sort of high density intracranial recordings or high field fMRI, where it's much harder to really align everything across subjects and just take the mean. And finally, it allows for this kind of inference from small samples or dense sample studies or clinical studies where you might have higher variability. Finally, just wanted to make a point that there was a very nice editorial in the Journal of Neuroscience this summer, which pointed out, this small n approaches, the possible advantages of it. But they say it should be acknowledged that this approach only allows for statements that pertain to the existence and magnitude of effects in those subjects, rather than the populations those subjects are drawn from. So I want to point it out that that's until now, we would say that this Bayesian prevalence sort of lets you make this leap from within participant inference. This summary just restates, I guess, what I had before, and I think I'm running out of time. So I'll just thank my co-authors the preprint is here and there's code in MATLAB, Python, and R, for a very simple method. But I hope that it's a different way to think about presenting your results and think about what we can learn about the populations we're interested in from the experiments we do. Thanks.
Awesome.
Yeah.
Thanks, Robin. That was very interesting. As someone who's played around with or tried to use mixed effects models before, it's nice to see some approach that might be more effective and potentially a little bit easier to use.
Yeah, I guess that really one goal is these, when we have in neuroimaging much more complicated nonlinear analysis, decoding, RSA, these sort of fancy things, it's not always easy to, put them into these kind of hierarchical models, at least down to the trial level.
Yeah.
I can't see the chat window with this. I can stop sharing.
Yeah. So yeah, if you have any questions for Robin, please enter them in the Q&A. We just have another minute or two. I guess while that's going on, so sort of going to what Eric's talk was about, like, are you able to put in a bunch of covariates as well when you look at this, or is it mostly looking at one effect across like the population and the individual subjects?
Yeah, I think the idea is really that you do your whatever you're interested in your experiment, you would sort of control for any sort of covariates on the trial to trial level. So you're moving away from thinking about the population, quantifying the effect of this group mean, but you think about each subject as their own experiment. So, you might have your sequence, you could do complex, modeling of behavior or sequences, and in that you might want to control for, various other things going on. So it doesn't make any sort of requirement on the analysis, just that it should be, properly controlled experiment with a false positive rate. And then what you're saying about the population is much simpler. All you're saying is that the population that I randomly sampled from, this proportion would be expected to have a true positive effect if I repeated the experiment on them.
Oh, that's great. Yeah, it's really exciting. Great. So I think we're going to move on to our last speaker, which is Tony Liu. And so Tony is a graduate researcher in computer science at the University of Pennsylvania in the labs of Lyle Unger and Conrad Cording. His talk is on quantifying causality with quasi-experiments. So take it away, Tony.
Okay. Thanks for the introduction, Steve. Yeah, today I'll be talking about quantifying causality more generally using the idea of quasi-experiments. So where are we headed? First, I'll talk about why estimating causality is important and why it's difficult, especially in settings when we don't have randomization of treatment. And then I'll talk about how causality can sometimes be estimated using quasi-experiments. So first, I wanted to lead with the argument that causality is at the heart of many questions that we care about as scientists, right? We ultimately want to understand causal mechanisms. Even if we don't explicitly state causality is our goal at the upfront of our study. Say if we want to examine the relationship between brain region A and brain region B, we might use a correlation analysis because that's sort of the best we can do with the data. But what we really care about is a causal relationship between brain region A and B. So we should really be putting causality at the front and center of our scientific studies. So let's get to one working definition of causality. You might be familiar with this in terms of the counterfactual framework. So let's have two variables, X and Y. And the question we ask is, if we change X, does Y change? And if Y does change, then we'll say that X has a causal effect on Y. We can represent this graphically with a directed edge going from X to Y, indicating the direction of the causal relationship. And we commonly call this upstream variable the treatment, and the downstream variable Y the outcome. And we usually want to measure the effect the treatment has on the outcome. Okay, so how do we do this? Now, we've all heard of the saying that correlation is not causation, but I also want to emphasize a related point, and that prediction is also not causation. Machine learning prediction, to be exact. Standard machine learning algorithms really only capture correlational relationships. And what we'll use throughout this, the rest of the talk is a motivating example coming from a real machine learning study back in the 90s on pneumonia. So let's say we're a team of data scientists, right? And the hospital gives us a data set. And the hospital wants us to help them with this decision of whether a pneumonia patient should be sent home or admitted to the hospital. Now, there's a related causal question in that what is the effect of hospitalization on our patient's pneumonia mortality risk, right? if we estimate the effect is small, then maybe we might be better off sending them home. Or if the effect is large, then we should probably admit them. So now, we're data scientists. We have all this healthcare data that the hospital gave us. Let's build a machine learning model. And we build a machine learning model to predict pneumonia mortality risk. And what we find in the results is something somewhat surprising. We see that our model predicts that being hospitalized actually increases a patient's pneumonia mortality risk. Now, this sounds completely backwards of what we think. And if we use this model for decision making, it would be sort of disastrous, right? Well, it's saying that hospitalization will make you worse, so we'll never hospitalize anyone ever. Like, why would we? That sounds completely nonsensical. But why did this machine learning algorithm come to this conclusion? The problem is what's known as hidden confounding. In this particular situation that I'm talking about with the study back in the 90s, asthma was a hidden confounder of both hospitalization and mortality risk, right? Asthmatic patients are not only more likely to go to the hospital, but they're also more likely to die from pneumonia because of complications. So if the machine learning model that we trained does not account for asthma, indicated by the dotted outline, it learns the erroneous relationship that hospitalization makes you more likely to die when the true relationship is being driven by the presence of asthma. So with this idea of confounding in mind, how do we actually go about estimating the causal effect of X on Y? Well, the gold standard in these situations is an experiment. And what I mean by an experiment is just any sort of study where we can randomize the assignment of X. And so what we have is our standard setup where the treatment, we're trying to measure the effect the treatment has on the outcome in the presence of confounding. And with the addition of random assignment, we are able to break the links between any confounders and the treatment and outcome. So if we randomly assign the treatment, we can know that the changes in the outcome Y due to the changes in X are because of X and not because of some spurious correlation with the confounder. And this allows us to estimate the causal effect of X on Y correctly. Now, this is sort of great. This is what we want. But in many cases, experiments are actually practically or unethically or ethically infeasible. Let's go back to our pneumonia example, right? If we wanted to run a clinical trial trying to figure out the relationship between hospitalization and mortality risk, this probably wouldn't fly in terms of IRB, right? If we randomly assign hospitalization to patients, we couldn't go through with that study on sort of ethical grounds. And in a lot of situations, we can't run experiments because of barriers like this, right? So what do we do? Well, we still have our original data that we ran our machine learning model on, right? But this is really what's known as observational data. And so we have to conduct an observational study. And in these situations, we do have to take into account the confounders. And what's commonly proposed in these situations is to control for the confounders that are present in our data. Suppose that we know from the onset that asthma is a confounder, and we can then subsequently include asthma in our analyses or regression of running regression of Y on X to account for any spurious correlations that it might induce, right? So we solve the confounding of asthma. But there's actually another problem. What if there's another hidden confounder that we didn't account for, such as smoking history? What if we didn't measure that? Well, we can say that maybe we anticipated that ahead of time and did measure smoking history, but the problem is we can sort of continue playing this game. We can keep on saying that there are additional confounders that we may not have recognized in these situations that could affect the relationship between X and Y. And so if we want to control for confounding correctly in an observational study, we need to control for all the possible confounders. And in many realistic scenarios, it's really, really difficult to claim this is true, right? Think of the brain. If we're trying to measure neural activity, it's conceivable that every other neuron could be a confounder, and we cannot possibly hope to measure all possible neurons. It's just not feasible, right? So in these observational settings, what can we do? There's an alternative called quasi-experiments that are proposed by, that have been proposed by economists and statisticians over the past decades. And the idea is to look for something in the data that randomizes the treatment X for us, hence quasi-experimental. And there are numerous number of quasi-experimental techniques that allow us to estimate causality from observational data. And what we'll do today is take a look at one technique, the regression discontinuity design, in detail and see how it allows us, see how the data randomizes X for us. Okay, so what is the regression discontinuity design, or RDD for short? So the key to this design is that there is a threshold on a continuous variable, say age, that determines treatment assignment. So individuals above a certain age will get the treatment, and below a certain age will not get the treatment. And we'll sort of make this concrete with our pneumonia example. So in medicine, there are a lot of clinical rules governing the hospitalization of pneumonia patients. One rule is that if their patient's respiration rate, number of times they breathe in a minute, is greater than 30, then you should admit that patient to the hospital. And if it's below 30, then you're safe to send them home. Okay, and of course, being admitted to the hospital is going to affect their pneumonia mortality risk. And so what we can do is plot as a function of the respiration rate, our individual patient's mortality risk. And what we'll see in this data, with x-axis being the breathing rate and the y-axis being mortality risk, that at that cutoff of 30, there's going to be sort of a discontinuous jump in the mortality risk of our patients. So how do we get causal effects from this scenario? Well, one approach might be to just look at the outcomes of people to the left of the threshold, not hospitalized, and the people to the right of the threshold who are hospitalized, and just compare how they're doing. Now, this approach will result in biased estimates because people on the left of the threshold and people on the right of the threshold are different. If you have a lower respiration rate, you're probably younger, you're probably healthier. If you have a higher respiration rate, you're probably older, you might even have asthma, right? So these comparisons are actually biased. But if we look at a vicinity around the threshold, I claim that everyone will look the same on average within a region around the threshold. Now, why is this? The key is that the respiration rate, like any continuous measurement, is going to be noisy. So if we continually remeasure a patient's respiration rate, sometimes it'll be 31, sometimes it'll be 29, sometimes it'll be 32. But on average, we'll see that within this narrow region around the threshold, the patients just to the left and just to the right are going to be comparable. We've essentially created an experiment local to the cutoff, and this enables us to estimate causal effects, at least within this small region. Okay, we can actually represent this graphically in the same sort of framework we had before, where we have a running variable determining a cutoff, which then determines our treatment assignment and affects our outcome. Mapping that to our pneumonia example, we have the respiration rate as a running variable, the cutoff of 30, and hospitalization and pneumonia risk as our treatments and outcomes. And now, like I said, when you're local to the cutoff, we have an experiment, and that allows us to break any incoming connections to our randomizer node, which is C in this case. And now what we have graphically is actually the same setup as that gold standard experiment that we saw at the beginning of the talk, right? Where C is providing randomization of the hospitalization. And this allows us to estimate causal effects without having to explicitly assign hospitalization ourselves. All right, so this is a medical example. Let's get to some neuroscience. I want to briefly talk about an example that one of my lab mates, Ben Lansdale, has worked on. So we can think about how neurons learn to fire and treat this as a RDD, where the threshold here is the input drive to their spiking threshold, and the output is some reward signal, say a dopamine response. And what he found is that in this causal setup, the neurons will converge to optimal weights much quicker in an RDD than in a non-causal learning framework. I also want to sort of briefly mention my lab mates Ari Benjamin's neuromatch talk this coming Friday on teaching causality in neuroscientists, where we also talk about causality and quasi-experimental designs. Now, these quasi-experimental techniques are not a silver bullet. There are actually quite a number of significant limitations to them. The first being that they require a very specific data generating process. If we don't have a threshold in our data, then we're sort of out of luck in terms of being able to apply a regression discontinuity design. Furthermore, these quasi-experiments are much more data hungry than true experiments. And this becomes an issue when we're trying to talk about small sample sizes, right? So what are a couple takeaways? The first is that I think causality should be considered in all scientific questions. When we're conducting a study, we should ask ourselves if the question we're really trying to answer is in fact a causal question. And if it is a causal question, we need to recognize things like correlations or ML will fall short, and we need to use techniques that actually measure causality. If we do want causality, experiments are the gold standard, but they may not be feasible on practical or ethical concerns. And these quasi-experiments that we've seen provide a handle into estimating causality in observational data, and we should be on the lookout for quasi-experiments in the data that we might encounter, say thresholds in the context of RDDs. With that, I'd like to thank my advisors, Lyle Unger and Conrad Cording, and I'll be happy to take any questions.
Thanks, Tony. Yeah, that was really it's really interesting to learn a little bit more about how causality is framed. So yeah, you have a question in the chat, which asks, I don't know how the RDD circumvents the problem of not being able to know all confounding variables, i.e. basically, understand the full causal model. Can you elaborate a little bit more on that?
Yeah, sure thing. Yeah, so we talk about, let me go back to that slide. So we talk about in general that, say, people with different levels of respiration rate might have confounding factors that influence whether or not they're treated or not treated, right? Asthma, for example. People who have higher respiration rates probably will have asthma, and that's a confounding factor there. And so on, if we consider the entirety of the running variable, that respiration rate, then yes, there will be confounding effects. But the key is that if we get close enough to the boundary, and close enough to the cutoff boundary, there is going to be a randomization effect in that there's noise being applied to the measurements that we're making. So even if you're asthmatic and sitting at 29 versus 31, you're comparable to another asthmatic person who might have a rating of just below the threshold when you're just above the threshold. So as long as you get close enough to the boundary, we let stochasticity present within the system to take care of the randomization for us. And there's always going to be issues of how wide we can make this boundary, which leads to extrapolation problems. But at least close to the cutoff, we can say that we're as good as random in terms of, and not have to worry about confounding factors. Cool.
Excellent. I guess just a quick question. Actually, I have another question too, where it talks about, the randomization by the threshold that you're talking about seems to rely on random noise. What happens if there's correlated noise?
Yeah, that's also a good question. So let's see, how do I want to phrase it? So that is a problem, and we can frame this as sort of a way where it's sort of a pitfall or something that we have to consider when we're applying our regression discontinuity, and that there might be so-called cheating at the threshold. So people might be aware of the threshold and have sort of a tendency to manipulate their own running variable to be on one side of the threshold or another side of the threshold. To keep it like a concrete example, say if like you're a pneumonia patient and you really want to be hospitalized for whatever reason, you can ask your doctor like, hey, can you make sure my reading is 31 so I can be hospitalized? And that produces a correlated noise, a correlated bias of people who appear on one side of the threshold versus the other. Now this is a problem, right? This will cause issues with our analysis. But what's nice about this technique is that you can actually detect these sorts of biases in the data. If we plot the sort of density of people to the left and people to the right of the threshold, it's sort of a robustness check of the feasibility of the design to say that, okay, the density should be continuous when we cross the threshold. And if we see sort of a spike in one direction or a spike in the other direction, then we say that we can't use an RDD in this situation. So yes, yeah, the correlated noise is a problem, but we can test for it.
That makes sense.
Yeah.
All right. Well, yeah, I'd just like to thank all three of our speakers again. Really fascinating talks. Thanks everyone for attending this session. And yeah, please attend some of the other ones.
Thank you.
Bye, everyone.
Thanks, guys.
In the last year. So it's been nice to have a bunch of motor control people around.
And
we go on.
Hey, I have a button that says start webinar. Do I do that?
No, I start. I still started.
Okay, there. All right, great. So you're starting the webinar. We're waiting for people to show up. Oh, they're coming. Oh, 7, 8. Okay, so we just gotta let them come in. Oh, here they are. Friedel's here. I see some of these names. So this is a great forum and a good opportunity. Plus, sometimes I don't miss the travel at all, and other times it's like, well, it would be nice to not have the blower going.
Well, SFN was nice this year because it was in DC, and I was going to take the whole lab because it's a 2-hour train ride, but
yeah, exactly.
Now they're all stuck here. So
Yeah, well, I should visit you. All right, we can get started. Okay. Welcome everyone to this NeuroMatched keynote lecture. And it is my great pleasure to introduce Jennifer Semrau, who is now at the University of Delaware in the Department of Kinesiology and Applied Physiology. And she'll be talking to us about proprioception after stroke, a complicated relationship with motor control and visual feedback. Before I start, I want to make sure that people know to go ahead and place your questions in the question and answer, and I will read them out afterwards in our discussion session. The talk will be about 30 minutes, and then we'll have 15 minutes for discussion. If you go in the chat, we'll see that, but we prefer to have all the questions in the Q&A, which is on the far right, on the bottom of your screen. Okay, with that, take it away, Jennifer.
Well, thanks, Lena. And so I'm really excited to give a talk for this forum today. I haven't given a virtual conference talk yet. So this is a really great first opportunity for me. And I think I think I've heard tons of great things about NeuroMatch. So as Lena said, I'm going to talk to you about proprioception after stroke today. And I'm really going to focus on two main kind of modalities, that being its relationship with visual feedback, and the other being the relationship between proprioception and motor control. And so I always like to start off my talks with kind of a really general example. And so when we think about this little guy trying to catch a ball, you know, one of the first things he needs to do is be able to visually identify that it's a ball, and he needs to know the trajectory at which it's traveling, and that dictates, you know, where his arms are going to go. And in order to do that, he needs proprioceptive information about where his limbs are in space, and then he needs to be able to actually execute the movement. And so we do these things fluidly every day, and that's kind of your general motor control intro that everybody has. But when we think about stroke, stroke is really complex when we think about it from a sensory motor standpoint. And so I have kind of these three boxes. We have motor control, vision, and proprioception. And I know that that is a gross overview of the systems, but in general, that's kind of how I think about the interaction between these things. And so, you know, we can, when you have a stroke, a lot of different things can be impacted, and you can have a stroke that really affects anywhere in the brain. And so, you know, maybe centers that control motor function are affected, maybe centers that control vision are affected, and maybe centers that control proprioception are affected. But to make it more complicated, maybe the connections between each of these different kind of modalities are actually impacted. And so when we think about not only, you know, these individual kind of groupings of behavior, but also the way that they interact, we have a really poor understanding of those interactions in the types of impairments that people have after stroke. And so I do like to preface, you know, talking about stroke, about, you know, why do I study stroke? Well, one of the things that's important to know is kind of the vascular organization of the brain. And a lot of the strokes that happen to the middle cerebral artery, which is, you know, runs kind of right up the middle of the brain here. And when we look at the actual territory that MCA, or the middle cerebral artery, is responsible for, it's actually a pretty large portion of the brain, and kind of smack dab in the middle of that are motor and somatosensory cortices. And so when we think about what happens after stroke, you know, movement is one of the things that is most commonly affected. And so when we think about it on a more individual level, you know, there's a lot of structures in the brain that can be differentially affected by stroke. And so I'm showing some examples here from a study that came out of Sean Duclos' lab in 2014 that actually looked at, you know, really focal lesions of people that had stroke, and then looked at their sensory motor impairments. In this case, they looked at motor impairments and proprioceptive impairments. And so the three examples I have here on the left are actually individuals that have thalamic lesions. And so you can see that, you know, these individual thalamic lesions, and these were focalized lesions, that you see, you know, a little bit of impairment here, you see more impairment here, and you see a significant amount of impairment here on this third example. And so despite the fact that these two lesions are fairly similar, this one's a bit bigger, you see kind of the gamut of, different forms of motor impairment. And here what people are doing, they're just making center out reaching movement. The same can be said for lesions that happen in the posterior limb of the internal capsule that carries really important fibers for sensory behavior. And so what we see is that, we have a lesion here and we have a small lesion here, but we can have really different phenotypes of different types of motor behavior and different types of impairment. And so, I like to put that in focus, just because when we think about the types of impairments that people have, everyone is different when we think about stroke. And so to frame kind of the bigger problem in stroke, a lot of people have upper limb impairment after stroke, and I study upper limb impairment. And so approximately 75% of individuals, but nearly everyone undergoes some form of rehabilitation. And so there's a couple problems that we face in trying to understand sensory motor impairment after stroke. And one is that often the clinical measures we use lack sensitivity to change. And so they can't pick up on a lot of the nuances that we are familiar with in the field of motor control, things like kinematics and kinetics. They can often be subjective. And so sometimes this is a good thing, but sometimes this can be detrimental. Another thing is floor and ceiling effects. And so the Fuchel-Meyer is a really commonly used motor score, and it has a really well-known ceiling effect where you can still have some sort of impairment, and particularly motor impairment, but that you actually, the scale actually can't pick up on that. But most importantly, it doesn't necessarily tell you the underlying cause of the deficit. And so, what I really care about is, developing better tools to really understand the types and the nature of the impairments that people have after they've had a stroke. And so the big thing that I care about is proprioception. I'm going to talk a lot about proprioception today. And, one of the things is that we don't really have good tools to measure proprioception in people that have motor impairment. And so hopefully I will convince you of that, that we're working towards a better goal for that by the end of the talk. And so, I always give an example of exactly how we measure proprioception clinically. And one way is you do kind of this self-report digit movement. And so your clinician, and so I'm in the camera, it's showing, I usually do demonstrations if we're in person, but you move your finger up, or the clinician moves your finger down, and then you're supposed to report, did you move it up or down, yes or no? But, when we think about that test for proprioception, that's not really that great of a test. We can also use something that's a bit more sensitive called the thumb localizer test. And what this test does is it actually measures a person's ability to find their thumb in space on a 4-point scale. And so, this one is a little bit better, but it's still not great. And so, one problem that has kind of been the case in the field of stroke recovery and stroke rehabilitation is that when we think about impairment, we naturally think about motor impairment after stroke. And so, motor control and proprioception are so tightly linked that we don't really know how impairments or deficits in proprioception can actually impact motor control. And so I'm going to talk mostly about that today. And so, in my lab and in the work I did in Sean Duclos' lab, we use robotic assessments for proprioceptive impairment. And so when I think about proprioception as a broad definition, there's several subtypes, some of which are not listed here. But, my work has really focused on two major subtypes of proprioception, the first being position sense, which we are not really going to talk about today. The second being kinesthesia, which is the sense of limb movement. And so if you were to close your eyes and kind of do a John Travolta disco move, you would know that your arm moved up and to the right about 45 degrees. And so the one thing to understand about proprioception, especially when we think about the clinical implications and the clinical problems, measuring proprioception in individuals that have a motor impairment is very difficult, because it is difficult to dissociate what is a motor impairment and what is a sensory impairment. And so we have to come up with creative ideas of how to actually approach that. The second thing is that, because we're not really that great at measuring proprioceptive impairment, that often proprioceptive deficits can be misdiagnosed as motor impairment. And so, one good example of this was a gentleman we had come through when I was in Calgary for my postdoc, but he was tested across 6 months on a lot of our measures, but he came back one day and talked to one of our therapists and said, I feel amazing. I feel like I've recovered. But the only thing is that when I'm putting on horseshoes, he was a farrier, so someone who puts on horseshoes. He said that when he was putting on horseshoes, he had to look at his hands. And that was something he never had to do before. And so, from the PT that worked in the lab, it was, her clear interpretation of that was that this was in fact a proprioceptive impairment and not a motor impairment, just based on the, 25 years of experience that she had. And so kind of the bigger thing is that proprioception is not often evaluated or treated in stroke. And so when I talk to clinicians, I usually ask them if they, if they measure proprioception, the vast majority of the time, that answer is no. And so we're working towards trying to build a better understanding of what these deficits actually look like. And so one of the first studies that I worked on in looking at proprioception after stroke was looking at kinesthesia, or the sense of movement. And so, this work was done in Sean Duclos' lab. And so what we use in that lab and what I use in my lab here at Delaware is a kid-arm robotic exoskeleton. And so the person comes in and is seated in the robot. There's motors that can control both the left and the right arms. And then the person is wheeled into this virtual reality environment. And so they're looking down on a horizontally mounted screen. And so this kinesthetic task, what happens in this task is that the robot moves one of the person's arms. And so in this cartoon example here, the robot's moving the right arm. And then as soon as the person feels the robot begin to move this arm, they're supposed to match that movement. They're supposed to match the speed, direction, and magnitude of that movement. And so this is a task that's designed to assess the person's sense of movement. And so, in this, what we do is we eliminate any source of vision. We take away any sort of visual feedback. We also take away any feedback they might get about their upper limbs by putting a bib on them. And so here we tested 113 individuals with stroke at approximately 10 days post-stroke. And so just for reference, in terms of, evaluating sensory motor behavior, this is a fairly large study. Having over 100 subjects, and we tested a lot of control subjects as well. And so I just want to show some videos of kind of what this looks like, and hopefully this will move smoothly. I've had some video Zoom problems. And so on the left here, we have someone who's a control. And so this control subject, the robot is going to move their left arm, and then the subject is going to move their right arm to match that movement. And so we have a setting movement in the beginning. The robot moves the left arm, and then the person matches right away. And so we'll let it cycle through a couple movements. The robot resets, and then the robot moves the left arm, and the person matches the right arm. And so this person is actually quite good at this task. And so if we look at what that data actually looks like, this is just an example robot trajectory, and this is an example speed profile of the robot. And we move people around in 6 directions for this. And so if we take all the trials from one set of movements, we can see that this person moves in a nice straight line. They move just about the distance to the target, and it closely mirrors what the robot did. The speed profile also matches up quite nicely. And so when we look at what someone with stroke looks like, and so here we're having the robot move the person's left arm, and that's the stroke affected arm. So the person is not actually creating any motor output with their left arm. We're using the right arm as a behavioral output of their sense of movement. And so the robot moves the left arm, subject moves the right arm. And so you could tell just from watching the video that those two were a bit different. And you can see that even here, the stroke participant is actually winding up in a different position based on what their initial planned trajectory was. And so if we do the same thing, we look at all of these movements that were supposed to be in this direction, we can see that there's a lot of variability with which the person moves, but we also see that the directional error is very high and that the speed profiles are actually very variable as well. And so, you know, one of the things that we found from this study, it was that 60% of individuals do have these kinesthetic impairments after stroke. And so we're evaluating people approximately 10 days after stroke, and that's quite a large portion of this population that we're looking at. And so I'm just showing the exemplars from the paper here. We have someone who has a left affected stroke, someone who has a right affected stroke. And the thing that I always like to point out is that, you know, this person here on the left in blue, they on average moved a little bit slower than the robot, probably half as much, whereas this person who had a right affected stroke actually moved twice as fast as the robot. And so the thing that I'd like to just kind of drive home is that it's not, the deficits that we see aren't an artifact of these people not being able to move with their unaffected arm. Remember, the unaffected arm, these are people that have unilateral stroke. Their unaffected arm typically does not have a motor impairment. And so we see that, you know, on a parameter by parameter basis, that these people are fairly impaired with, you know, sense of direction and their timing relative to actually generating that movement. And so we're looking at here at a cumulative sum histogram. And so here, you know, if we draw over to the percentage, you know, it's, you know, between 30 and 50% of people are actually getting the direction right. And so, and that's, you know, that's not great. It's good that we understand this, but this is actually, you know, a pretty significant problem when we think about the fact that proprioception is impaired after stroke. And so this led to, you know, a lot of subsequent studies. But the one thing that we noticed was that when we allowed people to do this task with vision, and so we gave them complete visual feedback of their arms and visual feedback of their hands underneath the mirror, that they actually didn't, there was a lot of weird behavior, meaning that a lot of people didn't improve. And so we did another study that was published in 2018. And this time we looked at approximately 281 individuals at 10 days post-stroke. And so here, what we did was we first had people do this task without the use of vision, and then had them do the task again now with vision. So they had complete visual feedback of their limbs in space. We took away the bib, we took away the screen on the mirror. And so one of the interesting things that happened is that, you know, when we look at what control performance actually looks like, which is right here, we see that the blue is no vision and the red is vision. The blue on average for most controls was a bit more variable than the vision condition. You give controls back and that performance improves. If we look at people who've had a stroke, we have this individual here in panel C at the top. When we look at the blue is no vision, and then we give them vision of their limbs, they actually improve quite a bit. You can see that when they don't have vision, they have these large curved trajectories that are sort of in the correct direction to the target. But these, when we give them vision, which is the red lines, this becomes a lot more regular. So we do have people that improve with vision, and that's great because that's a strategy that therapists use. When OTs and PTs say, okay, now look at your hands, I need you to pick up that cup. They're using visual information to help their motor and proprioceptive information. The last panel here is an example of an individual who had a stroke but doesn't show improvement, meaning that their no vision condition and their vision condition looks identical. You wouldn't be able to tell the difference between these two. So there's something weird here that they actually cannot use the visual representation of their hands or their arms to actually improve the behavior output of this particular task. And so what we did is we looked at all of the different magnitudes of how these people improved. And so what we're looking at is a cumulative sum histogram here of on this axis here is all these closed squares are performance of the task without vision. And then kind of these lines jutting out from the left and the right, these are actually the open squares is the person's performance on the task with vision. And so if you're moving this way to the left, that means that you're improving. And so we had some people who significantly improved, and that's the folks in the blue. And so these are people that do really poorly without vision, but actually improved to this control level, which is this gray box. But we have all these people in purple who don't improve to control levels. We have some people who improve a lot. We have some people who do the same, and we have some people that get worse. And so this was a really kind of weird, puzzling question that, you know, is this a problem with vision? Well, you know, we screen everybody for visual impairment. We screen everybody for field deficits, and we screen everybody for spatial neglect. And yes, there are some people with spatial neglect in this sample, but I think that approximately 50% of these people were people who didn't have neglect. And so we have this kind of larger subpopulation of people who are not able to use vision to their advantage when we give it to them. And so, you know, this is, you know, my theory behind this is that this is probably a problem with the integration of these two different forms of information. And, you know, stay tuned, because that's where I'm headed right now, and we're doing some of that work in my lab. So I'm going to switch gears a little bit and talk. So we talked about, you know, the relationship of vision and proprioception, particularly kinesthesia, but I'm also going to talk a bit about motor control. And so, you know, one thing that I think that people who care about sensory motor control and stroke really care about is, you know, do people improve? How do they improve? What are the characteristics of those different types of impairments? And so, you know, what I'm showing you here on the left is a slide from the Copenhagen stroke study that was done in the 90s. And what they found from that study, or the main take home of that study, was that regardless of stroke severity, the people plateau by 3 months post-stroke. And so they failed to make any other improvements beyond that. But, you know, one of the biggest problems, I think, and that's noted by, you know, many researchers, is that there's a lot of variability in stroke. And so how do we actually address that problem? And so a study that we did in 2015 actually looked at the, you know, inter-individual variability in terms of motor and proprioceptive function, but we also looked at a lot of clinical measures as well. And so the FIM is a measure that's actually used to discharge individuals from the hospital, and is used by insurance companies. And so if you look at this average curve, it definitely looks like people plateau right at 12 weeks. But if you start overlaying, you know, the actual data on top of it, we see that when we look at these individual recovery curves, that everyone is different. And we, you know, we have a pretty significant ceiling effect, but everyone is different too. And so one of the ways that we can kind of address that is actually using robotics to really try and understand how people recover, how their motor and proprioceptive function recover after stroke. And so the study that we did in 2015 actually looked at 2 different motor tasks. I'm only going to talk about our visually guided reaching task here today, and 2 different proprioceptive tasks. And so I'm only going to talk about the position matching task today. And so the position matching task is very similar to the kinesthetic matching task. And instead of matching the movement, people are now matching the location. So it's a measure of static position sense. And so, you know, people came in over the course of 6 months for this study at 1, 6, 12, and 26 weeks. So we had them, you know, we had regular visits for a while, and we're really interested in understanding, you know, what does recovery look like for everyone, and how do these different modalities of sensory motor control actually interact? And so if we look at this, I present a lot of examples in this section just to give you an idea of kind of what this data actually looks like. And so when we think about, you know, inter-individual variability, you know, we have different timelines of motor recovery. And so I'm showing you 2 subjects here, 2 participants here. The first person, this person has significant motor impairment at 1 week post-stroke. So the timing is labeled under here. But by 6 weeks, they have within control range motor behavior. If we look at the second individual, this person has really impaired motor performance at 1 week, 6 weeks. And then they make kind of this big jump from 6 weeks to 12 weeks, and they continue to improve all the way out to 6 months. While they're still very impaired, you know, there is changes happening across this 6-month trajectory. And so we see that, you know, depending on a person-to-person basis, that we do see differences in the timing of the person's recovery for motor control. You know, on the flip side, if we look at what proprioceptive recovery looks like, I'm showing you 2 examples of our position sense task. And so for, you know, good performance on this task, you can think of someone just being able to replicate this black square right here. And so what we see here is this person, so you see these big ellipses, this really indicates a high level of variability in that person's ability to sense where their arm is being moved. But by 6 weeks, they fall into, you know, control level or normal levels of behavior for a position sense task. The person on the bottom, this person has really impaired position sense at every time point that we tested them. And so, you know, we see these big differences in different types of recovery and when they happen. And so, you know, when we think about an individual basis, you know, I think we need to be able to compare, make, you know, objective comparisons between what their performance looks like at, you know, any given time point. So when we think about the recovery of individual parameters, you know, we could think about, you know, averaging over the group, which is, you know, what a lot of studies long ago have done to actually characterize behavior. And so anything below this dotted line here is normal. And so we look at the average, then we put the standard deviation on it, and then we actually put the individual trajectories on it. And this is, you know, it looks like a mess because it is a mess. You know, everyone has their own trajectory of recovery and own trajectory of behavior. And so one of the things that I was really interested after seeing this was, how do these 2 things intersect? And by 2 things, I mean their motor control. So this is a measure from our reaching task, and our position sense, or one of our proprioceptive tasks. So this is position sense variability. And so typical variability is about, you know, 4.5 centimeters for someone who hasn't had a stroke. And so for individual subjects, this is an example from someone who has really impaired motor recovery. And so I think in the original paper, we referred to this as incomplete recovery because they were still impaired at 6 months. So this person had pretty impaired motor function. You know, they improve a bit, but they still have impairments at 6 months. But they never had an impairment in their position sense. So we have intact position sense, but impaired motor control. On the other side, we can also have people who have intact motor control, but completely impaired position sense. And so you can see that this person makes nice reaching trajectories, whereas when we look at the bottom, all you see is kind of a garbled mess, essentially, of data. And that's really what it is, because this person has no idea where their limb is being moved. And so, you know, it's interesting to think about, you know, these different modalities and how they can be dissociated based on the different types of injuries that individuals can have. And so, you know, I was really interested in looking at this from kind of an individual level. And so what I'm showing you here is performance on the motor task. This is the motor task score. It's a composite of a lot of measures that come out of the visually guided reaching task. And we do the same thing for the position sense task as well. So this is essentially, on the left is motor performance, on the right this is proprioceptive performance. And so for this person, both of these blue lines are from the same person on the left and the right. You know, this person actually, they do improve over time. So this is all the way out to 6 months, but they never reach this gray area. And what this gray area is this is controls. This is the control level. For this person who has impaired position sense, what happens is that, you know, they improve their position sense and they actually get down to control levels by 12 weeks. So they're their motor function doesn't really recover, but their proprioceptive function or their position sense does recover by 12 weeks. We have a second example here. And so this person recovered motor function by 12 weeks, but never had a position sense impairment. And then we have this individual who recovered motor function by 6 weeks, but had significantly impaired position sense. And so you see all these different patterns of, motor and proprioceptive behavior depending on who the individual actually is. And so I have examples for days of this, of different patterns of behavior. But the take home of this really is that, there are people who have timelines of motor and proprioceptive function that actually work nicely together, that those two timelines actually match up. But the take home of this is that, there's 30% of these people. And so 30% out of 116 people that we had in the original study actually have motor and proprioceptive recovery that do not match. And what this means is that, you may have someone who's in rehab and maybe they're still a bit clumsy, and that may not actually be a motor control problem. It may actually be a problem with proprioception. And so being able to really, carefully identify these different types of deficits is, I think, paramount to really developing individualized courses of, treatment. And so just to kind of wrap up a little bit, the big take home points from today is that, I use robotics in a lot of my work, and, we're looking at measurements. And so the first thing is that, we see that proprioceptive impairments are quite common after stroke, and we see this kind of complicated relationship with vision. And when we think about the fact that many rehab strategies actually rely on the use of vision, that kind of complicates it a bit more. And so that may, information like that may actually re-guide a treatment strategy or a rehab strategy. And then, the last part of the talk really focused on the relationship between motor control and proprioception. And so, we need to be able to realize that, just because you have an impairment in motor control, that doesn't mean you won't have an impairment in proprioception. And if you have an impairment in proprioception, maybe you don't have an impairment in motor control. And I think that a careful identification of those things will help us develop a bigger snot or a more specific snapshot of what individuals look like after their injuries. And so I just have a kind of a, what does it all mean? And so, I'm a big fan of looking at sensory impairments because I just gave an entire talk about it. I think they're important. I think we don't know enough about them. And so, my lab's really focusing on developing kind of some finer grain tests and analyses for looking at these different types of impairments, and specifically into this idea of sensory motor integration after stroke, because I think that it's actually quite important. All right, last slide is an acknowledgement slide. Thanks to my lab at the University of Delaware. They are a fantastic group. And, this work was done with Sean Ducalo at the University of Calgary, along with Steve Scott at Queen's University and Troy Herder, who's at the University of South Carolina. So thank you very much for listening.
Great. Thank you, Jennifer. I'll clap for everybody. And looks like we have a good crowd who wants to ask questions. Cole Hurwitz says, amazing talk. So I'm going to start with a question. The first question from Katinka Vanderkooy. Thanks for an interesting talk. Why did you choose a matching task instead of, for instance, a discrimination task? Perhaps the discrimination task would be more suitable to study proprioception without motor involvement.
So so that's actually the next steps. So we're actually working on a speed discrimination task in my lab right now, because it's something that I am, I'm not aware that exists, but I think it's something that will be useful and give us some information about, what those thresholds are for different modes of proprioception. And so I know that Catherine Lowry, who's a research associate in Steve Scott's lab at Queen's, she actually just published, I think in 2019, a positional discrimination task paper, which is very good. So I recommend that you go have a look at it.
Right, and so let's see. I guess Aaron Wong has a related question about doing a matching task to assess proprioception, and are there downsides to, how that might equivalently assess proprioception on the ipsilesional side, i.e. to confirm a lack of impairment? So I guess the question is more about how does in locomotion, we say less affected side affect your results?
Well, and I think that, so I think there's two questions here. I think assessing proprioception of the ipsilesional side is exceedingly difficult in people who have impairments on the affected side. And so one thing that we've done in all of the papers that we've published is we actually assess for motor impairments, so ipsilesional impairments, through a variety of clinical and robotic measures. And so every paper that we've published, I've actually taken out everyone who had a score below perfect. So we typically use the CMSA, which is the Shidok-McMaster for arm assessment, and we use the visually guided reaching task to assess for some motor issues. And so we take those people out if they do have an ipsilesional impairment of sorts, and rerun the analyses. And there has never been a difference between leaving those subjects in and taking them out.
Okay, and we have a question from JJ Gorban. I can't say your last name because it's not here. I'm sorry. Jean-Jacques, would the motor task not be more linked to proprioception if there was no visual feedback during the reaching movements?
Yeah, and so that's actually, so everyone's ruining the surprises that are coming down the pipe.
No, we're just anxious to know about them.
Yeah, well, and that's, and that's one of the things that we have been working on, is looking at, how do, not only the proprioceptive measurements that we're looking at, so these kind of active sensing measurements, as well as, discrimination thresholds, as well as exactly what JJ has said in the chat. And so, I think that it's something that I think is important to evaluate, and that really starts to get at the active versus passive usages of proprioception, which I think it's a kind of it is a difficult problem to try and tease apart. And I think we're just at the beginning of really figuring out exactly how these things look in an impaired population.
Yeah, I think that there's so many things to measure. I wanted to follow up on the discrimination task, having done some things like that. What's the relative amount of time that it's going to take you to identify a discrimination threshold versus going through your whole battery of matching tasks? And, what kind of trade-off do you see there?
Well, and so, I think that, this has not been my baby in terms of driving the times down for these tasks, but Sean and Steve have really focused on the patient and, how they are going to experience these different measurement tools. And so, one of the things is that, I don't know how many Sean's doing in his lab now, but that whole battery of tasks, which is probably 8 to 10, take probably 45 minutes. And so the timing of a lot of these tasks are for the position sense and kinesthesia tasks are actually quite short. I'm talking about 3 to 4 minutes. If everyone goes and reads Catherine's paper, they actually look at reducing that the discrimination samples, I think around to maybe 50 movements, where they were able to actually accurately predict kind of what someone's discrimination threshold was. So, and I think that's really useful because, I think in the field of motor control, we are used to doing, upper limb tasks that take, 45 minutes to an hour, and expect, we have willing and wonderful subjects that come in and do these things. But, if you're reasonably thinking about bringing someone in a couple days after they've had a stroke, or maybe even, a couple months after they've had a stroke, they don't want to sit there and, go through a kind of an onerous task for an hour. And so there's a I think there's a big trade-off in terms of, what we're measuring, what questions we're asking, and, the specific design of how we're implementing these different measurements.
So with that, I'm gonna bring a question from Kelvin Jones to follow up. Have you thought about more pragmatic clinical measures that could be more widely deployed to clinics, but was consistent with the robotic assessments?
So COVID is the inspiration in my lab right now. And so what I started working on right at the beginning of this, I had 2 really talented summer students who were working remotely. And so we've actually started working on some tasks that can be easily ported over into the clinic, into somebody's home using a tablet. And so, we're hopefully going to be making comparisons between different devices to see, can we make measurements, can we validate them, and will they be useful to the people that are actually going to be using them?
Okay, I'll go back to the top. Hannah Block wants to know, was there any difference in the lesions of patients who improved with the addition of vision versus those who didn't?
So yes, for the people that did have issues with vision, I think that most of the time, and I'm a little embarrassed that I have to go back to the paper and reread exactly what was in there, because there was a lot of different areas, because the 2018 paper, we actually do some structural lesion analysis. And so, I think that for the most part, those people had lesions kind of centered around, those, parietal areas that are responsible for integration of information, which really, it wasn't a huge surprise, but to find some sort of, neurophysiologic commonality, I think was helpful in terms of trying to narrow it down.
Okay. All right, so Cole Hurwitz says, it seems though most, if not all, assistive or therapeutic brain-computer interfaces for stroke recovery target the restoration of motor control. Do you think your work provides a new research direction for those devices or for new therapies in the form of restoring proprioception?
Yes, yes. So we actually wrote a grant about that because, I think that's, I think that, that is one area that has been a little underappreciated. And I have a graph somewhere floating around that I made looking at all of the publications that focused on stroke and robotics and motor control and proprioception. And so, if I were to make a graph of my hands, the stroke and motor control is, up here and the proprioception is down here. And so I think there's a lot of room to move and lots of room for investigation in terms of, one, how do we use these particular measurements? How do we actually integrate them into a therapeutic system? How do we tap into that? I think that that's a really, really good question. And, if we're thinking about BCIs, that's way beyond my expertise at this point. So.
All right, I'm going to hop to a question that I'm personally interested in from Carly Sombrick. If there are lower limb proprioceptive deficits following stroke, do you expect that vision would reduce impairments in some patients similarly to the upper extremity?
Oh, I don't know. So I'll be honest and say that I attend a very large motor control lab meeting every week with several lower limb motor learning folks, and it just brings to light the differences between, upper limb and lower limb and kind of how different they operate. And I don't like to make assumptions when we talk about upper limb and then port them over to lower limb, because, I think that even the learning processes are very different for, how we learn or how we retrain gait versus how we learn or retrain the upper limb. And so when you think about how we use visual information for each of those, I don't want to refer to them as independent systems, but, for each of those systems, we use vision very differently for gait than we do for our upper limb, because we don't look at our feet a lot of the time. I mean, I look at my feet when I'm, trying to get in and out of the snow to know how much I have to lift them up. But, we typically don't use vision a lot to actually guide the actual body in that case, but we use vision to actually, take cues from our surroundings. But with, an arm, it's constantly in our periphery, and, we are using that information to actually guide where we are. So I would expect them to be a bit different, but I'm not willing to put money on it because anything's possible. So.
Great. And then Friedel DeGroot says, how do you know that impairments in motor are not impairments in using vision?
So how do you know impairments in motor are not impairments?
So if you have a motor impairment and it's not proprioceptive, how do you know it's not like that you can't?
Yeah, well, and so the one thing that we do is that we screen for visual impairments, right? And so we screen for, just regular vision. Can someone see in 20/20 vision? We also screen for field deficits. And we also screen for things like visual neglect.
So there could be, but you're not actually testing for that right now.
Well, and so in the vision study, we do see that a lot of folks that have neglect can't improve with vision, but people with neglect are their own kind of special storm of impairments. But the important thing there is that we actually have a lot of people who don't have a field deficit, who don't have neglect, that actually show these kind of weird vision impairments. So, I don't think it's a problem with sight. I think that's what you're trying to get at, and rather there's a miscommunication between those two different forms of sensory information.
Okay, we are about out of time. I'm going to ask one quick last question from Kelvin Jones. Did you examine your outcome measures at week 1 that on an individual basis that were prognostic for recovery?
No. So that's a very short answer. Yeah. Okay. So, yeah. And so with
maybe
that,
it's coming.
What? Sorry?
Maybe it's coming.
Well, and so, that work that I was doing really we weren't trying to make predictions at that time. And so there's a lot of work out there that has tried to make predictions. And I think that there's some use in that. And I think it takes some far more sophisticated skills than I have in terms of, implementing computational methods to really get a nice predictive picture of what recovery actually looks like, or will look like, I guess, if we're predicting it.
Well, thanks. I think our time is up, and I really appreciate your talk. It was really, really food for thought and clearly generated a lot of discussion, and everyone's giving you applause. Thank you.
Yep.
Looks like a lot of the same people from the last time. It's good. We had an upper limb, and now we're doing lower limb.
Yeah.
Okay, I'm going to give it a few more seconds while people come in. Numbers keep going. Okay, well, I'll get started just by introducing you. Welcome, everybody. I see a lot of the same faces, or not faces. I'm imagining your faces, but I see your names from the prior session. And I want to remind everyone again that the chat can be seen by everyone, and it's great to introduce yourselves or to talk amongst yourselves. If you have questions directed at the speaker, please put them in the question and answer session. You can vote them up and down. And at the end of the talk, I'll be reading them off in order of the most votes or my own personal preference. Okay, so today it is my pleasure to introduce my colleague, Tricia Kazar, who is now an associate professor in the Department of Rehabilitation Medicine in the Division of Physical Therapy at Emory University. She directs the motion analysis lab there. And today she's going to talk to us about biomechanics and neural correlates of post-stroke gait retraining. Okay, with that, Tricia, take it away.
Okay, thank you, Lena, for the introduction. And I'll start by saying I'm really excited to be here. And thank you to the organizers for inviting me. Before I dive into the science, I'll start out with the most important stuff, which is giving credit where it's due. So I want to thank my collaborators and colleagues at Emory, especially Dr. Boric, Lena Ding, who's right here, our MC, Steve and Gordon, Gordon is an emerging collaborator and Steve's a long-standing collaborator. And of course, trainees and personnel in the lab, especially a shout out to Alejandro Lopez, who's a PhD student, and he's been instrumental in a lot of the neurophysiology data that I'll present. And of course, last but not the least, funding agencies and funding sources. So our lab engages in translational research, and the population that we study is people with chronic post-stroke hemiparesis. As you can see with some of the numbers on this slide, this is an important and leading cause of disability. Immense medical cost is incurred because of a stroke. We're doing a much better job with acute management and survival after stroke. However, there are still at discharge from rehab residual deficits and persistent disability with regards to walking, and walking function after stroke is the primary focus of our lab. And happens to be World Stroke Day is coming up this week, so that's a good coincidence. And with regards to rehabilitation of walking after stroke and recovery of walking function after stroke, there are several gaps in the literature. There has been a lot of research, clinical and neuroscience research, and there have been RCTs done comparing interventions. However, the mechanism underlying these interventions, and more importantly, why they work, if they do elicit a positive response in terms of improvements in walking, and for whom these interventions are best suited, is largely unknown. So that's the thrust of the questions that we ask in the lab. And this quote here from a recent stroke review textbook does a nice job of also emphasizing the need for, instead of starting out with RCTs or clinical studies, going in with a mechanistic foundation and understanding the neural and the biomechanical correlates, which is essentially the title of the talk today. So I'll talk about some of the examples of neuromechanics mechanisms that we've probed with regards to stroke gait, both understanding and treating gait. So our approach includes evaluating the biomechanical correlates of stroke using kinematics, kinetics, as well as, of course, measures of clinical walking function. We also study the neurobiology using non-invasive stimulation techniques to probe neurophysiology. And we use neuromechanics measures in context of clinically relevant treatments. For example, electrical stimulation or biofeedback are the two examples I'll use today. So starting out with a focus on the biomechanical correlates, just as an overview, stroke impairments in gait after stroke are profound. We see deficits that affect all phases of the gait cycle, all lower limb joints, and it's a complex problem because of the multitude of deficits, and because even starting out and evaluating which of these impairments should we focus on, whether it should be swing phase, stance phase, ankle, hip, compensations, etc., is a complex or is still fairly poorly understood. And objective decision-making criteria for treating and diagnosing stroke gait are heavily required in the literature. One of the biomechanical impairments that we study, and is the focus of a lot of the interventions I'll talk about today, is decreased paretic propulsion. So this figure here shows ground reaction force data from a force platform embedded within a treadmill. The green is the paretic leg, which shows a decrease in the forward push-off or propulsive component compared to the non-paretic leg. And there's a spectrum of disability. Different people will show different magnitudes of the deficit, but this is very common and highly prevalent in people with stroke. The reason we see this deficit in propulsion is, of course, because of decreased force generating ability, weakness, and lack of coordination in the plantar flexor muscles, which are the primary muscles that generate propulsion, and also inappropriate positioning. Trailing limb angle is a term we use in the biomechanics literature, at stance to swing transition of the paretic leg. The reason propulsion is important is because a series of cross-sectional studies over the past 10, 12 years have shown that this decreased propulsion is related not just to the stance phase deficit that it is, but also other swing phase deficits, such as decreased knee flexion, slowed walking speed, which is an important functional outcome and treatment goal after stroke, as well as asymmetries in gait. post-stroke. So we focus on this decreased propulsion and try to target it using different kinds of treatments and use them as paradigms in turn to study the neural and the biomechanical changes that underlie these interventions. In terms of the neural correlates, we use non-invasive stimulation, like I said before, and this schematic just gives a rough overview, a cartoon version of the corticospinal tract and some of the primary circuitry for lower limb motor control. So we use non-invasive transcranial magnetic stimulation, or TMS, to evaluate corticospinal tract output using MEPs. Ankle muscles are the target muscles because they're the target for propulsion as well as for the interventions in terms of therapeutically the stimulation or the biofeedback. And a lot of research has already shown this, and our work just confirms that in people with stroke, the paretic or the lesioned corticospinal tract shows a decrease in MEP amplitude or overall excitability. In addition to TMS, we also use peripheral nerve stimulation, or PNS, to probe spinal reflex excitability using the Hoffman's reflex or H reflex amplitude. And this gives us a readout of the 1A alpha motor neuron segmental reflex circuitry. Again, similar to MEPs, H reflexes in stroke have been studied previously, and there is an increase in H reflex amplitude or hyperreflexia, and that's one of the sequelae post-stroke. In addition, with TMS and PNS, we also probe paired stimulation approaches at a range of interstimulus intervals to evaluate the descending projections between the cortex and the spinal lower motor neuron or spinal reflex circuitry. And unlike TMS and H reflexes, the effects of stroke on these descending projections, direct, fast conducting, as well as indirect projections, is largely unknown. So our work is interested in evaluating how stroke affects these different parts or different levels of the neuraxis, and how these neurophysiological correlates at these multiple levels of the nervous system respond to treatments. In terms of the gait treatments or approaches that we use to study as intervention paradigms, like I said, I'm going to give examples of work with FES or electrical stimulation, as well as biofeedback as treatments that target propulsion. So this just gives you a collage of the number of different kinds of interventions and treatment approaches that are used in the clinic. We'll use FES, treadmill training, and biofeedback as one example. So starting out with the fast FES intervention, just to give you an overview of what the intervention comprises of or what the active ingredients are. This is high intensity, fast speed treadmill walking. So there's bilateral stepping practice at fast speed, combined with functional electrical stimulation that's delivered to ankle muscles. And the muscles we stimulate non-invasively, again, during the gait cycle, are dorsiflexors to restore foot drop and plantarflexors to increase paretic propulsion. This has been fairly well studied when it comes to the biomechanical and the functional effects of a 12-week intervention. But the neural correlates of fast FES are poorly understood, which is probably common for a lot of other gait interventions as well. So this intervention provides targeted practice through stimulation that's delivered just to the paretic limb, to the paretic ankle muscles, and it's been shown to increase in the short term and long term propulsion and propulsion related variables, as well as improve overall gait function. But the missing link, or the hypotheses that we'd like to test, is if fast FES induces a preferential increase in the corticospinal excitability and normalization of segmental circuitry at the spinal cord in the lesioned corticospinal tract, which in turn is the source for, or the mechanism underlying, the biomechanical and functional benefits of fast FES. The control treatment that we use is fast walking, which is dose matched and intensity matched, except there's absence of FES. So it's good, it's still an evidence-supported intervention, it's high-intensity treadmill training, but it's not targeted to the paretic leg or to specific deficits on the affected limb. The recent study that we completed for evaluating the neural correlates of fast FES was on individuals with chronic stroke, and we used an AB-BA crossover design. So we have a pre or a baseline evaluation of neurophysiology, gait biomechanics, and walking function. And then the individuals get randomized to either 3 sessions of fast FES stimulation and 3 training sessions 3 days a week, or fast walking, which is the same treatment without FES. They complete a post-eval of the same outcome measures after 3 sessions of training. And then after a 3-week, at least a 3-week, much longer in some individuals, washout, they cross over to the other kind of training. And then we again collect pre and post data. So this is a repeated measures design, which helps us take account for or reduce the effects of inter-individual variability on the training-induced changes in neurophysiology and biomechanics. From this study, before I dive into the results, actually, this just gives you a schematic of each training session, what it entails. So things to look out for is that they're very well matched in terms of structure, dosage, and components. There are 6 training bouts. The only difference is, it's the same individual, similar speeds for training, and the only difference is the presence of intermittent FES for the fast FES group. So the results from this crossover study showed we evaluated soleus and TA MEPs bilaterally. And I'm just showing data for the paretic soleus and TA here, just to save time. This shows raw MEPs from an individual's paretic limb. The before versus after fast FES, we see an increase in MEP amplitude, but with fast, the same individual does not show an increase in MEP amplitude. So we don't see that increase with fast training. Group data and individual subject data show, again, more consistent increases with fast FES post versus pre-training, and more variability or a decline with fast. When we look at soleus MEP amplitudes normalized to baseline, so here, this is just showing individual subject data, and an increase would show an upslope, and a downslope would show a drop in MEP amplitude. So a majority of individuals, because it's a small sample study, we can look at and show an increase or upregulation. For TA MEP amplitude, again, a majority of individuals show this upregulation. So in summary, when we also didn't see, and I didn't show that, similar changes in the non-paretic leg. So we're able to target the lesioned corticospinal tract and show preferential increases in both the muscles that were targeted through training on the affected leg. This slide gives you other examples of some summary variables derived using TMS. So now it's delta change in MEP amplitude for fast FES versus fast. So a bigger number above 0 would show an advantage or greater benefit of fast FES for the same person. So for seated, TA MEPs collected in seated, intracortical facilitation, as well as soleus MEPs collected in both seated and standing postures, we see this consistent effect of more individuals, a majority of people are showing a benefit of fast FES. So transitioning to changes induced in spinal segmental or H reflex data with the same 3-session data set. This shows pre and post H2M ratios on the top and H2M slopes at the bottom panel. And you see pretty much lots of variability, but there's no consistent change from pre to post for fast, which was our control. For fast FES, there's still considerable variability, but there was a significant increase in H2M ratio and the slope after versus before training. When we look at the delta, though, it's not statistically significant. So there's no between intervention significant effect. So we're not confident based on the current data. We're still continuing to probe and analyze this, but we think that a larger sample and more robust, maybe greater dosage study is needed to try to parse out the effects of this intervention on corticospinal versus spinal segmental circuitry and how that influences its effects on gait. The paired stimulation approach that I talked about, I'm not going to show you the fast FES effects data because we did not actually see a change in SLF or LLF. But like I said, this hasn't been studied in stroke before. So here we're just showing examples of stimulation pulses, PNS alone, which elicits an unconditioned H shown with the bold line, and the paired conditioning, which is short interval or short latency facilitation, SLF, and that conditioned H reflex, the larger amplitude, compared to the unconditioned, that ratio is used as an indirect probe of the strength of descending circuits, both direct fast and indirect slower conducting. So this long latency facilitation, long interval facilitation is the measure of those indirect slower pathways. And in people with stroke, we found compared to able-bodied controls, a reduction in both SLF and LLF. And this is work that's part of Alejandro's dissertation, as well as a product of collaboration with Dr. Boric's lab. So a shout out to both of them. So we're still studying this. There still needs to be more work done, but there seems to be an effective stroke on these pathways, and especially the long latency pathway shows some nuanced differences in that it's not completely obliterated. There's a reduction, and there's some evidence for ipsilateral or from the contralesional cortex longer latency effects as well. So this is certainly something we're continuing to look into. Finally, because we're interested in stroke gait, biomechanics, and function, we also want to know if the neurophysiology metrics are related to gait impairments and walking function. And this just shows 3 examples of scatter plots of those 3 levels of the neuraxis that I showed data for, that people who have more impairment in neural circuit function, so smaller MEPs or lesser ICF, intracortical facilitation, or a higher H2M ratio, also show more impairment in either their biomechanics or walking function as measured by gait speed. This is a more recent study with Dr. Ting's lab, where Jessica Allen, who's now a faculty at West Virginia, looked at EMG, changes in EMG activation at a range of walking speeds, and this was overground walking after versus before fast FES training. And here we looked at 2 responders and 1 non-responder. The reason this is important is because no intervention, like none, is going to be a one-size-fits-all and effective on all individuals. And it's important to understand what are the factors that correlate with non-response. So in this case, Dr. Allen's study found that individuals who showed a decrease in soleus TA coactivation, so more normal EMG activation during the gait cycle, were also the ones who were responders and showed a greater improvement in walking speed, but non-responders actually showed a slight increase or worsening of coactivation. So again, another direction that needs to be established more concretely with longer term studies and larger sample studies. So to summarize, when we evaluate the neural correlates of fast FES as a probe and build upon the biomechanical or clinical data that we have, it's messy. It's not as straightforward, but there certainly is evidence for, preliminary evidence for, an increase in lesion corticospinal tract output, some modulation of spinal segmental circuitry that needs to be further studied, and also using EMG data as a probe for this, again, neural mechanisms, and understanding what are multiple, using all these measures, how does fast FES work? And can we now predict, using some baseline neurophysiology or baseline biomechanical data, the long-term response for an individual before they start out with this intervention? Switching to biomechanical correlates, I'm going to talk, this is less detailed, a little bit more brief, but we'll see. Hopefully that works out. So the biomechanical correlates that I'll talk about just focus more on interventions, biofeedback as one example, and how they influence multiple biomechanical variables in people with stroke. So before I talk about biofeedback, I'll illustrate one example of a study that looks at a much simpler and much more broadly used kind of feedback, and that's verbal feedback or verbal instruction. So in the context of that same intervention, fast FES, we asked the question whether or not adding individualized verbal instructions by a neurorehab clinician augment the motor learning that's observed by one session of fast FES training. So this shows the same schematic, fast FES training. The control was now fast FES, and we compared it to fast FES with incorporation, with addition of verbal instructions in a faded format throughout the training session. And we tried to give some guidelines to the clinician, but it was not prescribed feedback. So we tried to mimic clinical setting feedback while trying to keep it as organized and robust as possible. And in response to a single session of training where we looked at both within session effects as well as retention 24 hours later, fast FES with verbal feedback, which is FF plus PT, showed a bigger increase in propulsion and propulsion related variables. We also looked at ankle angle at initial contact, which is AAIC here, and that wasn't as responsive. But certainly the push-off variables in TLA showed an effect. This shows estimation plots for the same data, but now we're looking at within session change scores on the top, retention change scores on the bottom. So a positive value would show an improvement. And we found, even though again it's a small sample study, moderate to large effect sizes for an advantage towards fast FES with PT feedback incorporated. So this verbal feedback that's heavily used in the clinic, one observational study shows once every 14 seconds does seem to enhance motor learning when added on top of systematic or controlled intervention. The second kind of feedback that we've studied in our lab is real-time gait biofeedback, which we can think of as a more high-tech, objective, quantifiable version of that verbal instruction or verbal feedback. And in this case, the goal is to provide the user or the stroke survivor information about biomechanical variables in an overt, timely, and meaningful manner to modify that targeted variable, to induce a change. This gives a video example that I'll just let play in the background. So we have the person walking on the instrumented treadmill. There's a feedback visual display, which is very simple, kind of boring almost. And the beep is the audio component of the feedback. So when they have a successful step, when they reach target, the feedback gives them an audio cue, and they constantly see the visual equivalent of their push-off forces on the screen. We looked at the feasibility of this feedback on able-bodied individuals first, and we found that for the targeted variable, which was in able-bodied people, their right leg, they showed an immediate increase in push-off as soon as biofeedback started. And for a short retention, 2 minutes retention test without biofeedback, they were able to maintain that increase. So it's able to induce a change in the targeted variable without changing the non-targeted variables. This is a unilateral feedback, and that would be promising for people with stroke who have that baseline asymmetry in their gait. So the effects on stroke gait, these data here show peak AGRF in response to biofeedback, pre-training, and then 3 retention periods after 1 training session. All these tests were done without feedback. In between, we had 3 6-minute bouts of AGRF feedback training using that audio-visual paradigm. The filled symbols are the paretic leg, and they show an increase that's maintained at the 30-minute retention. The non-paretic leg, which is the open symbols, stay flat, they don't change, and that's an advantage because that leads to this deficit or interlimb asymmetry reducing after biofeedback training. We also find trailing limb angle shows a similar effect, as well as plantar flexor moment, step length, so some of the other variables that would expect to be related to push-off. Biofeedback about limb position versus force generation is the recent study that we did. So instead of using AGRF as a target variable, here we're using TLA or trailing limb angle as the biofeedback target. And again, the format of the feedback is very similar, but we're trying to compare the effects of, compared to no feedback, AGRF given as a target versus TLA. And for AGRF, they both seem to be equally effective. When we look at TLA, there again is an equal effect of both interventions, but TLA feedback has a slight advantage significantly compared to biofeedback for targeting that TLA variable. TLA feedback is more promising because the wearable sensors and even visual observation, a PT or a clinician can assess trailing limb angle more easily than push-off, so it might have more potential for translation. Moving on to the next step in terms of the biofeedback, we have an ongoing project that's trying to gamify the biofeedback interface to make it more engaging, fun, to give this score that gives knowledge of performance or knowledge of results, keeps track of number of successful steps, and hopefully that'll help enhance motivation and further improve the long-term efficacy of biofeedback as a gait training paradigm. So to summarize, in terms of the neural correlates that we used fast FES as a paradigm for, there's still a lot of work to be done, right? So the ongoing work we're doing is at the beginning stages of looking at a five-year project to evaluate 18 sessions of fast FES, and before, during, and after training with fast as a control, looking at neurophysiology, biomechanics, clinical function, as well as energy cost as multimodal outcomes for these interventions. In the long term, by understanding the neurobiological correlates, we'd like to develop neuromodulation adjuvants to fast FES that are based on where is the best place or target in the nervous system to further enhance therapeutic effects, as well as baseline predictors of long-term response, so we can have a better decision-making criteria for whom fast FES is best for and who might be better off with fast training. For biomechanical correlates with the biofeedback work, again, there's lots of next steps, and some of them Dr. Ting's lab is collaborating with us on. So a lot of this work has focused on push-off or TLA, so certain individual variables, and we're interested in having a more holistic or comprehensive analysis of gait and how it evolves or changes, as well as including progression and speed as variables on top of that biofeedback paradigm. So using these approaches, our long-term goal or long-term vision is to develop individualized, precise approaches that can enhance both gait function, gait speed, endurance, and also gait quality. So we can have this optimization of speed and gait quality in people with stroke, and in the process, maximize quality of life of individuals with stroke. I was debating if I should include this, but I had this made, a collage of 2020 and all the things it's been. But I think the one thing, despite all the pandemic and all the stuff that's going on, that worries me at least, and this is just like me saying this because Neuromatch is a scientist and forum of experts, I don't know how we can establish the validity of facts and expertise again, and what we can do as citizens and more importantly scientists to sort of get that back as the data are what matter. But anyway, so that's more of an open question or almost lament. But again, thank you, and I'll field all the questions that you might have.
Great, thank you. I'm clapping for the audience, and I'd like to encourage the audience to go ahead and put their questions in the Q&A box. I'll start by just looking at these results, Tricia, because I haven't actually seen them in this format. Was the TLA feedback, the trailing limb angle feedback, actually better than the push-off feedback? Yeah. On push-off. So you're telling them one variable and then they actually do the desirable one better. And do you have some ideas about why that might be?
Yeah. So equally effective, but TLA in especially people with stroke was more effective, not in the controls, was more effective at increasing TLA, the targeted variable. Also, the one interesting finding, which again needs to be corroborated with larger sample and more sessions of feedback, was that in response to TLA feedback, there was less unilaterality of the effects. They showed bilateral increases in TLA. With AGRF feedback, there was a unilateral, only the paretic leg increased AGRF. So there were some minor nuances which might—
Wait, what?
Yeah. So TLA wasn't as unilateral in its effect.
So if you tell them to improve trailing limb angle of the paretic leg, they also increase it on the non-paretic leg.
Yeah, so there was a significant increase in stroke survivors of the non-paretic TLA, and it could be that it was a difference in interlimb asymmetry in TLA versus push-off, that how they're dependent on each other more so than push-off. So those were the two key differences. But when we look at just push-off, they were both equally effective. There was no inter-feedback differences for people with stroke.
That's weird. Do you think that means that we have some internal symmetry generator?
It could be. It also brings back this notion of, and you've discussed this, about dependencies and interdependencies amongst variables. And feedback can be a good probe to evaluate that, because we can try to change one thing and see how it cascades into other variables. And if one causes one effect, the opposite might have, the differences between what you're targeting and what the secondary effects are might vary. And that can help us inform more tailored feedback strategies, maybe.
Interesting. Let's see. I know many of you have questions out there, so put them in the chat. I actually want to go back to the findings, the neural correlates. So you said the corticospinal excitability improves after fast FES.
Yes. Yeah. From the 3-session study.
So why?
That with longer training sessions.
I mean, did you expect that? Because if you're really stimulating peripherally, you might think that you were doing some kind of spinal upregulation, or I guess it should be downregulation. And does that mean that, talk to me about where the locus of that improvement should or is happening.
Yeah, so I think in terms of whether we expected it, that was part of the hypotheses that we would expect to see upregulation of corticospinal excitability. And we wanted to see if this upregulation would be unilateral or more preferential to the lesioned hemisphere or the lesioned corticospinal tract, because that's the one that's getting the stim. The other leg isn't getting the stim. And what you asked before about the locus of change or the mechanism, non-FES, like somatosensory stimulation or other kinds of just sensory level stim, has been shown because of that barrage of sensory input, which in turn causes upregulation of motor output, to increase corticospinal excitability. So tense and somatosensory input is already known to be an upregulator. And in this case, our hope, or we posit, that when you have task specificity, when you add that motor component and the practice of the movement that's occurring concurrently with the stim, that should actually have an even bigger effect on CST excitability. There are some studies that speculate that aerobic exercise also induces plasticity. So the control intervention should also might also cause a change in corticospinal excitability, but that might be more bilateral. It wouldn't be specific to the paretic leg.
I see the questions, but I had another thought, which was, if people are learning early on, right, do you think that you're increasing their voluntary control of that leg, and then after they learn it for a while, like maybe do more sessions and maybe that moves downward? Do you think that you would be able to test that with your neural correlates?
Yes, I think that would be awesome if we could parse that out, the time course of changes and how they might evolve or shift from spinal to cortical levels, because that's one of the implicit goals of the study, to not just measure before and after 18 sessions of training, but every 3 sessions or so, and see which one starts to show changes or modulation first, and how they stabilize or evolve over the course of training.
I hadn't thought about that hypothesis before. Okay, well, Jasmine Mirdamati has a question of, for the neurophys studies, how many patients were MEP positive? And do individuals with or without an MEP show differences in spinal excitability?
Yeah, that's a good question, Jasmine, because especially for lower limb TMS, MEP negative or eliciting MEPs is even harder than upper limb. So we used a batwing coil, like a specialized coil for lower limb, and we, at least for TA, so LEAS it's fewer, but we were able to get MEPs for a majority of the individuals with that specialized coil. So if I had, I think 3 out of the 14, and the data I showed was for people where we could elicit an MEP at baseline. It might not be 50 microvolts. That's discussion we've had with Dr. Boric back and forth. But we had active condition, resting conditions, standing MEPs, and that increased our chances of eliciting a response, even if at seated rest we couldn't. So we tried to do a range of tasks and record MEPs from those tasks, and we only looked at the changes in people who had MEPs. So that would be an interesting follow-up to look at with the larger sample study to see if presence or absence of MEP, how that changes the long-term response, the neurophysiology response. That's an open question, I think.
So you're saying that you didn't do the H reflex if people didn't have an MEP, because then you couldn't do the conditioning?
Well, that's a good point, Lena, because we were trying to get the best data set we could and make the most of every participant who walked through the door. If we couldn't get MEPs, or if they were actually contraindications for MEPs, we would still do the spinal excitability component and 3 sessions of fast FES. So there were differential cohorts, some people that had just H data, some had TMS, H, and paired pulse data. So
right, so the question was, with if they didn't have an MEP, was their spinal excitability, so their H reflex response, different from the group that got it?
Well, now I get it, yes. So the short answer is there was a relationship between not so much MEP plus or minus, but we looked at the relationship between the size of MEP amplitude and the H to M ratio. And it was an inverse relationship. So people who had high corticospinal excitability tended to also show worse or hyperreflexia in spinal circuitry, if I remember correctly. So there was a relationship, but we didn't look at it with respect to positive or negative.
Oh, and so does that mean, do you think there's an inhibitory effect of the descending control or
Yes,
excitatory?
So this whole notion of the stroke causes disruption of descending projections, which in turn disrupt the inhibitory normal inhibition on spinal segmental reflexes, and that in turn causes hyperreflexia. So some of those relationships did match that overall hypothesis. However, I think there was another question about examining inhibitory pathways in the spinal cord. I think it's still complicated. Like we have these multiple probes, right? We have TMS, intracortical facilitation, H reflex, and we can look at paired pulse, the TMS and spinal pairing. And even with that, there's going to be questions that remain, right? So reciprocal inhibition, presynaptic inhibition, if we had enough time and resources, and we have a 3-hour neurophys session before and after as a function of that training. So I think the challenge is fitting in those extra measures and still not having fatigue or the time course of the session influencing them. So ideally, the follow-up of that follow-up study should be even more zoomed in view at intraspinal inhibition, presynaptic or descending inhibition, Renshaw cells. Like there's a whole array of follow-up probes that we could, maybe this is a good way of starting out with where is the signal, where's the locus of change, like Lena asked, and then we can follow up with now a more fine-tooth comb to see even more of the zoomed in view of that change.
Great. So I thought your gaming thing was hilarious.
Yeah.
In
a good way or a bad
No, it was
way?
great. I was wondering why your biofeedback goes horizontally. I would just naturally think about it going vertically.
I agree. I think the first biofeedback, the old one, was Motion Monitor was the package we used, and that was the default, that we were just happy to be able to do biofeedback in real time, and we didn't try to play around or modulate it. It doesn't allow much in terms of range. You can do bar plots, but that zigzag tracking is horizontal. And I do agree, especially for people with stroke, even though in our preliminary, the biofeedback study, they didn't seem to have any trouble with following instructions and matching biofeedback, that could cause a mismatch, because ideally you want something that goes forward backward in sync with your leg, which is moving in that forward backward direction. So there could be some follow-up interesting questions about what's the best way to provide feedback that's intuitive and matches with the task, and does that in fact affect someone's interpretation and success with biofeedback.
Right. And so when you went to this hammer with the exploding balloons, is it a binary thing, or does it give some feedback as to the amplitude of whatever variable you're tracking?
So the goal, the vision is, and it's still like the binary part works, but the goal is to have the movement of the hammer, which again, I think it's the backward forward, which matches it's backward forward, the movement of the hammer to give an indication of the excursion that the person's AGRF is going through. So ideally it needs to be both the amount, how you approached that push-off, and then whether or not you achieved successful increase in push-off.
Okay, fun. Great. Well, with that, our time is about up. If there are no more questions, I'm going to thank Tricia again for a really interesting talk, and thank the audience for being here and for their questions as well.
Yeah, thank you very much for having me.
A postdoc researcher at Conrad Coding's lab, and I'm very delightful to have 3 amazing speakers today that are talking about the visual system. I'm really looking forward to hearing their talk. This is an interactive talk, so each speaker have like 5 minutes to talk, and then you can bombard him or her with your questions. And feel free to use the Q&A button or chat to get involved in the whole discussion. Remember that no question is dumb and everybody is welcome here. Okay, let's first start with the first speaker. We know that the visual system is a feedback loop. You constantly get information and you comment with your eyes, with the saccadic eye movement to get more information. Like right now that you are looking at the screen, you're not just looking at me, you're probably going around the saccadic eye movement to get as much as possible the information. And there are a bunch of studies that they present like a static image to animal and get information that way. But how about the natural case, in the natural settings? I have the first speaker, Atanas, that he's working on this question, and he has an interesting setting to investigate this question. Atanas is a PhD student studying neurobiology at City University of New York. He's computationally focused and interested in the mechanism of how we experience the world throughout the perception and interaction, specifically in the visual perception and action. He is talking about the pre-saccadic stimulus affects post-saccadic neural activity during realistic visual search. Thank you so much, Atanas, and we are looking forward to hear your talk.
Thank you. I'm going to share my screen now. Okay. Welcome to my talk. My name is Atanas Stankov, and as my title was said, it's pre-saccadic visual stimuli affect post-saccadic neural activity during realistic detection task. I only have 5 minutes, so I'll go quickly, and please ask any questions afterwards. Okay, so in this, I have this task where people were sat in front of a computer and they were asked to do a target detection task in a military setting between threatening and non-threatening targets, while we recorded their brain activity using EEG and recording their eyes using an eye tracker. So in this task, the people were driven through a city and they had targets pop up, which they had to identify as threatening or non-threatening. In other parlance, they're either targets or distractors. And here I'll show you video of all the subjects' eye tracking and what the task looks like. So people are being driven through this environment, targets pop up, people shift their gaze, and through button response, they say whether a target is threatening or non-threatening. Okay. So with this task, we had two hypotheses. Target revoked potentials, that is people's brain activity when a target is presented, is modulated by visibility conditions. Either people see their target clearly or in a foggy, or where the target is relative to people's gaze, whether it's in the central part of their vision or in their periphery. And this was determined by looking at person's gaze and where the target was at target presentation, and then using the median of the distribution to say whether it's central, i.e. closer, or peripheral, i.e. farther away. So here I go into the data of people's behavioral and neural responses for all the trials. Just to note, panels A, B, and D are target onset locked, and panel E is saccade locked. So at target onset, the target is presented. Naturally, people saccade to the target. So in blue and A, you see the majority of eye saccades, and then the corresponding button responses. And then in gray is all the saccades that we chose for analysis. B is people's brain activity during target presentation in all conditions, with interesting peaks around 180 milliseconds, 305 milliseconds, and also at 750 milliseconds. Naturally, people moved their eyes faster towards the target. And also for saccade responses, there's two interesting time points at 125 milliseconds and 195 milliseconds. So then we go into ask the question first, what is the brain's response when a target's present, when a target's not present? So here in A is the difference between B, when a target's present and people saccade to it, and then when a target's not present. So you'll see two interesting time activations, one between 0 and 150 milliseconds, and then anything afterwards, which is attributed to button responses in panel D. But for the first two time points, I showed topological plots showing a neural increase when a target's present and a person saccads to it versus when a person saccads to anything else and the target's not present. There's a occipital increase at 125 milliseconds and a corresponding central brain pattern increase at 195 milliseconds with a depression in the occipital region. Now, one could say this is, maybe because people are moving their eyes faster during spontaneous saccades versus not target saccades. But interestingly, people move their eyes slower during target saccades. And in panel G, I also want to show that people's gaze diverts laterally when a target's present at 200 milliseconds into the saccade versus when they at 0 when they start saccading. Okay, so how much time do I have?
You have 2-3 minutes to go.
Great, thank you. So the first question is, how does target visibility affect behavior and brain activity? Naturally, people saccade faster to the target when the conditions are clear and saccade slower to the target when the conditions are not clear. And this has a corresponding target locked activity. And for those two time regions I talked about, 180 milliseconds and 305 milliseconds, there's an increase in activity and with some frontal decrease at 305 milliseconds. Naturally, also people's button response was faster when the conditions were clear. And complementative to the target locked data, there's a saccade lock evoke response difference with a decrease at 305 milliseconds and an increase at 195 milliseconds. Subjectively, people move their eyes faster in the clear condition, though that is not significant. I wanted to show that. And then the saccade lock difference is attributed either to a shift in activity or an actual difference in magnitude in activity. So channel 28 represents the upper half of panel E, and channel 48 represents mostly the bottom half of panel E. And then when it comes to target location, people do saccade more and faster to peripherally presented targets. And interestingly, they do not, that does not affect when they respond to it. So where the target pops up does not influence target response time. And for the most part, neuroactivity is inconclusive to where the target pops up in their vision. Though people do perform better when the target's presented in the center of vision versus the periphery. Let's see. And with that, I would like to thank my lab, my PI, Lucas Pera, and then my corresponding fellow lab mates, Jason and Max, and also the collaborators at the Army Research Lab, Jordan and Stephen. That I conclude with any questions or comments. Thank you.
Thank you so much, Atanas. That was a great talk. We have a Q&A and the chat to discuss. Feel free to ask the question, or if you want to be on a screen, you can ask to be on a screen. There's the one question I see. Do you see any ramping activity leading to the saccades?
So if this is more specifically any activity prior to saccade onset, no. I did not see any ramping. There was some differences, but I don't, they were too early. They were before the baselining that I used. So no, I would say no.
Yeah, it was a little bit like at first that you were presenting, I thought that's the reverse. I thought that like it's foggy. Probably the peak of the saccade is higher because we might search for the in the screen, like more curious and like keen about the small changes. But as you see in the one of the presentation, is lower than the case that it's clear. Is that right?
Let's see. Can you be a little more exact?
Yeah. So like the peak of this red and blue one are for the foggy and clear one, right?
Yes.
And that was a little bit like controversial for me. Like that's like I thought that the blue one should be higher, but that's interesting that you find that's a reverse.
Are you talking about the neuroactivity or the histograms?
The histograms.
Oh, yes.
The first one, yeah.
Yes, people do find. I want to say that people find the targets later. Yeah, so that's why they saccade later, and people find the targets earlier, so the concentration is sharper for the clear. But I'm curious, why do you say that you expected for the fog the peak to be sharper?
No, so that is the time to find them, right? That's not a neural activity. I thought that's a neural activity. I'm sorry.
Oh no, that's when they when the person actually saccaded to the target.
Yeah, sorry.
Yeah.
There's a question I see. Julie asked that I have two questions. Did you see a modulation of the signal or saccade reaction times by target tweets levels?
And then there's a follow-up.
Yeah, the follow-up is that for the visibility, target visibility, did you look at the fixation time? I wouldn't expect to change the saccade reaction time, but maybe fixation time would increase or decrease for the information processing times.
No, I did not see a difference in target threat levels. I mean, it's either threatening or not threatening, so there's no like spectrum to it. So but I did not see changes there. And for saccade fixations, the method I use for saccade detection is good for saccade detection, but not necessarily fixation times. So I can't make any comments on fixation. I don't have the ability. The data is not allowing for that kind of. I would say though that honestly I don't know much about. So there's saccade times and there's fixation times, but they generally follow the same trend. I just don't know the nuances between the two to give a better answer, so I apologize.
You use the same object for all of them, right? Or does the object change?
The object changes. If I go back earlier.
And does it change the reaction time?
Oh, I see. The question is whether the object type changes the reaction time?
I guess part of that also might be object-based. I'm not sure. Julie, if you want to clarify your question, we'd be happy to hear.
Oops. I can't navigate through Zoom, so I apologize for switching the slides.
No worries. Actually, we don't have that much time. We have to wrap up in a few seconds to go to the next talk. Sorry that we can't completely answer this question. If you want to ask a question, I also write down the Twitter account of Atanas, and you can also get the email address if you want to follow up. But let me upload for you. Thank you so much. That was a great talk.
Thank you.
And let me move on to the next speaker. Thank you again.
Thank you for having me.
Okay, so the first time that I learned about the color blindness was the time that I was submitting a paper in a journal, and they told me that you can use the red and green in one figure because some people are blind to that. There is a concept comes back to the John Locke, the great English philosopher, that It's possible that two people use exact vocabulary for the colors, but they are systemically talking about different colors. Today we have Ariel that he's going to present an interesting experiment that we are learning more about this concept. Ariel is in the Monash University, and the title of his talk is "Are Color Experience Equivalent Across the Visual Field?" Thank Ariel for joining us, and we are very looking forward to your talk.
Thanks, everyone. It's a little early here in Australia, so I've pre-recorded. If there's issues with the sound, then let me know and I can do it live, but otherwise I'll just, hopefully this works. Hi, everyone. My name's Ariel, and I'm going to talk about an ongoing project to determine whether color experiences are the same across the visual field. So my first guess at an answer to this would be obviously yes. My naive impression is that there's some external world, light bounces off of stuff in it and into my eyes. This generates color experiences across my visual field, and I'm able to report these to you all. But there's evidence from neurophysiology and psychophysics to be suspicious of this naive view. Cone photoreceptor densities drop off dramatically with eccentricity, with most cones packed into just a couple of degrees of retinal eccentricity in the fovea. Correspondingly, visual acuity drops off rapidly with eccentricity. You start to get effects like crowding, where individual stimuli become harder to distinguish, and color contrast sensitivity diminishes quickly with eccentricity. So maybe the naive view isn't correct. How do we test this then? We need some way of capturing color experiences. One way to do this is to show people colored stimuli, which generate color experiences, and then get them to rate how similar these experiences are to each other. An intuitive take is that any particular color experience can be captured by its relationship to all the other color experiences. So to take red, for example, a red experience can be described as something that's identical to another red experience, very different from a green experience, and very similar to, but not identical to, a pink experience. Collectively, this set of similarity judgments describes color experience. Now, previous work has looked at these color similarity judgments in central vision, but to the best of my knowledge, no one's done this yet in peripheral vision. Researchers have looked at how people rate the same stimuli shown in central and peripheral vision along predefined axes, such as hue or saturation, but no one has collected actual similarity judgments from these peripheral stimuli. We want to examine similarity judgments from central to peripheral comparisons, and from peripheral to peripheral comparisons. Now, just before I get started on the experimental details, I want to flag that there's some controversy in the field as to whether we should be accounting for cortical magnification or not. Like with the photoreceptor densities, there's an uneven amount of cortical resources dedicated to visual processing across eccentricity, with much more being allocated for foveal vision than peripheral vision. Whether it's appropriate to scale the stimuli size to match cortical magnification is disputed, so I'll be showing examples of both cases. Now, to get into the experimental details themselves, we recruited our participants from Amazon Mechanical Turk. We did screen size and viewing distance calibrations initially to make sure participants saw stimuli all of the same size. In terms of eccentricity, the central stimuli were displayed at 1 degree of visual angle, and the peripheral stimuli were displayed at 10 degrees of visual angle. We used a range of different stimuli size where the peripherally scaled stimuli were shown using V2 magnification, which was 4.5 times the central stimuli at 10 degrees of visual angle. Obviously, for fixed stimuli, they were kept the same size. We used 9 different colors with 250 milliseconds of presentation to try and prevent saccading, and the whole thing took about 30 minutes. You can see an example here on the right where fixation, stimuli presentation, response. So what were the actual results? Now, very briefly, and I don't expect you to be able to interpret this, I'm just showing you the individual subject data to give you some idea of how varied and rich it is before I move on to the necessarily simplistic summaries. But you can see here there's quite a variety of individual responses. So to the actual data, you can see here that with cortically magnified stimuli, the color similarity judgments from central to central comparisons, central to peripheral comparisons, and peripheral to peripheral comparisons were all extremely highly correlated. suggesting that color experience structures are equivalent across the visual field. Looking again at this cortically magnified data, but now breaking it down by stimuli size, you can see that the central to central and peripheral to peripheral judgments remain highly correlated, regardless of stimuli size. However, looking at the fixed size stimuli data, you can see that below a certain size of stimulus, here below about 0.25 degrees of visual angle, these correlations start to break down. Now, this suggests that color experiences may not be equivalent when generated from sufficiently small stimuli. Now, one further question we had that comes from the philosophical thought experiment of color inversion is, how do I know that my red isn't equivalent to your green? This motivates the question of within subject color inversion, which is for the same stimulus and the same response, might the subject be having a different color experience? And if so, would we be able to detect this in the similarity structure data? So obviously we can't get at this directly because subjects wouldn't necessarily be able to notice it. So instead we simulated it and we saw whether we could detect the inversion. To do this, we take some set of stimuli and the similarity judgments that correspond to these stimuli. Then we swap the similarity judgments that apply to particular stimuli. So for example, here in the periphery, the judgments that apply to orange now apply to purple and vice versa. And you can see here what the similarity judgment matrix might look like once we re-index the judgments according to the original stimuli ordering. So what we want to know now is whether the central to central and peripheral to peripheral judgments will still be highly correlated with each other, or whether there'll be some statistically detectable decrease in correlation between the two apparent after we've switched which stimuli correspond to which similarity judgments. What we found is that in general, these inversions are detectable. You can see here on the left, one particular inversion that we simulated. So this resulted in a detectable decrease in center to periphery correlation across basically every subject, suggesting that if participants did indeed have this color experience inversion from central to peripheral vision, as described by that swapping you can see there on the color wheel, we would be able to detect it through a change in the similarity structure. So what do I want you all to take away from this talk? Firstly, that color experiences are probably equivalent across the visual field. Now, we've only looked here at up to 10 degrees of eccentricity, which is about the limit with what we can do with an online study on participants' own screens, but it's already well outside the fovea, as you can see on the bottom left there. After COVID, we'll try and follow this up in lab. Secondly, color experiences across the visual field when generated using sufficiently small stimuli aren't necessarily equivalent, but this isn't the case when taking into account cortical magnification. And lastly, although I only showed you one example, there's reason to believe that most within subject color inversions across eccentricity would be detectable. Now, all of this together is evidence that irrespective of veridicality, the naive impression of color experience across the visual field may be accurate, that the entire visual field is colored. Now, I'd just like to thank all of those who helped me with the work and to thank you for your attention. Additionally, if I could get you to really to fill out one really brief survey about your opinions about color experiences, I'd be extremely grateful. Okay, any questions?
Thanks.
Thank you, Ariel. That was a great study. I will wait a few moments to audience write the question. I'm pretty sure there are a bunch of questions right now. Yeah, so you also submit the document here. Do you want to add anything on that before audience start questioning?
No, I mean, people are, there's a question. There's already some questions.
Okay, awesome. Yes, I see the questions. Okay, Julie asked that 10 degree may be outside of the fovea, but it's still considered in the peripheral regions. Do you expect the same result further in the periphery, like 20?
Yeah, so it's difficult to get at with the methods that we have at the moment,
given we can't do in-laboratory studies. I would still expect the same results. I think if you do the cortical magnification and you use large enough stimuli, I'm suspicious you get the same results out. I don't know, but like considerably further. But that's really only based on my naive impression that my own color experiences extend pretty far out, rather than based on like good empirical data. Though if you look at the studies where they don't look at similarity ratings, they just look at how people rate the colors on like already predefined axes, you see that those are reasonably stable, I think, until a bit further out as well, with using, I think they're like 2 degree of visual angle stimuli or so. So my guess would be yes, but obviously I can't assert that without data.
Perfect. But I guess you have that in your experiments, the like up to 10, 20 degree, right?
Well, we have up to 10 degrees, which is into peripheral vision, but it's not super duper peripheral, given humans can see up to about 200 degrees in total.
Awesome. A.M. Hound also asked, that's nice. Did you include RG diachromats as subject? Can distinguish the result from normal?
Yeah, so that's a good question. We didn't exclude anyone on the basis of saying that they had color blindness, but we did collect a post-experimental survey asking if they would report any color vision impairments or visual impairments or any of those sorts of things. So on my list of things to do is to go through those surveys, see if there's anyone who said that they were colorblind, and then see if they can see if their data looks different. But I don't have an answer for you yet.
How many subjects you had for that?
So each particular task has about 15 subjects in it, and I think there's like 10 different conditions. So there's about 150 subjects total. Yeah, it will be a good test to see if we can see the differences. Although it'd be interesting if I suppose you'd have to have compression along some axes, even if the relationships are still. Yeah, I agree. Sorry, I'm speaking to the chat. The comment was just that it would be a good test of the method if we can pull the colorblind people out.
Yeah. Oh, did you try on the colorblind people also? Yeah, that's what. Yeah. Okay, awesome. Thank you. Great. Also, if you want to come on a screen, just ask me to be on a screen for asking a question, Mario. Otherwise, thank you, Ariel, for your great talk. And let's move on to the last talk. Yeah, for the last talk, we have Caroline from York University, and she's a master's student that works with Dr. Falla. She's going to talk about the effect of distractors and saccade trajectories. Her title is Spatial Profile of Distractors Modify Saccade Trajectories. Thank you, Caroline, for your talk, and we are looking to hear your talk. Thanks.
Almost forgot to unmute myself. Okay, thanks for the introduction. Oops. Sorry, having a bit of an issue here. There we go. All right. When you're making a saccadic eye movement from a central fixation cross to a target object, such as this red circle here, if the target is the only thing on the display, the saccade you make will be relatively straight towards the target, and it'll follow the direct path. Whereas when there are multiple objects on the display, such as a target object and a distractor object, like this blue circle, both objects will compete for your attention according to previous research, and you'll end up with a saccade that initially angles itself at the average of the two object locations, and then it'll curve to compensate as it goes to the target. And this curvature towards the distractor object is seen for short saccadic reaction times, or SRTs, which reflects the distractor's ability to capture gaze. For longer reaction times, we're able to fully cortically process both objects and inhibit the distractor, and so we end up with a curvature that points away from the distractor. Previous studies that have investigated the distractor's effect on saccades have kept the distractor at a set distance to the target, as well as keeping it very similar to the target. So to better understand the target-distractor interaction, we varied the distance of the distractor to the target, as well as the similarity. And so when there's enough time for this cortical inhibition of the distractor at these long reaction times, we expect that as angular distance changes, saccade curvature will vary according to this spatial suppressive surround annulus. And so this is the idea that when the target and distractor are close enough together, they're in an attentional or excitatory spotlight, but as the distractor moves further away, it is suppressed according to this gradient, where we get the most suppression at a medium distance away from the target. And as the distractor moves even further, we get suppression that's reduced. And since a suppressive surround has also been found in feature space for simple features like color and direction of motion, we expect that there could be a complex object space suppressive surround, which we hope would see a similar modulation of curvature when we vary the similarity between the target and distractor. Participants were first shown a preview of the target, and then were later asked to fixate on the central fixation cross, use their peripheral vision to figure out which object is the target, and then make a saccade to the target. And we varied the distance by what we called angular distance in an egocentric reference frame. And the target could be placed at any of these positions around the display, but for today's discussion, we'll just use the top location. And the distractor would then be placed relative to the target at one of these 10 positions, either on the clockwise or counterclockwise side. We also varied the similarity between the objects, and these are examples of the novel complex stimuli we used, from which we were able to create objective similarity differences. So similarity difference 1 would mean there's 1 line difference between the stimuli, and 2 would mean there are 2 line differences. We went up to 4 for this study, and so just keep in mind that a higher similarity difference means that the objects are less similar to each other. And in another study, we found that perceptual similarity also follows this objective similarity. We used a variety of metrics to measure the saccade trajectory, such as initial angle, which is taking the 20% point along the length of the saccade, and the angle between that and the direct path. And then the sum curvature, which is taking all the deviations away from the direct path and adding them all up. We split our results by saccade reaction time due to the differences in curvature we saw earlier. So looking at short reaction times, we see that as angular distance increases, the initial angle decreases, whereas sum curvature increases and then plateaus around angular distance 3. Both these effects are significant, and it's indicative of not having enough time to inhibit the distractor. And so we get that the saccade angles itself at the average of the object locations, and then curves when both objects are far enough away from each other to cause competition for your attention. And we see a different effect for long reaction times. We get this U-shaped curve for both initial angle and sum curvature, which supports the idea of the spatial suppressive surround. So at angular distance 3, we see the most curvature away from the distractor because we get the most suppression of the distractor. For similarity, as expected for short reaction times, we don't see any effects on the curvature. But for long, we see an effect on initial angle only, whereas the objects become less similar to each other, the saccade is initially angling more away from the distractor. And in conclusion, angular distance affects the saccade curvature and initial angle for both short and long reaction times, but it's at these long reaction times when we find this pattern of inhibition that supports the spatial suppressive surround because we're able to cortically process both objects. Whereas for similarity, we only see this affecting the initial angle for long reaction times, which suggests that more complex objects do not have an object space suppressive surround like the simple features do. And what we did see is that the less similar the objects get, we're able to inhibit the distractor more. So with that, I'd like to say thanks, and thanks for listening.
Thank you so much. That was great. I'll wait a few moments that audience can write the question. Just for curiosity, some of these objects that you make is probably look like the letter or digits. Does it change the reactions?
We were mainly planning that they didn't look like letters or objects. I can kind of go back if you want. Yeah, there we go. I guess you can kind of see some of them if you really look at them might. But no, we didn't look at any differences based on if they might look like letters or numbers. We were hoping they didn't.
But that's interesting to know because that like memory kind of thing may affect some way.
I did have a participant that strategy was to sort of pick what the different objects looked like, and he would talk out loud too, which was difficult with the eye tracker.
If no one has a question, I have a question.
Sure.
Would you say, as I understood it, people change this curvature is because like they're like, oh wait, this is the wrong thing I'm looking at. I'm gonna change my so does that insinuate that people are thinking about the object before they even saccade? So yeah.
Yeah, so I guess for our study, they had to stare, actually this panel that's highlighted here, they had to stare at the cross and use their peripheral vision to figure out which one was the target. And we only included correct trials for this study, like for this analysis at least. So I guess they would have seen in their peripheral vision that the target is the one that they wanted to look at as they were deciding to plan for the saccade. And so that's why we see with the longer reaction times, when they had more time to plan and more time to decide which object was the correct one, that the saccade curves differently, because they're able to, like, the distractor doesn't have as much of an effect on the saccade curvature.
Oh, I see. So you wouldn't say that they were doing any kind of perception while doing the saccade? Because, you know, there's the whole saccadic inhibition.
Yeah. I think maybe sort of unconsciously, yeah. I know when I was doing it myself, it goes really quickly, and I guess you end up seeing these effects even though you're sort of unconsciously deciding and you're just like, I'm gonna look at that one.
Okay.
Or at least it feels like it's unconscious.
Can I ask one more? Is it?
Sure, yeah, if nobody else has questions, sure.
Yeah, so the distance is the same, but you're saying that there's variance to the reaction time for the same distance?
Yeah, so it's more, the reaction time was more the time the participant just took to stare at the cross.
Right.
So as soon as they started moving their eyes, that was the end of the reaction time period. So it was just variance in participants' like trials, yeah.
But so the speed is the same for all the saccades, because they're all, because of the same distance. I assume like the speed distribution is probably really tight.
That makes sense. I actually haven't looked at speed, but I probably should.
Okay. There's like all this. There's all this like old literature like showing the correlation between saccade size and eye speed, and it's like really, it's kind of a tight correlation. I'm just curious if you see that.
Yeah, thanks for the suggestion.
Yeah, and the trajectory are different when you have a further object, or the same almost?
Pardon?
The trajectory of eye movements, are they changing when you look at the further objects, or?
So both objects were equidistant to the target. We kept them at the same distance every trial. They were just different distances from each other.
Oh, but overall distance is fixed here.
Yeah. So it was at a 7.5 degrees of visual angle was that circle that they were placed around.
Sorry, then I missed that one that you changed probably that angle. Oh, so they are the angular distance. And yeah,
so actually right here, so the target was placed somewhere around the circle, and then the distractor was relative to the target, 5 positions on either side away, up to 5 positions on either side.
And that was symmetric for left and right, right? The result.
Yeah, we kept the same amount of trials left and right, yeah. We just grouped them together for the analysis.
I've got a more basic question.
Yeah.
Sorry. No, I mean,
yeah.
If other people have better questions, they should ask theirs.
Oh,
but...
yeah. So there is not any question. Sorry, I have to clean up the last question, but I don't see new question coming. Yeah, go ahead if you want to ask.
So is so I'm assuming that if you only use one target, people move in a straight line to the target. There's no curvature in like a typical saccade from the central fixation to the target.
Yeah, I think that there's, sorry, were you done?
Oh, and then I had my follow-up to that is that if there are like 2 objects, and there's like a target and a distractor. Is the idea just that they make some like vector sum eye movement of like the attraction to the target and the other one, as opposed to like they like normally would go in a straight line to one and then maybe go to the other one next? It's interesting that like there's some like sort of summation of attractive forces from the two different objects that are there. That's what I'm confused about or curious about.
Yeah, so I think the idea is that, yeah, when there's only one object on the screen, it's relatively straight. There is sort of like a underlying curvature to a saccade, which is why we would normalize our data. But when there are the multiple objects, we do get this curvature. Yeah, sort of like you said, there's an averaging of the vector, like saccades to both, and you sort of average the vectors. And that's why we get that initial angle that's outwards and then the curvature to sort of pull you back in to go to the correct object. But then you see differences in the direction based on how long you take, for example, which is what we were mainly looking at. I hope that answers.
And does it change over time? Like, do you have an adaptation here?
Pardon?
Does it change over time? Do you have like adaptation for this curve over time, like the subjects are doing the experiment?
Oh, like as they do the experiment, does their curve? I didn't look into if later on in the experiment they had differences. That's a good point, though. Yeah, but I didn't look into that yet.
Okay, thank you so much. If there isn't any further question, let's thanks again all the speakers, and that was a great talk for all of you. I hope to see you again in the rest of NeuroMatch Academy. Don't forget to tweet or retweet about the NeuroMatch. Use NMC3 hashtag if you want to find the relevant news. Thank you so much again, and have a good rest of the day, all of you.
Thank you.
Bye.
Thank you.
And hello, everybody. Whatever your time of day is, it's lovely to see you. My name is Ina Puce, and I'm going to be the MC for today. And the host, who's doing all of the back-end work, is Shekufe Hedayati today. So thank you, Shekufe, for helping out. We have some really interesting talks today. Shekufe is also going to be watching for questions, as I will be. And so feel free to put your questions in the chat window, and we can monitor those so we can ask those for speakers later. The common theme today is really the resting state network. But that said, we've got a very interesting set of talks that are coming up. And our first one, which is coming from Clément Garin, is on primate resting state networks. So a comparison between human and primate resting state networks, which is going to be very, very interesting to see. So without further ado, Clément, the floor is yours.
Sure, thank you very much. I'm going to start to share my screen. So you should now see it.
Yes, we do.
Great.
And then if you can, yep, lovely. Thank you.
Thank you for the very kind introduction. Today, as you said, we're going to talk about non-nominate primate network, and especially we're going to focus on the default network that we're going to try to compare this default network from the primate to the human one. So as you may know, the default network is a network that is particularly involved and is particularly involved in high cognitive abilities such as self-directed thought. This network is highly active at rest, and we can actually divide this network in 5 clusters in which we can find the parietal cluster, the temporal cluster, the posterior cingulate cortex cluster, and the medial prefrontal cluster. The medial prefrontal cluster is actually an important cluster since it has been shown to be involved in a gateway function of self-representation in humans. In mammals, some default mode network-like has been proposed. One of them has been explained, has been detailed by Grandjean et al. In 2019 on what we can see here is that a seed, so meaning a small region, has been created in the anterior cingulate area, and after we can analyze the correlation to this region. So that's what you can see. And this is the default mode network that has been proposed in that species. We can see that we have some parietal temporal regions that are correlated to this seed, as well as some frontal region. Exactly the same thing has been done a long time ago in rats. And in macaques, since we have a much bigger brain, we are able to actually have a bigger scale and to see with much more detail the region involved in this network. So in 2011, some clusters such as the medial prefrontal cortex, PPC, PCC, and dorsolateral prefrontal cortex has been demonstrated to be involved in the default mode network-like of the macaques. More recently, in marmosets, Liu et al. Found that the medial prefrontal cortex seemed actually to be missing from the default mode network-like of this primate. So what they have done here is they analyze the various correlations that they can observe between few seeds. And you can see that, for example, here, the correlation between the PCC and the dorsolateral prefrontal cortex seems to be high, especially when compared to the correlation between the PCC and the medial prefrontal cortex. So that is actually very interesting for the function that I described earlier. So this result actually also been confirmed with tracer injection that you can see here on your screen. So what the purpose of my work here? I could create one big question, that is the medial prefrontal cortex role in the default mode networks further developed in human than in other primates? So to do so, the first thing that I have done is to try to compare a statistical tool, a statistical analysis that will allow me to compare the role of the medial prefrontal cortex through different species, because of course we have different signal, we have different SNR, so it's actually more difficult than we think. Then I'm going to try to answer to that question. In the third time, I think if the medial prefrontal cortex is not involved in the default mode network in non-hominid primate, in which network is it involved? So I'm going to also try to answer to that question. So here we studied 3 different primate species: human, macaques, and mouse lemur. Mouse lemur is a very small endemic species from Madagascar. And as you can see, the macaques were anesthetized, but not humans. So the first method that I choose to extract the network into the macaque's brain and into the human brains, and you will see not in the mouse lemur brain, this first part, I choose to use atlases. And these atlases actually display some homologous region, which is a great thing for comparison. And as I said, since mouse lemur doesn't have this atlas, I'm not going to talk about the mouse lemur in this first part. So we extracted some seeds in various anatomical area of macaques on human. And as you can see here is a correlation, the statistical map that you can observe with similar region. And we use first the area 8AD, and you can see that we observe a region that we choose to call here the frontoparietal network that seems to actually be very close to the literature and to what has been called the default mode network-like. In human, using a seed in the area 8AD, we obtain a default mode network, a classical one. Using a seeds in the medial prefrontal cortex, I obtain a network that links the medial prefrontal cortex or the 9D, but we also tested with the area, for example, 32. We have exactly the same network. We have a correlation of this region with some temporal area. When in human, we obtain the classical default mode network. Using a seeds in the area, anterior area of the cingulate cortex for 24C, we obtain again the frontotemporal network. When in human, we obtain the default mode network. And to the end, I choose to show you the area 46D that led also to the frontotemporal network in macaques, but to the executive control network in human. So you can already see that we might have some important discrepancy between the organization of the network in macaque and human. So the third species has been done to actually include all our 3 species, and I choose to perform on our bold images a dictionary analysis. This analysis is highly similar to ICA, but seems to have better decomposition of the overlapping signal. So classically, this method is going to produce statistical map, and this statistical map can be concatenated into functional atlas, as has been done, for example, by Yeo et al. In 2011. So since this study is a gold standard and find that we could segmented the brain network into 7 networks in human, I choose to use 7 networks in all R3 parameters. So here you can see the results in R3 spaces, and I only show you 2 statistical map for each spaces, and you can see classically that we are able to extract the defaultment network, the frontoparietal control network in human, and in macaques that we recovered the 2 networks that I described to you earlier, the frontoparietal network and the frontotemporal network. Interestingly, in mouse lemur, we find a frontoparietal network that doesn't seem also to involve the medial prefrontal cortex. We find another network that involves the medial prefrontal cortex on some temporal area. So then we concatenated all these 7 statistical maps to build 1 functional atlas for each species. And we segmented all this cluster based on the anatomy. So most of the region has been segmented automatically, but some of them has to be segmented manually in mouse lemurs. So here I wanted to reproduce what I've been done here, but with a statistical comparison. So as I said, we created a method that we call statistical comparison of the relative correlation difference between functional and anatomical region, which sounds complicated, but basically it's the same thing as asking, when compared with medial prefrontal cortex, is the dorsolateral prefrontal cortex more or less connected to the PCC? So what I've done is that I extracted in all my spaces the correlation between the PCC and dorsolateral prefrontal cortex in each individual, done the same thing between PCC and medial prefrontal cortex, and then perform a statistical test to see which one is greater than the other one. So when I've done that on human macaques and mouse lemur, for example, for the connection between the temporal and the dorsolateral prefrontal cortex and the medial prefrontal cortex, I obtained that the medial prefrontal cortex seems to always be more connected than the dorsolateral prefrontal cortex to the temporal area. However, if I compare human macaques, mouse lemur now for the correlation between PCC, dorsolateral prefrontal cortex, and medial prefrontal cortex, I found that the medial prefrontal cortex is strongly connected to the medial prefrontal cortex, but that was not the case in macaques and mouse lemur. So then, since the functional atlas might have some ROE problems since it doesn't follow the anatomical boundaries, I also repeated this test on the 2 atlases that have similar region in macaque and human. And I obtained that the area 8AD is actually not less connected than the medial prefrontal cortex, but as connected in human. And if I, for example, repeat this test with the posterior anatomical area that is usually integrated into the deformant network, I obtain exactly the same results. And finally, in macaques, I obtain the opposite results, meaning that the 9M is always less correlated than the 8AD. So in conclusion, here we saw that it seemed that the PCC and the medial prefrontal cortex were always, the connection were always weaker in mouse, lemur, and macaques, but not in humans. We might have found a new network that involves the medial prefrontal cortex, and that seems to be similar to non-hominid primate, but not to human. And finally, this area 8AD, that seems very interesting. Is it an ancestral characteristic of the primate frontoparietal network? So if this is the case, and we have 2 different networks, the hypothesis will be, do we have a reorganization or a fusion of the frontotemporal network or the frontoparietal network that could be a singularity of the human defaultment network and potentially explain its specificity. So thank you for your attention.
Thank you very much. That was absolutely fascinating. And let me see if Shakufa has given me any questions. No, because we have a shy audience, and so we don't have questions. But of course, I have lots. So I'm going to ask you, Clement, a couple of questions. One big picture question, and that relates to you've got quite a difference in level between the human subjects and then macaque and mouse lemurs. And I want to get you to speculate about what you think might happen if you were to do the same thing for a great ape brain. What would you expect? Would they be more like the human one with the connection between the PCC and the medial prefrontal cortex? Or would you expect it to be more like the macaque?
That's a very interesting question. I mean, the day I would have this data set, I will be really happy to do the analysis. It appeared that only one paper has been published on the chimpanzee default mode network. And it seems that the medial prefrontal cortex is involved in the default mode network like of this primate. So that's why I chose the title as non-hominid, because I don't want it to say non-primate, since there is a study that said that it might be not a human specificity, but a non-hominid specificity.
Yeah, I wondered about that title because I thought there was a backstory behind it.
Exactly.
Yeah, great. Thank you for that. And yeah, if we find that paper later, maybe you can share that paper in the chat when the next presenters, because I'm sure other people might be interested in seeing that. And that goes too for the audience. If you know of any interesting papers that might relate to some of these studies, feel free to share those web links in the chat, because that's what makes these sessions online much, much more interesting for everybody. Now, I have a second question, which is a more nuts and bolts question for you, Clement, and that relates to these brains of these little primates, the lemur, the mouse lemurs, and the macaques, of course, are much, much smaller than the human brain. And so I'm thinking about the kinds of changes in blood flow, in the fluctuations, and the kinds of frequencies that these fluctuations occur on. And are they vastly different relative to, say, what goes on in the human brain? Because I know people in the human brain with resting state fluctuations have looked at wavelets type analyses and looked at the frequencies of, of that, the various bold signals. What kind of frequency changes do you see in these very small primates?
I mean, I didn't actually really look at the comparison of the frequency, but yeah, since all the frequency were band pass between 0.1 and 0.001. It's actually something, that could be interesting to see if there is some changes, because I know, for example, that studying the blood flow in mouse lemur is actually very difficult because it has a specific heartbeat. I'm not an expert on that, but I know that we tried in my previous lab and it was almost impossible. So yeah, it might be interesting to study that more in depth. Yeah.
Well, congratulations on getting a mouse lemur fMRI signal given those difficulties. That's a tour de force, and that is just amazing. Beautiful data. Thank you so much for sharing that with us today. So we will move on now, because it's time to move on to our next speaker. And our next speaker is going to be Catherine Devaney. And now we're sticking in the default mode network, but this time we're going to the human brain, and we're going and looking at how focused attention networks and the dorsal mode network change in people who practice meditation, expert Vipassana meditators versus controls. So a completely different change of gears. So take it away, Catherine. Thank you.
Okay, thank you. Let me make sure my arrow keys work here. They do. Okay, so today I'm going to talk about some work I've done using fMRI to investigate the neural correlates of cognitive processes involved in attention and meditation. And as we've all experienced, the visual world contains more information than we can simultaneously process. So in order to be able to successfully interact with the environment, humans need to be able to attend to an object of interest, like this man attending to some probably very important stock information on his cell phone, while filtering out irrelevant distractors like street noise, etc. And attention enables us to select for further processing one of the many competing external and internal sources of information. So, as we see, he's very focused on his phone. However, if a taxi were to come around the corner, it would be advantageous for this man to disengage from his phone and re-engage on the approaching predator. So this purpose of disengaging and re-engaging can be adaptive and can prevent us from getting eaten by taxis. However, it can also work to our detriment by causing us to get distracted when we really want to be focusing. So there are two complementary networks in the brain that these functions map onto. There's the dorsal attention network, which comes online when people are doing sustained focused attention tasks. And then there's the so-called ventral attention network, which acts as a circuit breaker into the dorsal attention network to reorient attention when a salient feature of the environment would be beneficial to pay attention to. And of course, attentional processes exist across multiple levels of the information processing hierarchy. But for my talk today, I'm going to focus on 3 cortical networks in particular. So this map should be familiar to all of you. We just saw it. So this is the 7-network map. And today I'm going to be talking about the dorsal attention network, the default mode network, which, as we were just talking about, tends to come online when people are doing self-referential thought, thinking about the past, planning for the future, and importantly, is suppressed when doing difficult tasks or when attending outward into the environment. And then finally, the ventral attention network, or the so-called circuit breaker attention network that I mentioned a moment ago. So ideally, these networks work together to optimize interaction with the environment, but sometimes they fail. And so optimizing the relationship between these networks is of great interest to this individual in particular, and to human beings in general. So how do we optimize these relationships? One candidate process is meditation. So in meditation, one is training attention in the absence of any stimulus-driven feedback or input. And there are multiple types of meditation, but in particular, what I'm going to focus on today is called focused attention meditation. So in focused attention meditation, you bring your attention to some object, usually the breath, and when there's an internal or an external distraction, you notice that you're distracted, and then you just bring your focus back to the object of attention. So the hypothesis we had going into it is, with repeated practice over time, this should alter the relationship between cortical attention networks. And there are many candidate networks and many cognitive processes involved in meditation. In particular, one has to sustain attention on the object. Then, presumably, the default mode network is coming online as one is getting distracted. Then there's salience and executive network, responsibility for noticing that one is distracted, coming back on task, and then reorienting attention back to the object of interest. So this is a relatively, we assume, high-level training at the cortical level. And there has recently been growing scientific interest in how meditation works in the brain, and different sort of candidate processes that are trained by meditation, so in research labs, as well as in popular culture, but does it work? So we had 2 goals here. One is to design a robust cross-sectional study to investigate attention networks in meditators and match controls. Given the differences between these populations, that turns out to be a non-trivial task. And then second, after we have that robust design, to examine the cortical correlates of sustained attention and attentional capture in meditators and controls. So we recruited highly experienced meditators who were all practicing one technique. So that eliminates some potential for confound by having people doing wildly different types of meditation. Everyone had a lot of experience. The average lifetime meditation hours was 5,000. These people are meditating on average for about 850 minutes per week. And they're all recruited out of the same center. And then we were very careful about how we found the controls. And importantly, in the interest of time, I won't go into too many details, but one thing that was a high priority for us was to equate the demand expectations between the groups. Because many studies that have been done before of meditators, you know, the meditators fill out these like 3-page questionnaires about their meditation experience, and then they just pull in the controls. And it's been shown that if you believe you're supposed to do better at a task, you will. And so we wanted to control for that. So we put the controls through the same exact kind of screening process that we put the meditators through, only we were just screening them for sort of nothing, just to normalize demand characteristics. So then we had 2 tasks, one that's designed optimally to recruit the dorsal attention network and suppress the default mode network, and another that's designed to recruit the ventral attention network. In the interest of time, I'm going to skip this demo, but this is the multiple object tracking task. Suffice it to say that in this task, what you have to do is fixate at the center and then sustain attention on these discs while they move. And then at the end, determine if one of these 4 discs was one that you were attending to. So this is a highly demanding sustained attention task. There's been a lot of work on this task for the last 5 decades or so. And then the ventral attention network localizer had 2 levels. One was a modified Posner task, which same thing, that's been used as a ventral attention network recruitment localizer for a long time. But to make it more robust, we added a RSVP stream of oddball images. And this is there's a Frontiers in Human Neuroscience paper just about this task, if you want to know more about it. So now we have our design. So let's move on to running the study. So first of all, in the sustained attention task, and I just want to draw your attention here to the fact that there are blocks in the multiple object tracking task where they're sustaining attention, and then there are blocks where they're not. All they have to do is fixate, and they just hit a random button at the end. So what we have there is all the bottom-up stimuli are always the same, and we can compare attentional state across an attentive state and a passive state. So we can compare attention versus passive in meditators and controls. And so when we do that, this is the grand group average of everybody. And what you can see, if you recall the map of attention networks from before, is that areas in the dorsal attention network, including the IPS and the frontal eye fields, are highly driven by this task. These are the areas you see in yellow. Whereas default mode network areas are suppressed in this task during attention. So now what if we split it up by group and we look at the meditators versus the non-meditators? So when we do that, even just looking at a glance, there's a striking difference between the meditators on the left and the non-meditators on the right. And you can see, first of all, that the magnitude and extent of dorsal attention network activation is greater, and also the extent of default mode network suppression is significantly greater in the meditators than in the non-meditators. And a between group full brain group average comparison and an ROI analysis bears this out. So to do the ROI analysis, we just pulled the whole network and we looked at percent signal change in attend versus passive in the dorsal attention network, the default mode network. And you can see the meditators in orange and the controls in purple. So both the level of activation in the dorsal attention network and the magnitude of suppression in the default mode network is greater significantly in the meditators than in the controls. So to turn our attention to attentional capture, we look here at oddball trials versus non-oddball trials. And what we see in the grand group average is ventral attention network structures, including the temporoparietal junction, are recruited by this task. However, when we compare the meditators to the non-meditators, we don't see any evident differences in the maps. And indeed, when we do a between group comparison, we don't see anything popping out in that contrast. And this was borne out by a region of interest analysis. So you can see that there's individual subject variability that's driving these ROI results, but the oddball versus non-oddball and the invalid versus valid trials were not significantly different between the meditators and the non-meditators. So these results taken together demonstrate that meditators show greater activation in dorsal attention networks and greater suppression in the default mode network when attending without a commensurate cost in attentional capture. And I'll quickly do future directions. As I mentioned, the sustained attention task was a block design. What I suspect was happening is that the controls were switching between doing the task and introspective thought, and the meditators were not. However, without any temporal information, I can't really draw strong conclusions about that. So my next goal is to look at sustained temporal attention in the meditators. I'd love to do some longitudinal studies someday, and then my ultimate goal, and connect me with anyone you know who's working on this, is to have a contemplative neuroscience retreat center where people are meditating and getting scanned, every other day or something. So that being said, I'd like to thank my mentors for this project, Dr. Sarah Lazar, Dr. David Summers, and Dr. Mark Halko. Keep in touch if you'd like, and thank you all for your attention.
And thank you. Thank you, Catherine, for that. That's fascinating. And I found that, of course, fascinating because I do meditate from time to time. And so I was dying to see what results you were going to show. And let's see, we have people who are shy of asking questions, but of course that gives me time to hog the floor and ask you. You've got a block design, which is great for the question that I want to ask. It takes a while for people to get into the groove of doing a task. And so it takes a while to focus one's attention. And I'm wondering whether you've had a chance to look at how in the group of meditators versus the group of controls, whether the meditators settle in to the task faster and they're not sort of being distracted, and whether those signals in those networks that you're showing, particularly in the dorsal network, whether that actually is a little bit more stable in the meditators early on in that block.
That's a great question. So Sarah, in collaboration with Mike Esterman, is doing some work looking at that in a graduated continuous performance task. So they're looking at performance decrease, which happens in non-meditators after about 7 minutes, but in meditators seems to be more stable. I will say everyone did practice runs of both tasks outside of the scanner, so they had a chance to ask questions or anything before, and that was my first indication that this experiment was going to work, is when I was showing 4 meditators, I was having them practice on the multiple object tracking task, and I said to them, so if you could just direct your attention to the laptop, and all 4 of them were like, boom, like right in. And generally with non-meditators, that you can tell they're kind of like zoning out, and so that was a promising sign. But that would be something that's definitely possible to look at, like a TR by TR analysis. Yeah.
Exactly, that's what I was thinking. Yeah, and also maybe to look at autonomic measures like pupil dilation or something like that. That will be really fascinating as well.
Yes.
Okay. Well, thank you very much for that study. And we're going to our last presenter of the session, and that's Braxton Hartman. and he is also going to talk about the dorsal mode network and interactions with dorsal attention networks in ASD. So this is going to be really interesting to hear. So Braxton, the floor is yours.
Great, thank you. So I will be discussing atypical functional connectivity between default dorsal attention and frontoparietal control networks in autism. Oops. So to begin with, what is autism? Autism spectrum disorders are characterized by difficulties in social interaction and communication and by repetitive and restricted behaviors. Now, a very large and growing number of studies have attempted to investigate the neurobiology of autism using functional connectivity fMRI, which, as I'm sure you all know, measures the correlation between hemodynamic response across brain regions to determine the degree of functional covariance. And this reflects, of course, a network approach to cognitive functions. So, right, and the idea is that networks that are observed during rest in the absence of any task are still expected to be topologically isomorphic to those that are driven by task activation. So for example, Biswall's seminal 1995 paper, you can see that the resting state activity in the motor cortex sort of drew out the same structures that you would see during a actual finger tapping task. So in particular, we're interested in looking at three networks. The default mode network, which comprises regions such as the medial prefrontal cortex, posterior cingulate, superior and inferior frontal gyri, the inferior parietal lobule, and so on. And this really deals with, as you heard, internally directed cognition. So things like introspection or prospection or theory of mind, and so on. The other network we want to look at is the dorsal attention network. This, of course, deals with your externally directed cognition. So when you're dealing with a difficult task, for example. And finally, we want to look at the frontoparietal control network. And this network is hypothesized to be able to flexibly couple with either the default or the dorsal attention network, depending upon task domain. And it's sort of anatomically interposed between the default and dorsal attention networks. It's comprised of areas like the lateral prefrontal cortex, precuneus, the medial superior prefrontal cortex, and the anterior insula, among others. And so on the right here, you can see a parcellation of the three networks from Nathan Sprang and colleagues' 2013 paper, where these were actually driven by tasks. So the default network activation was driven by a autobiographical planning task. The dorsal attention network was driven by a Tower of London task, and the frontoparietal network, as theorized, had covariant fluctuations with either default or dorsal attention network, depending on domain. And so the idea of this three network model is that the frontoparietal control network can, depending on whether you're focusing your attention internally or externally, it can flexibly couple with either the default or dorsal attention networks. It's sort of a third-party mediator. And so this is a Spring embedded graph, again, from the Sprang paper in 2013. And you can see in the blue here are all the default network nodes. In the green are the frontoparietal control network nodes, and in the red are the dorsal attention nodes. And so these are this parcellation of 43 nodes is what we're going to be applying to our ASD subjects. And so this is really important because alterations in the composition and interactions of these three networks using this model have been widely studied in a number of cognitive domains, including aging, depression, and bilingualism. And we see things like the breakdown of these networks in aging, a loss of the sort of canonical anti-correlated relationship between the default and dorsal attention networks and general fractionation of networks. And so these three networks have been pretty well studied in isolation, the default network at least. The default mode network has been pretty widely studied in autism because it's known to be involved in cognitive modalities which are impaired in autism, such as prospection, theory of mind, and so forth. But results have really varied wildly. We've seen results showing overactivation, underactivation, overconnectivity, underconnectivity, and so on. There's a lot less work on the dorsal attention network and frontoparietal control networks, but again, results are quite inconsistent with what little we do have. And there could be various reasons, especially methodological concerns. For instance, a number of researchers will use task regress data as opposed to pure resting state data. There are concerns with the ongoing debate over global signal regression. There are concerns with, let's say, motion parameters. Maybe the people with autism move around more, and this could introduce a confound. But really what we want to address is the gap in the literature and asking what are the interactions between these three specific networks, not looking at them in isolation, but in terms of their relationship. So our ASD data set comprises 25 young adult males with autism, as well as controls who are matched on IQ and, crucially, motion parameters. And we pre-processed our data using a modified version of the Anaticore procedure, and no global signal regression was used in pre-processing. Okay, so to begin with, our network analyses, we use the voxel time series averaged within each of the 43 ROIs in the Sprague parcellation. The coefficient for each pairwise connection of the 43 nodes was calculated. These were Fisher-Ardisy transformed. And then to establish the connectional modularity of these 43 nodes, we used average linkage clustering algorithm, with cluster distance being defined by covariance in the bold fluctuations between pairs of ROIs at the base level and in clusters at each level up. And so here is what this looks like for our TD subjects. So these blue dots represent the default mode network, red is dorsal attention, and green is frontoparietal control. And as you can see from the heat map, we have a very nice clean delineation of the three networks. All the default, our default network cluster sort of up here in the upper left corner, dorsal attention in the middle, and on the lower right corner, we have our frontoparietal control network. And if you actually look at the hierarchical clustering in the dendrogram off to the side of the heat map, you can see that indeed the highest levels of clustering are indeed for the three networks. So the highest levels of clustering are for the default mode network, for the dorsal attention network, and the frontoparietal control network. And there's obviously a lot of dimensionality underneath that, but at the highest level, we're seeing the three networks, as we would expect. Things look a little bit different for our autistic subjects. As you can see here, even just looking at the heat map, there's a lot less of a clear delineation, a lot less of a clear block structure that you would expect to see between networks. It just seems sort of generally messier and less delineated, excuse me. And especially interesting is when you look at the hierarchical solution and the dendrogram here, you see that the highest level solution is not for our three networks. In fact, you see that at the two sort of highest level solutions is for one component containing the default network and then half of the frontoparietal control network. And then on the other hand, the dorsal attention network and the second half of the frontoparietal control network. The frontoparietal control network seems to be split rather than acting as a sort of neutral arbiter between the two other networks. It seems to be split between them. And so this is interesting because in healthy adults, what you expect to see is, again, three nice separate networks. Some of the nodes of a frontoparietal control network are what we call promiscuous nodes when it sort of becomes, when you're having either an internal or externally directed cognition, certain nodes will switch affiliation from the frontoparietal control network to either the default or dorsal attention network. But here we're seeing that it's completely split even during rest, which you would not expect to see. And here's just the side-by-side, so you can sort of really clearly visualize the difference here. Again, on the left is your typically developing, and on the right is our autistic population. We also did some seed to voxel analyses. I'll skip the methods here and just go to the surface projections of the seed to voxel analyses of the left frontal eye field. This was chosen because it's an important dorsal attention node. And so as you can see here on the surface projection, we have, for the autistic group, hyperconnectivity of the anterior cingulate and sort of medial prefrontal cortex to the frontal eye fields in autism versus typically developing, but also hypoconnectivity in autism to the frontal operculum and insula area, as well as the inferior superior parietal lobule area over here, again in autism compared to controls. And this is just a table of the various select peak regions of between group differences. And so finally, we also took a look at betweenness centrality, which is a graph theoretical measure. And this was selected over other network measures because of its ability to explicitly detect interconnector nodes. Specifically, we looked at the frontal middle gyrus Brodmann area 6, because this is sort of the core interconnector node. Okay, moving along quickly here. And as you can see across various tie densities, it was in autism actually an increase, interestingly enough, of betweenness centrality in this area. And then finally, I'd like to look at differences in anti-correlation. So we performed permutation testing to see how many reliable anti-correlations between default and dorsal attention nodes there were. As you can see in TD, there are quite a few pairwise anti-correlations between nodes of a default and dorsal attention network. In autism, it's much sparser. There are much fewer reliable anti-correlated relations in the permutation testing. And so what does this mean? Well, in healthy younger adults, the magnitude of anti-correlation has been linked to individual differences in task performance. And in aging, we tend to see that as adults get older, we see reduced anti-correlations between these networks in rest. So it's very interesting that we would see in young adults with autism, this reduction in the canonical anti-correlated relationship. And that third network, the frontoparietal control network, which is supposed to mediate that relationship and mediate the task switching between internal and external cognition, it didn't seem to be clearly differentiated in autism. And if the FPC is indeed responsible for mediating task switching between internal and external cognition, does this perhaps reflect in autism an architecture which is impaired at task switching? Could this perhaps account for some of the symptomology of autism, the perseverations, for instance? And finally, the key findings from the left frontal eye field, which is a region which is, of course, very important for fixation, pursuit, vergence, and so on, and which we know is implicated in autism. It shows deficient response inhibition and hypoactivation generally. And the area that it is hyperconnected to is a region that is thought to be implicated in social cognition and associations of context and emotional response. So just hypothesizing, perhaps this could reflect an inability in autism to properly attenuate that relationship as a function of task-specific attentional demands. Again, the insula, another area that was implicated, is thought to facilitate between other distributed networks depending on internal or external cognition. So that further supports the idea that there could be impaired task switching. And finally, the left Brodmann area, BA6, actually had higher betweenness centrality, but I don't really have time to discuss that too much. So in summary, our results demonstrate that the relationship between these three networks is altered in autism. Crucially, there is reduced anti-correlation between default and dorsal attention and a lack of a differentiated frontoparietal control. And we think this could reflect a functional architecture which is impaired at task switching. Thank you so much.
Thank you, Braxton. That was really, really fascinating. I have a quick question for you related to that split in that frontoparietal control network. You know, in the dendrogram, it splits. And is it because of that hypoconnectivity, do you think, in those ventral structures? And then notice, you know, the insula is involved in that too. There's hypoconnectivity. Is that what is maybe creating that split, do you think?
That's an interesting question, and that's definitely something that I would want to look into a little further. There are a number of statistical techniques that I've read about in the literature that I'm really interested in applying to this data set to try to tease apart what is it exactly that's causing this split in the frontoparietal control network. And I think really the next steps would be to look at task-driven activation. If we were to replicate like what the Spring paper did with the Tower of London and autobiographical planning test, would these differences in resting state also show up as differences in task switching, for example, during tasks?
Yeah, and it made me also think about the fact that the activity in the insula, you know, Ralph Adolphs and Stanley Adolphs and Kennedy Adolphs have the empathy network, which, you know, includes the insula. And so that's what I was thinking of when you were presenting the data. So it's very, very interesting work. Thank you so much for that presentation. Now I have a question, it's for you, Braxton. What method did you use to estimate the distance between regions in hierarchical clustering analysis? And that's from Hee-Ching Hu, and he's got a second one, but I'll let you answer that first one first.
Right. So for the agglomerative hierarchical clustering, we used average linkage clustering. We had actually, we were thinking about which methods to use, and that was ultimately the method that was used in the Spring paper, which we replicated. And we implemented that using a Python pipeline.
Great. And then his second question is, why did you focus on adults with autism instead of on children? Is there any special reason for that?
Certainly. Well, one of the things that I really like to do is actually have multiple age cohorts, to have, you know, everything from very young children to very old adults. Unfortunately, it is very difficult to get young children with autism to sit still in a scanner. And also with respect to older adults, we actually don't have as many, partly because it's now that we're starting to really diagnose people with autism. And so, and of course, there's the fact that on a university campus, you mainly have access to young adults.
Yeah, that's true. Okay, well, look, thank you, Braxton, and thank you to all the presenters for an absolutely fascinating session. Thank you to the audience, and thank you also to Shakufe Hedayati for maintaining a really tight back end of this session. So thank you so much, and enjoy the rest of the conference. Bye-bye, everyone.
Bye, thank you.
Okay, and so we are now live. So as the attendees file into the room, it takes a couple moments for them to be let in by the software. I will just go ahead and introduce this session. So my name is Megan Peters. I'm one of the co-organizers of NeuroMatch. And with me here today, we have three excellent talks from Tyle, Daniel, and Electra. And so we are going to hear them each give some great talks. Just to orient you guys to the software, if you haven't done this before, at the bottom of your screen, there is a Q&A button. Please drop your questions into that Q&A box. And then during the Q&A period, I will read them out. And if we have time for any follow-ups, we can also hand you a microphone and you can chat from the audience as well. Also in your chat box, you have the option to do both panelists and attendees. So if you want everybody else in the room to be able to see what you're saying, you'll want to set it to all panelists and attendees. Okay, so without any further ado, oh, and I also want to say, sorry, that Yeshi Guo is our back-end host from NeuroMatch, and she's doing an excellent job keeping the tech running. So without any further ado, we will go to our first speaker. So we will first hear from Tyle Grutzbagers from Western Sydney University, and he's going to be talking about the neural dynamics underlying prioritization of task-relevant information. So I will ask the other speakers to please turn off your video and microphone, and Tyle, you can go ahead and share your screen. Looks good to me. Okay, whenever you're ready, take it away.
Thank you, Megan, and thanks everyone for joining us in your late evening, early mornings, middle of the nights. I'm fairly comfortable here at lunchtime in Sydney. My name is Tyle Grutzbagers, and I'm going to talk to you about a recent study of ours. And it's about information prioritization. And there's a lot going on in the world around us. And if we are not prioritizing the right incoming information, this has obvious consequences. So this is a fascinating topic that has seen a lot of attention, pun intended, over the decades. This question about at what neurostage and at what time in the brain's response to incoming information is task-relevant information prioritized. So I'm going to use task-relevant and attention a bit interchangeably. And we know a lot about this. We know about things that are presented in specific locations, at what time points those are enhanced. We know that certain features can be amplified and enhanced in neural responses. And in streams of incoming information, we know that relevant targets in these streams are see a very specific enhancement around 270 milliseconds. So there's a lot of research on this topic already, but what this often has in common is that the stimuli are presented in isolation. So it's often a cue, one stimulus, and you do something with the stimulus. And we were wondering what about stimuli that are presented in the same location and at the same time? So when there's a lot of competition going on. So what about stimuli that overlap in space and time? So that's what we focused on in this study. So basically the question is how attentional prioritization works when stimuli occupy the same space and are presented at the same time, and specifically which processing stages, so how does the time course of this, how is the time course affected by attention here? And we use so this is generally very difficult to disentangle, because when you present two stimuli at the same time or very close in time at the same location, it's very unclear which signal in your neuroimaging response belongs to which stimulus. And we've shown in a couple of papers last year that when you use so fast streams of information, so RSVP, in combination with EEG recordings and MVPA, so classification, you can actually disentangle the overlapping information that is specific to each of these stimuli in the stream. So we've got a couple of publications from last year that really nicely show this. So for this current study, we're doing the same thing. So we're presenting stimuli in relatively fast streams, and we're recording EEG, and we're using classifiers to figure out what information belongs to which stimulus and how this changes depending on task demand. So here are stimuli. We use 16 different stimuli. There's some birds, fish, boats, and planes, but generally we're interested in the decoding of the individual stimulus in the sequence. So we present these in relatively quick sequences. So these sequences are at 2.5 Hertz, and they're on. So each stimulus is on for 200 milliseconds and then gets taken off the screen for 200 milliseconds, and then we have the next stimulus coming on. And we asked subjects to do a two-back detection task on the stimuli. And to make it even more complicated, there's two stimuli on the screen. So one is the objects that I was just showing you, so those 16 images, and we presented them overlaid with letter stimuli. So we had 16 letters, and they were presented at the center of the object on the screen. And we asked subjects in half of the sequences to respond to two-back events on the object stimuli. So here that would be the boat is repeating in a two-back event, so then they would press a button to report this. In the other half of the sequences, which were actually yoked, as in we've shown the exact same sequence, but we asked subjects to respond to the letter stimuli and two-back events on the letters. So here they would not press the button whenever the boat was repeating, but they would press a button when that letter U repeated, two stimuli after. Now, as you may notice, there's a co-varying feature in this display, as in the letters are always the small element on the screen compared to the objects, which are presented at a much larger eccentricity. So to look at the interaction effect here of the relative size between the stimuli, we repeated this exact same experiment in a new set of subjects where we switched the location of the two stimuli. So now we have small objects overlaid on large letters. So this allows us to really nicely disentangle these effects. So they were doing the exact same task, and we presented the stimuli in the exact same order as well, but they are different subjects. So to illustrate this, I'm going to show you the two experiments, sort of to give you an idea of how fast this is going. This is a pretty difficult task, and people can do this task, but you find that they are, for example, they were not responding, they were not giving any false positives on the unattended stimulus. So if they were asked to do objects, they would not give you any letter two-backs. So this tells us that the manipulation works really well, right? So we can direct their attention to one set of the display really easily in this case. So we have 20 participants in each experiment, and we did minimal EEG processing as we've done in our previous work as well. So we do some filtering between 0.1 and 100 hertz, downsample the signal to 250 hertz, and then create epochs relative to the onset of each individual stimulus in the sequence. And these epochs lasted 100 milliseconds before stimulus onset up to 1,000 milliseconds after. So there's actually multiple events in this sequence, but we can align them to the onset of each individual stimulus. And then we use MVPA to decode the overlapping responses to the individual stimuli. So the first thing that we did was discard all the two-back target events. So we do not actually look at the target processing. So we were mostly interested in how does attention prioritize the information, all the stimuli in the sequence. And all the stimuli could have been targets, but were not in this case. So we discard the two-back targets. So all the stimuli are still relevant. And we use a leave one sequence out cross-validation. So we train classifiers on all but one sequence, and we test the classifier on the remaining sequence. And we train the classifier on distinguishing between the 16 objects, between all pairwise combinations there, but also between all the 16 letters that we used. And then we repeat this process separately for object tasks and letter tasks. So when they were responding to object two-backs and for letter two-backs. And then we repeat this also over all the time points in the EEG epoch. So we get a decoding accuracy over time relative to the onset of the stimulus. So here's the results for object decoding. So here we're decoding pairwise objects. So chance level is 50%. And we see really nicely that the classifier is indeed a chance before the stimulus comes on. So that's our sanity check. And then we see that decoding accuracy shoots up to around almost 60% accurate. For both tasks, so regardless of whether people were responding to objects or for letters, we see that decoding accuracy increases. And we see that the lines start to diverge around 200 milliseconds here between the different tasks. So in blue, we have the object task, so when the objects were attended, and in the dashed orange line, we have the letter task, so where the objects were not attended, but we could still decode the objects. Here are the associated topographical maps. So we see that the signal originated from occipital senses. And here are associated statistics. So here I put a blue dot in each time point whenever the decoding accuracy was significantly above chance. I also use Bayes factors in this case. So there's a blue dot in the second row whenever the Bayes factor was showing evidence for above chance decoding. And there's a blue dot in the bottom row when the Bayes factor was showing evidence for the null, so no decodable information. And in blue, we have the attended. In orange, we have the unattended. And in black, we have the difference. So we see that the difference starts to come on just after 200 milliseconds. So this is when the object was large and the letter was small. And in the second experiment, we swapped these two displays around. So I'll put them side by side here. And we see a very similar picture here, except for the very strong first response. So that strong early peak is gone. But we still see a difference coming on around 200 milliseconds. And in fact, it looks like it starts a little bit earlier when the object was the small item on the screen. So we see an enhancement of task relevant information in this case around 200 milliseconds, but it seems like that was happening earlier when the object was a small stimulus. Now, we also decoded the letter stimuli in these sequences relative to their task. So here's the results for letters. So I think the first interesting thing is that, yes, we could decode between individual letters. It looks slightly worse than for the objects, but it was still possible, but letters are a bit less complex stimuli. So this is, I think, why it's a little bit lower. And well, we see some interesting effects here. So we see an earlier difference for letters whenever the letter stimulus was small. So this difference is started around 100 milliseconds compared to the objects around 200 milliseconds. And when the letter was large, we again see that the difference seems to start a little later. So we see an early low-level attentional enhancement for letter stimuli when they were small on the screen. And the effect was stronger earlier when the letters were small. So in conclusion, what we found that regardless of the tasks that people were doing, early neural responses to stimuli in these fast changing streams display remarkable detail about the sensory environment. So we could decode between individual objects, we could decode between letters, regardless of whether they were attended or not. And the effects of attention seem to interact in a manner dependent on the perceptual characteristics of the display. So whether the object was small or large, and whether the letters were a small or large item on the screen. So with that, I would like to thank all the people involved in this project, and I will point you to the relevant preprint that summarizes this research on bioRxiv, and I would like to thank you for your attention.
Great, thank you so much. And in lieu of applause, because we can't do that in Zoom, I'd like to invite the audience to drop some praise into the chat if you enjoyed the talk. So thanks for the great talk. If you do have questions for Tile, please drop them in the Q&A box as well, or you can put them in the chat. I'll see them either way. But while we're waiting for some of those questions to come in, I was just wondering if you might tell us a little bit more about whether you're doing so you have this decoding performance that's kind of comparable, at least between the small and the large, and also between like the letters and the objects. Do you see though differences in, I don't remember whether you said what kind of decoder you're using, but differences in the informativeness of the features in terms of the anatomical location of like which electrodes are really strongly contributing? I don't know if you've done any statistical analysis on that yet.
We don't have any, that's a good question. We don't have any statistical analysis on this because it really becomes, it explodes in terms of complexity as soon as you start looking at individual electrodes and at individual time points. But sure. We do talk a little bit about different, it seems like it's a complex story. So that it looks like there might be differences in the type of lateralization that you see between these two effects. We talk a little bit more about this in the preprint, but there's no strong, reliable differences that we concluded from this.
Great. Cool. Okay. Well, if you do have lingering questions for Tile, please do drop them in the Q&A, and I'm sure he can answer them in chat on the back side. But if there's no other questions, I guess we will go on to our next speaker. So thank you again for the great talk. And we will move on to our next talk in this session, which will be by Daniel. I am going to mangle your last name, so I would prefer that you say it yourself.
I mangle my own last name every day. I think it's Furigal, but let's go with that.
Great. So thank you. So Daniel is going to be telling us about visual mismatch responses are a product of surprise, but not fulfilled expectations. And so this looks great. So whenever you are ready, please take it away. Thanks.
All right. Yeah, hi, everybody. My name is Daniel Furfuru, and I'll be talking about oddball sequences, visual mismatch responses, and surprise. So imagine you're walking down the street and you see a dog. So computer malfunction there. But of course, you wouldn't see a dog in isolation. You would typically see a dog in the context of some environment. Perhaps you're out in a green field. And based on this, you might expect to see perhaps another dog or another dog. You might be in a dog park, which would be a happy place to be. And so based on this sort of information, you wouldn't expect to see something like a toaster appear. And so here's a fairly silly example of how the context and our prior expectations and knowledge come to bear on influencing our expectations about what we will see in the immediate future, which can help us more appropriately interact or perceive these types of objects. So people have investigated this in the lab using oddball sequences in which individual images are presented one at a time. So here we have rooster, 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 toaster. And so based on this sequence, you might come to expect the rooster to appear because of its higher prevalence rather than the sort of rarer toaster image. And conversely, you might have the opposite, so toaster, 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 wait for it, rooster. And here we can compare evoked brain responses by the same images in each of these two contexts, labeled the standard and the deviant. And so there's been hundreds of ERP studies comparing these images, and they typically find more negative going evoked potentials over bilateral posterior sites for deviants compared to standards. And that's about the extent of where we agree in the field, and it's controversy from here on in. So there's been a very long-standing debate about what visual mismatch responses reflect, and actually what experimental manipulations are necessary to produce them. So for example, it could be prediction error signaling as specified in different predictive coding counts, novelty detection or detection of these unusual events, or something like repetition suppression or adaptation by feed-forward or intrinsic mechanisms, which could even sort of account or facilitate these other two things here. And I think the reason why this has been controversial is because different effects are intermixed within these sorts of designs. So the standard and the deviant are also common and rare, expected and surprising, and also repeated and immediately unrepeated images. And so to sort of boil this down into a series of small hypotheses or easily testable ones, we can think about what is driving this mismatch response or this ERP difference wave between the deviant and the standard stimuli. So for example, we might have a response enhancement for deviant stimuli without much of an effect on the expected stimuli here. We could have a response suppression for the standards, which are repeated and expected, without such effects on the deviants. And so we could think of this as repetition effects or expectation suppression, or we could have a mixture of both suppression and enhancement for the standards and the deviants, respectively. And so we've gotten partway to answering this question by including equal probable control sequences, whereby a large number of different images are presented within a sequence. So participants can't form expectations for any particular image, and none of them are immediately repeated. So by comparing the equal probable control and the deviant conditions, we can get effects of surprise fairly easily. However, effects of expectations seem to be still conflated with effects of stimulus repetition in these designs. And so what we wanted to do is to try to isolate and quantify effects of fulfilled expectations and surprise relative to some sort of expectation neutral condition in a cleaner way. And also just figure out whether we could find graded effects of expectation or surprise by stimulus appearance probability. And this speaks to some theories that would hypothesize that, the objective appearance probability determines how big these effects are. So using existing designs, this would have been very difficult because of the sheer numbers of trials and stimuli that you need to present. And when manipulating appearance probability, this gets even worse. So I took the advice of my grandmother, who said, if you walk faster, you get there faster. Accordingly, we used fast periodic visual stimulation, or FPVS, developed in Bruno's lab. And here's an example from Joanne Liu Shuang's seminal paper, where a base face image, similar to Tile's design, is presented at a very rapid rate, here at 6 times a second. And then every 7th face image is replaced by a face of a different identity to these base faces, known as the oddball. And here I'll specify that we're actually, in the experiment I'll present, you can have expected or surprising oddballs dependent on this context. And participants' task was to detect a fixation cross color change from blue to red. So in these sequences, briefly, people usually see a fixation cross, then the contrast ramps up over 2 seconds. We have a full contrast stimulation period, whereas the oddball and the base stimuli each appear at their respective periodicities, then a ramp down and a fixation cross period. And in these designs, you can manipulate the relative probabilities of each oddball that might appear in the sequence. So here's an example from an older study where we had the base face, and then the oddball could either be the same face identity as this base face or a different face identity. And the relative probability of these can be manipulated. And because these oddballs are periodic, this means that participants can come to expect specific oddball face identities at specific times. And one of the nice things about this is that basically by presenting a whole bunch of base phases between these, you can control for immediate stimulus repetition effects by continuously adapting the system to that base phase. So in the experiment we ran, we had a base phase and two different oddball identities from the base phase. And we manipulated the probability with which each phase could appear. So we had a common and rare, so a 90-10 split, 80-20 splits, 70-30, 60-40, and an equal 50-50 split condition. And these were presented, of course, in different stimulation sequences. And one important thing is that we use different base and oddball faces for each sequence type so that we didn't get carryover of expectations that were linked to these specific images. Okay, so there are a number of different outcomes we could expect. So we have the different probability conditions at the bottom, and then some sort of normalized amplitude on the y-axis. First, there was the null model, which we hoped wouldn't happen because that would be quite boring. Then we had the expectation only model, where expected stimuli had a dampened response compared to the 50% neutral and the surprising conditions. A graded version of this, where the stimulus probability sort of had a gradual proportional effect on this. A surprise-only condition where the neutral and expected conditions were quite similar, and there was only an effect of surprise, a graded version of this, and then, of course, a combination of expectation and surprise effects. So, what did we find? Looking at the grand average ERPs, we can see this sort of gradual, more negative going ERP waveform for the surprising compared to the neutral and the expected conditions. And we'll see this a bit clearly in a second. And here we can identify two time windows over which there are surprising expected differences. So between 200 and 350 milliseconds and between 500 and 1000 milliseconds. And by looking at the surprising expected differences, you can see these sort of difference waveforms quite clearly. And this is how we derived our regions of interest. So we compared surprising and expected, but then for subsequent analyses, we compared either surprising or expected to the neutral condition and never against each other. So we did this to avoid circularity in our analyses. And here you can see that the surprising has more negative going ERPs than neutral, but we don't see much of an effect of expectation here. This is also mirrored by the scout maps. So you can see that the neutral and expected are similar and the surprising ones are quite different. And for reference, here are these sort of individual condition maps, which also shows this quite clearly. Okay, so we then got a region of interest averaged, mean amplitudes. So to orient you again, here we have the individual probability conditions at the bottom, and then the amplitude difference to the neutral condition as the y-axis. So these are all compared to the 50-50 condition here. And what we found was that surprising stimuli evoked more negative going ERPs across these conditions compared to the neutral condition. Whereas we didn't really find this for expected stimuli. So if anything, the 60% was slightly more negative, and for the others, the base factors indicated evidence preferring the null. And this was also found for the later time window, and almost sort of, this came out a bit more clearly even here. So out of these three competing hypotheses, I think that we have a clear winner, that there is sort of effects, there are effects of surprise in particular, but not really effects of expectation suppression. And this might be surprising to you based on the literature. So we actually ran a review, did a review looking at expectation in the visual system, and we found that there's actually very scant or inconsistent evidence for this compared to neutral conditions, and especially almost absent effects in electrophysiological recordings as well. So how do we understand visual mismatch responses? To finish up, visual mismatch responses seem to index a surprise response, which seem to be shown all or none response, so no clear graded effects of stimulus probability. I didn't cover that, but this is what we found in our study. They seem to be gated by attention or the task relevance of a stimulus, at least in some studies here, cited here. And they're also larger following changes in one's visual environment compared to sort of unexpected repetitions of the same stimulus. And so we think that this might reflect something, a precursor to the orienting response, or something that might be necessary for things like post-surprise or post-error decision-making adjustments and things linked to reward learning and these sorts of things. We have a preprint, as was nicely posted, and our article was recently accepted for publication. To finish up, I'd very much like to thank my collaborators, especially Jane, who really sharpened up the experiment design and collected all of this data, but also Jen, Hintze, and Stefan, whose expertise really benefited this project at every step. Again, here are all of our Twitter handles, and thank you for listening.
Fantastic. Thank you, Daniel. And if you enjoyed the talk, please drop some praise in the chat because we don't have any applause here. So we have a couple questions here, and maybe we can do them relatively in quick succession. It looks like maybe the first one got answered already, but Mohammed, I'm actually going to give you a microphone and let you know, let you answer. Did your question get answered? Aren't repetition suppression and prediction error almost the same arguments?
Well, I think I probably missed it if it was answered.
Okay, great. Then we can do this question.
Oh, fantastic. This is one of my favorite ones. So yeah, there is the argument that repetition suppression and prediction error signaling are the same thing. I think this is something that's pushed a lot in certain veins of the predictive coding literature. But one of the things is that it actually relies on certain assumptions about when we observe expectation suppression and how this corresponds to repetition effects. A lot of papers that have purported to find expectation suppression actually have this stimulus repetition confound. So you have to almost assume that they are the same to find evidence that they are the same sort of the same thing. That said, there are more nuanced arguments. There are competing models of the predictive coding account, especially sort of Marta Garrido's work, which has this sort of very repetition-driven error signaling approach, which seems to hold water. There are also sort of circuit-based local and feed-forward accounts of these models, which actually predict quite a lot of the same thing as predictive coding. So there's a lot of model mimicry, but they can account for some other things, sort of inherited adaptation and other effects like that. So this is really an open question, but if you're wondering, do effects of repeating an image, are they the same as these probabilistic queuing sort of designs, or effects of entraining people's expectations to see a particular image? I'd say that the evidence says no, these are different effects.
Okay.
Hopefully that helped.
Great.
I guess I can clap since I'm unmuted. So.
Great. Fantastic. And Mohammed, I'm going to ask maybe Daniel to answer your other question in chat, because I think that it's a more pointed question. And then the last question that we have time for here is from Byron Price, who asks, what are the average time differences between oddball stimuli in the different conditions? For example, from one 10% oddball stimulus to the next versus the 90% oddball to the next.
So this is a fantastic question, and it speaks to the one shortcoming of our design. So the oddball intervals, if you like, were 1.16 seconds. So we had this just to have a reasonable number of base stimuli, but also to sort of try to keep these surprise-related ERPs away from the next stimulus as well. So I guess for the 90%, typically it was about 1.16 seconds. And for the 10%, it could have been 10 seconds or more. So here there is a sort of delayed repetition or sort of a recency confound, which we haven't fully accounted for here in our design. We did control for immediate repetition, but these sort of longer lag design effects are insidious and occur quite a lot in other designs which try to control for this as well. I think that I'm happy to talk about this more in the chat, but one thing I think this doesn't completely explain our findings is that we found the step sort of function difference between the surprising and the neutral conditions, which according to these sorts of delayed repetition models, I don't think they could really capture that or it's a little bit problematic for them to capture this pattern in our data specifically.
Okay, great. So I encourage you guys to continue the discussion in the Q&A box and in the chat. And with that, we'll thank Daniel again for the excellent talk and the good Q&A session. And we will go to our last speaker, who is Electra Schubert. And Electra is from the University of Melbourne, and she is going to tell us about decoding explicit and implicit representations of food, health, and taste attributes in the human brain. So whenever you're ready, Electra, take it away.
Thank you. Can everybody see that? Does that work? Yeah.
Yep, that looks good. And I'll ask you to speak up a little tiny bit because you're a little soft.
Okay, no worries. So hi everyone, thanks for tuning in. My talk is on decoding representations of food, health and taste attributes in the human brain. So as you're probably aware, there's a global obesity health crisis, and excess body weight has all kinds of other health problems that it's associated with, including heart disease, high blood pressure, type 2 diabetes, and various cancers. So part of the cause of this is poor food choices. So it all kind of comes down to this situation in the supermarket or the convenience store where we're faced with all kinds of food cues. So we might see a Kit Kat packet, we might see a popcorn packet, and it's important to understand how people process these food cues in order to understand how we make food choices and how we might improve these food choices. So when we make food choices, there are two main attributes that go into these decisions. The first of these is taste, which is a hedonic and fairly low-level representation. So it's thought to be integrated into decisions fairly quickly and automatically. The second attribute is health, which is a more abstract and high-level representation, and it's thought to be integrated into food choices more slowly. So because these attributes are often processed fairly quickly and automatically, it's useful to use neuroimaging methods such as EEG to understand how they occur. And a couple of studies have done this, including this one by Schwab et al. They looked at an ERP component called the late positive potential, which occurs around 450 to 1000 milliseconds after a stimulus or a food image occurs. And they found that it was more positive in amplitude when people viewed images of sweets and meat compared to when they viewed vegetables. So on the graph here, if we compare the red and blue lines, which were the sweets and meat, to the green lines, we can see that the more high calorie foods had a higher LPP amplitude associated with them. So they suggested that the LPP might reflect the processing of high versus low calorie foods. And in a recent study in our lab, we found kind of a similar thing. So we looked at differences in LPP amplitude between sugary drinks that people had rated as tasty compared to drinks they had rated as not so tasty. And similarly, we found that the LPP was higher in amplitude for the high taste drinks compared to the drinks that they rated as less tasty. So it seems like the LPP reflects the taste value or caloric value of foods and drinks. And it seems that we might have information at a very early stage in the brain about the attribute of taste, but it's not so clear whether people also process health quite as quickly. So that's why in our study, we decided to conduct 2 experiments to see whether it was possible to decode both of these attributes from brain activity during the first second of people viewing a food cue. So in experiment 1, we had 37 participants, and they viewed 174 images of foods from this FoodPix database. And here are some example images down here. We chose the images so that they varied a lot in both tastiness and healthiness. So there was representation from lots of different food categories. The task itself was quite simple. So this is basically how it worked. They would view a food image for 2 seconds, and then they would rate on this continuous sliding scale either how much they enjoy the taste of the food or how healthy they consider the food to be. And each image was presented twice, so once for each of the 2 questions, which resulted in 348 trials. And this occurred in 12 blocks. And within each of these 12 blocks, only one of the 2 questions was presented. So within one block, they would only be rating either how tasty or how healthy they considered each food to be. And during the task, EEG was recorded. And this is how we analyze the EEG data. So we used a technique called MVPA, which Tyle talked about in his talk already. But our approach differs a little bit in that we used support vector regression instead of classification. So this involves training an algorithm on a set of data to use these patterns that are distributed across multiple electrodes to predict, in our study, the health and taste ratings. And its performance is then tested on the remaining data. So I'll kind of walk you through what this image is describing here. So we had a 20 millisecond window that moved through the EEG data in overlapping steps of 10 milliseconds. And these windows were then transformed into spatiotemporal vectors. And we also had vectors of the taste and health ratings for each trial. We then trained an algorithm on 90% of the data to predict the taste and health ratings based on these vectors of the brain activity. And then its ability to predict the taste and health ratings was tested on the remaining 10% of the data. And then what this gives us is a graph that looks like this. So we have time on the x-axis, and then on the y-axis, we have the correlation between the real labels, so the real taste and health ratings, and the taste and health ratings that were predicted by the algorithm. And then the blue line here is the real decoding, so it was the algorithm's performance. And then the yellow line here is a chance distribution. So it's basically the same analysis that I described in the previous slide, but with all of the taste and health ratings randomly shuffled. So then to compare the classifier's performance to chance, a t-test is performed at each time point, and we have significant time points highlighted in purple, pink here. So we can see that for the taste decoding results, it was possible to decode taste based on the brain activity from a little bit after 500 milliseconds. And this seems to last as a fairly sustained representation up until almost 1000 milliseconds. So then we can compare that to the results for health, which showed a weaker and kind of later decoding onset. So decoding was possible from a little bit after 600 milliseconds instead, and it doesn't seem to be anywhere near as strong or sustained as the results for taste were in the previous slide. So based on this, we decided to reanalyze data from a previous study in our lab to look at whether these representations are still present or whether they're different when people are making dietary decisions and are not explicitly instructed to think about the taste and health of the images. So this study was already published by Rosenblatt et al. In 2018. There were 85 participants and they completed 2 phases. The first of these was a rating phase where they just rated the taste and health of a series of snack food images. And then in the decision phase, they viewed a subset of these same images and they rated how much they wanted to consume each food instead. And this is what each trial in the decision phase looked like. So they would view the food image for 3 seconds, and then they would rate on a sliding scale similar to the one used in experiment 1, how much they wanted to eat each food at the end of the experiment. And there were 50 trials in this stage, and it all occurred in one block, while EEG was again recorded. So our analysis was basically the exact same approach as the one that we used in experiment 1. So we have similar figures here. These are the results for taste. But interestingly, the taste decoding was a little bit later in this experiment compared to experiment 1. So we can see that significant decoding started from about 750 milliseconds, but this particular window didn't remain significant after corrections for multiple comparisons. So the significant decoding that we have is in this window from 1100 milliseconds, which is quite a bit later than the decoding from experiment 1. And then when we look at the health decoding results, on the other hand, it was possible to decode health fairly earlier, so from a little bit after 500 milliseconds, which is about the same time as taste was decodable in experiment 1. And I'll talk a little bit later about why this may have been the case. But we also did one additional analysis in experiment 2, which was decoding the decision strength, which was the ratings of how much people wanted to eat each food at the end of the experiment. And we can see here that we have significant decoding a little bit after 800 milliseconds, which if we look at all the 3 graphs together for this analysis, the decision decoding was a little bit later than the decoding of both of these attributes. So what we might be seeing here is the integration of taste, even though this time window wasn't significant after corrections for multiple comparisons, but we might be seeing the integration of taste and health into these decisions if they're firstly processed and then integrated into the decision. Okay, so what does all this mean? Firstly, we found evidence that information on both taste and health is activated in the brain during the first second of dietary decision making. So previous studies had already kind of found evidence that taste information was present, but we found evidence that people also process health at this early stage. And this was also possible even when people were not explicitly instructed to think about taste and health. So this might be saying something about the way people make decisions in real life or how they process food cues in real life. Now, as I mentioned, taste was decodable earlier than health in experiment 1, but then this was kind of reversed in experiment 2. So I don't know exactly why this was, but I've put a couple of speculations here. So firstly, in experiment 2, the stimuli had a lot of variation in health, but not a lot of variation in taste, because the images were all snack foods, which were fairly appetizing, even the healthy ones, which were images of fruits and things like that. So people rated all of the stimuli as highly tasty. So it might be that because they saw all the images as being a similar taste level, they gave health processing greater priority. Another possibility is that because participants had already been exposed to the stimuli in the rating stage, their automatic craving reactions, which reflect taste value, may not have been as strong by the time they got to the decision phase. Whereas in experiment 1, they were viewing all of the stimuli for the first time. So for future studies, we might use these results to determine whether interventions might alter either these temporal processing dynamics or the strength of these representations of taste and health, so such as nutrition labeling or health warning labels. And thank you everyone for listening, and thank you to everyone who's been involved in this project.
Fantastic. Thank you, Electra. And if you enjoyed the talk, please drop some praise in the chat, because we do not have any applause here. So we have time for a couple questions, which is great. The first question comes from Mohammed Abdelhak in the Q&A box, and he is asking, are the results in experiment 2 based on training from the data of experiment 1?
So that's a good question. No, the 2 experiments were completely independent of each other. So the training was based on data from experiment 2 completely.
Great, thanks. And there was also, Tyler is putting something in the chat here. Us panelists can't use that Q&A box. So Tyler is saying there's probably heaps of between subject and between trial variance at the later time points in the epoch. You might get better predictions if you can somehow realign the time points.
I think that's a really good suggestion. Yeah, I'm not really sure how to do that, but if it was possible to realign the time points, that would be really good. So maybe I'll talk to my supervisor about that, I guess. Thanks, Kyle.
Great. And you guys can also talk afterwards. And can I ask a very, very quick question before we close up? Unless there's another one that's coming from the audience, but it looks like you're training 3 different classifiers in experiment 2 or in study 2. Have you thought about taking the classifiers for taste and for health and applying them to the desirability to consume data and seeing which one might be able to kind of predict better, or when they can predict better, and how that might influence those choices?
No, I haven't done that, but I think that would be a really good idea. Maybe something to do in the future. We're still working on getting this paper written up, so it's not all finalized yet.
Sure.
That's a great.
Cool. Yeah, like a cross-decoding approach. That might be kind of interesting and informative. Great. Well, if there are no more questions for Electra and for any of our panelists, we will go ahead and close up. But if the rest of my panelists might turn on their cameras briefly so we can wave, that might be nice. And then I'll also encourage you guys to meet up with your NeuroMatch matches, or if you guys want to talk offline, there's always ways to connect. So thank you very much again to all 3 speakers for today's session. And we will see you next time in the next NeuroMatch session. Thanks.
Thanks
Bye, guys.
for hosting. Bye, everyone.
Thanks, everybody.